Okay, great. If you could all find your way to your seats, we're going to get started. So my name is Kate Hartley. I'm a professor of psychology at New York University. I also did my PhD with Liz Phelps at New York University, which makes me Joe's academic grandchild. And I had the pleasure of helping to organize this event today. In the interest, so I'm going to chair the first session and try and keep things running on time. In the interest of time, I'm not going to take another minute, but I'd like to turn it over to Mike Gazzaniga, who is going to be our first speaker. And I'm not going to introduce the subsequent speakers, so if you could each, as you come up, introduce yourselves, that will help us save time. Thanks, Mike.
So do I hit this button?
Yes, hit the space bar to advance.
Oh, there we go. All right, well, thank you, and great to be here and tell you my side of the Joe LeDoux story. So, no results. The first thing, this sounds like the old days. Everything has disappeared. Where did it all go? So to advance the slide, I'll use that. Yeah, there we go. We're here obviously to celebrate Joe and his work and the body of his work that a lot of the general area of neuroscience thinks of as his intricate work and the animal models of really bringing a serious science to the study of emotion. And he has done that with gusto and brilliance. But I'm not going to talk about that because a lot of people are. I'm going to talk about where Joe started, which is this trailer. And that was his first lab. And that's the family car pulling it up to the New England area where we were lucky enough to be studying the first set of East Coast split brain patients. And what we had done was we converted that trailer to a lab, and it had slide projectors, kistoscopes, recording materials, and the like. And we would drive that trailer to the patient's trailer because a lot of these patients lived in trailers in the New England area and were served by the Dartmouth Medical Center. And it was there that we found our first real split brain patient in the East Coast, and it was case PS. And a big part of those studies was Joe. And I wanted to show you just two short clips of how exciting it was for us to discover Joe, for Joe and I to discover case PS. This is case PS being flashed. We don't have to get into the technical parts of it, but being flashed a question to his disconnected right hemisphere. Right hemisphere couldn't talk, only the left. And we asked him, we flashed the question to the right hemisphere of who are you? And here he answers the question.
What's it got?
Is there a sound? So he's taking from a pot full of Scrabble letters and spells out his name, Paul. So here we have.
What was the question?
Who is?
Huh?
Who is what?
Didn't
What
get
was
it.
the word that was flashed?
I didn't get it.
And then we went on. So there's the right hemisphere answering that he is Paul, and the left hemisphere doesn't seem to know about it. Well, no, not
I'm
that.
going to ask you another one of these questions about what's your favorite, and you see the word flash, all right?
My favorite guy?
And you spell it out for us.
All right, my favorite
Ready?
guy, yeah.
And look right at the black dot.
And here he is answering
What's
the
your
question, who's his girlfriend, basically. And you will see a young Joe LeDoux here in a minute.
Good.
Oh, there he is.
What was the question, Paul?
What's your favorite girl?
Think that's what it was?
No.
What was it?
So there was Joe. The whole point of this story is that Joe started
did you spell
his
Liz?
career studying these special patients that have taught us so much. And
Joey,
this
he knows.
is where he became fascinated with
I
emotional
have
questions
fortified
and
too.
how they get
My
propagated
sister-in-law.
into
Her.
our cognitive life. And we went on
Get your brother
to
bring his ass
write a
up here
book
one of these
about it
days. Wait for
that
it.
resulted in this book, The Integrated Mind, and it was a real signature time, and Joe was at the center of it. And so we moved on to New York, and, and other than having a lot of fun, that's when Joe decided to go head on into studying emotions with animal models. But he left us a gift at the end. I had to put in for a grant, as we all do, and we had our eye on something better than the trailer. And so I said, why don't we get one of those fancy GMC homes? So we put in an NSF, and Joe, I asked Joe, would you write the budget justification? You know, you've been through the whole thing. And so he did, and 9 months later, you get that call from the NSF program officer, says, well, there's good news and there's bad news. And I said, what's the bad news? He didn't get the grant. I said, well, what's the good news? You did get the van. He said the review committee sounded like it was time with travels with Charlie, and they just thought it was the greatest idea in the world. And so we've always been in debt for Joe. But let me just conclude with this little story which connects with Joe. This is Amanda Gorman, as you all know, the poet laureate who spoke at President Biden's inauguration, and this fantastic talent out of Harvard. And Oprah, of course, immediately got onto it and wanted to know more about her and who mentored that person, who mentored this young talent. And it turns out it was my daughter, Zaz, who Joe used to babysit. And so there's some connection there. But anyway, I asked Zaz, I asked, Oprah asked me, so what was the trick? And she says, how do you mentor young talent? And she said, just stay out of their way. And that's exactly what my feeling is that we all knew about with Joe. Just stay out of his way, and you'll be in good shape. And I now am going to stay out of your way so the show can go on. Thank you.
Not yet. Okay, not yet. Good. Yeah, Joe has a few words for us.
All right, thank you, Mike. I have to thank a lot of people before we get started here. There are just so many of you that have contributed something. But I want to start first with my family. I would not be here, standing here talking to you without them. Nancy, you've been amazingly supportive as a partner and friend. Milo, you're a brilliant and thoughtful son, and we're incredibly proud of you. Anna, you're not here, but I think you'll be here later. We're happy to have you in our family. And Jacob, we miss you every moment of every day. Mike, you come next. An amazing cascade of falling dominoes led me from the bayous of Louisiana and degrees in marketing to you. You treated me as an adult and told me to think big. Thank you for your support and friendship for the past 50 years. It's hard to believe it's really been 50 years. You know, there's another part to the GMC story was I ended up living in that thing for a while. I broke up with a girlfriend, and the GMC was parked next to the lab I was working in, so Mike let me live in the GMC in New York City. It was like fun. So next, I just want to thank the 6 incredible women who put this together. I don't know where the men were, but that's the difference. You cannot imagine how much time they put into this. It's a big job. I did some of the work getting this place organized, so I know how big it is, but they did the really big job. So Liz Phelps, Daniela Schiller, Kate Hartley, Liz Romansky, Jackie Reitzes, and Claudia Ford. The two Liz's go back to the early days at NYU, where Liz Romansky was my first graduate student and Liz Phelps one of my first postdocs. Thanks to each and every one of you for all you've done for this thing. Yeah. So 3 amazing people helped me run my lab. Claudia's been with me since 1987. Mian Hu who's here, 1995, he joined us, and Will Chang since the early 2000s. I couldn't have really done it without them. You guys were amazing all these years, and thank you for still being here. So the lab survived and thrived with generous support from NIH, especially NIDA and NIMH. But we've also been supported by private donors, especially Jennifer Brout and Tim Somner of Knockout Noise, who not only contributed to my research, but also produced the Amygdaloid's second album. And they also produced a documentary about me on Amazon, directed by Kurt Feldhun, who is going to be around. Kurt, are you here right now? Okay, he'll be around some point. And in recent years, the lab has been funded by Bill Slater's Vulnerable Brain Project. Bill has become a very close friend and is a really terrific guy. Major thanks also goes to all the researchers who contributed to all the work we did over the decades. Many of you are here today, but whether you are here or not, thank you for being partners in the adventure known as the LeDoux Lab. Thanks to all of you who traveled here to speak, to those who could not come but sent videos, and to those who contributed to the special issue of the Cerebral Cortex magazine, where some things were said. Thanks also to all the staff here at 370 J Street, especially the Media Commons and the Clive David Institute. And last but not least, thanks to Tyler Volk and Daniela Schiller, who with me formed the Amygdaloids in 2006. And to others who have played with us, including Amanda Thorpe and Colin Dempsey, both of who have spent many years with us and continue to contribute. Both are here now. And at the end of the day, we'll get together on the stage here and play some tunes from our trademark genre, heavy mental. But I have to say one thing before closing. Amygdaloids, out of the blue, you've changed the nature and quality of my life. Thank you. I'm going to clap for you. One final note, this is a kind of PR thing. Werner Herzog has a new film called Theater of Thought. It can be seen at the Film Forum starting Friday. I'm in it, and we'll do a Q&A on Saturday at the 5:30 screening. But most important, 3 of my songs, Amygdaloid songs, are in it. And you'll hear some of those a little bit later. So it's a tight schedule, so let's move on. Thank you.
Catch up some time if I can. Which one is? Is it this one? I mean, I can get this out if I have to. Okay. All right. So, such an honor to be here, and I want to thank everybody who made the trip. It's really been remarkable to see how all of you have honored Joe today, and many of you coming from other places overseas. So, thank you. So, I'm going to say a little bit about my science and career with Joe. And one question, when I was thinking back, like, how the hell did I get here? I have been collaborating with Joe for pretty much my entire career, which I never, ever actually intended to do when I first contacted Joe. But I contacted Joe when I was about 28. Here I am, about that age. I don't know how old you were here, Joe, but I figured you looked kind of like that back in the day. Yeah. Yeah. Okay, so maybe a little bit after that, but beautiful picture with Nancy. So I contacted Joe then to, I was at this stage in my career. It was around 1990. I'd received my PhD from Princeton. Mike Gazzaniga offered me a great postdoc. And I was living in New York my last few years of grad school, and I was a little worried about moving to Dartmouth. And Mike said, try it for 6 months. If you hate living here, you can leave. So I did. I tried it for 6 months. I hated it, and I left. And I was kind of unemployed, but Mike graciously let me continue to do some research, kind of on a part-time basis. So we actually got a few papers out, and he couldn't have been more wonderful about it. Mike, more than anybody, appreciates that life's too short. But I was, trying to figure out what to do with my life, and I thought, if I'm going to stay in this field, I really need to think about what matters to me. And I realized I really wanted to dig deeper in the neuroscience. I really wanted to expand my memory research to the things that I think were important in memory, which was particularly emotion. Luckily for me, the McDonald Foundation around the time was funding postdocs to enable sort of cross-disciplinary training in cognitive neuroscience. I decided to write one to study emotional memory, and the amygdala. I approached Joe. He said, if I get the grant, I can come, and on my second try, I did. So that's how I ended up with Joe. And we've been working together ever since. So my first project, we proposed to look at the role of the amygdala in 2 tasks. Joe knew about this paper of mere exposure in rats, and we thought we can take this study that's been done in humans, mere exposure, this idea that if you see things more, you like them, or you hear them more, you like them. So we would do that in rats and humans with amygdala damage. And then we also would do threat conditioning in humans with amygdala damage. And there were some studies of threat conditioning that we used as a sort of jumping off point. So in 1991, I got to the lab. I took all the neuroscience graduate classes to get my training. I discovered studying the amygdala was much harder than studying amnesia. There weren't that many patients around that didn't have memory problems. There was no fMRI. There was only one documented case of a patient with amygdala damage at that time. So I started with the rat study. I just decided, what the heck, I'll apply for 3 faculty jobs. I did get 1 interview. This was the only study of a patient with amygdala damage at the time, just looking at skin conductance. So I started with the rat study, which was rather interesting, based on this sort of crazy study from 1967, where they played rats either, hold on, my mouse is now not working here. Let me just see if this works. Huh? But I can't know. It's not letting me play. That's okay. I don't need to. They played rats either Mozart or Schoenberg. I don't know if you know Schoenberg. I have a little clip here, but I can't play it. But Schoenberg's like this weird composer that goes, I don't know. I can't even imitate it. And the rats that, and this was Joe pointing me in this direction. The rats that heard Schoenberg didn't develop a preference, but the rats who heard Mozart did, not surprisingly. So we decided to try this with tones. And guess what? No mere exposure effect in rats. It really isn't that good in humans either. I learned a lot of neuroscience. I got a job at Yale. I joined the faculty. So basically, I got nothing done but learning neuroscience. Joe still wanted to work with me after that, in spite of the fact that I did nothing. And the McDonald Foundation let me use the funds I had with Joe to take to Yale. And Kevin LeBar, here's a young Kevin LeBar. Is Kevin here? Hi, Kevin. Joins the project. So Kevin really made this all come together. He develops this great fear conditioning paradigm. The Yale neurosurgeons helped me identify patients with amygdala damage, and we were able to do the study, and lo and behold, we found impaired fear conditioning in patients with amygdala damage. Soon after that, we identified a rare bilateral patient. There's Kevin testing her, and also showed that she showed this impairment. So this was really our first success. Soon after that, imaging came along. Yale was a leader in this technology, and we could further our work. So this was just the start of my career working with Joe. In 1999, I left Yale. I joined the faculty at NYU, and here our collaboration really took off. Here's our most highly cited research publication, 23,000 as of 2,300 as of this morning. Here's our most highly cited review paper. But that's really just the tip of the iceberg. So far, we have 21 publications. The most recent was this year. We've got one that's under review. Actually, Joe, it was rejected, but just recently, with maybe a revision. We'll see. Sorry, I didn't tell you that. All right. So 30 years later, I'm still working with Joe. Never intended. I thought I'd do that one fear conditioning study and then I'd be done. But you can see we've changed a little bit in all those years. And now I'm thinking, what is my post-LeDoux career going to look like? Because I've never actually had a career without Joe LeDoux. And then I realized when I really thought about it, there's no such thing. We may not still collaborate, he may not have an active research lab, but its influence is part of all of my science. So sorry. His ability to dig deep into his research and be precise while also constantly evaluating his approach and pivoting when he needs to, and at the same time always coming back to the big picture are lessons that have shaped my science and will continue to shape my science. So I don't think I'm ever going to have a post-LeDoux period. And I am forever grateful, Joe. So thank you for everything.
I read very quickly, and I left out an important thing. I want to thank the faculty of Arts and Sciences and the neuroscience in particular for supporting the LDU. It's been a great place to work. So I do that.
So I'm really honored to be here. I'm Kerry Ressler. I'm at McLean and Harvard now after being at Emory for 18 years. First, thanks to Liz and Jackie and everyone for letting me speak in the first section because I'm heading out shortly to ACMP in Phoenix. That starts tonight. And then Liz set me up a little bit because I can't follow that. And I said, Liz, what should I talk about? She said, whatever you want. So I'm going to throw in some stories. Don't pay attention. There's some graphs in science, but it's really to facilitate the story. So apologies in advance for that. I think the main reason I'm here is because I think of the people here, I probably grew up closest to where Joe grew up in Eunice, Louisiana. So Eunice is sort of right in here, right, Joe? And I grew up in Ocean Springs, Mississippi, which is right here. So I lived in an apartment on a bayou on the Mississippi Gulf Coast, which is sort of part of the overall bayou system. So with that, from having a lot of Cajun influence, how y'all are, as Justin Wilson would say, and we love. So I first heard about Joe as I did my graduate work at Harvard and was trained in the molecular biology of the olfactory system. I'd never planned on working on sensory. I really wanted to work on learning and memory and emotion, and there was nobody there doing it. So I had really good timing to be able to be involved in the early olfactory work. And I went to Emory for a number of reasons. They had great psychiatric biology, but then heard about this person, Mike Davis, who had just moved down from Yale. So as opposed to being officially scientifically related to Joe, I am, I guess, sort of a cousin. So I think everybody here, all the scientists know, but the people who are not in the field may, for most of the decades of the amygdala, it was the LeDoux family, the Fanzelo family, and the Davis family. So I'm sort of representing with Sheena and maybe a couple others the Davis side of the amygdala lineage. So I was trying to figure out how does one become a physician, scientist, psychiatrist, and actually study anything worthwhile. An oncologist, you can study, the cancer in a dish, but how can you do that in psychiatry? And then I heard about the amygdala story and threat and fear, and I was just blown away. So that's what I've been doing for the last 25 or 30 years is studying post-traumatic stress disorder. So I thought what I would do is tell just a little bit about my story. Has been so influenced by the work of Joe and all of his colleagues, and how exciting it is that we're almost, I think, to a neurobiology of PTSD that really comes from all of this groundbreaking basic work. So the work that we've been focused on for a long time is that post-traumatic stress disorder is tractable. It has a shared neural circuitry of threat and fear, which of course comes from all of the amygdala work. We know when it starts. It's one of the few disorders in psychiatry where we know when it starts that can really be well modeled by threat conditioning in terms of the trauma experience. And of course it involved learning and memory, which so much of which the work from the LeDoux lab has done. So our focus has been to identify a molecular neurobiology of PTSD based on the groundbreaking basic science to really have novel therapeutics and interventions. So everybody knows this graph, and it along with the Scientific American story here, and then when I read Emotional Brain, so the books that most inspired me as a young scientist were Oliver Sacks and The Emotional Brain and Synaptic Self and Joe's various works over the years. I think they were just some of the most beautiful ways of describing the problem in a really precise scientific way while also being very accessible to the lay public. And so we really, initially the high road and the low road, and we've done a lot of stuff in the sensory systems, but again, being able to focus on the amygdala as sort of this hub reflex that really could be dysregulated in our fear disorders was just a very powerful insight for which, as we all know, hundreds of labs have been focused on. In neurology, really the concept of a reflex, whether it's the patellar reflex or others, led to so many insights. And similarly, the concept of amygdala activation, be it chemical, electrical, or otherwise, leads to these hardwired symptoms that essentially, if we look at the hardwired symptoms from the work of the neuroanatomy from LeDoux, Fanzelo, Davis, and others, really the hardwired downstream subcortical areas lead to all the symptoms of a panic attack and all the symptoms with a triggered response of like PTSD and trauma. And that's a remarkably powerful thing. It's probably one of the few, if not only, places in psychiatry and psychology where we can explain a whole set of symptoms that people experience and feel like they're going crazy. And it's no, it's this. It's your body trying to keep you alive through this mechanism. So when I was in Mike's lab as a fellow, as a resident and a fellow, we were reading all of the work from this lab, and I was particularly interested in learning and memory. One of the first most salient memories of that time was when the reconsolidation with Khmer came out. I think we got to see it a little bit early. I think Mike was actually a reviewer. I don't know if you ever knew that. But obviously, I'm sure somebody today will talk about reconsolidation and the amazing stuff there. But to me, one of the earliest important findings was this idea that long-term potentiation, this basic physiology mechanism that had been long argued to be a measure of plasticity in learning, could really be identified in vivo in the amygdala in rodents with threat learning. And building on other work of consolidation, Kandel and McGaw and others with Mike, we started to work to really identify large sets of synaptic plasticity genes in the amygdala during threat consolidation to say what are the processes, and there's Eric and many others here who've done work in parallel lines with starting with Joe for decades, but really identifying short-term, long-term, and long-lasting memories with the basic idea of if we could understand consolidation of threat, might we be able to inhibit that initial consolidation in the aftermath of trauma and prevent PTSD, or if we could understand the consolidation of extinction, could we enhance extinction? And LTP and NMDA work, and Stefan Hoffman and I had collaborations over the years with D-cycloserine and other NMDA agonists to expand that work. So again, the idea that everybody knows here is that consolidation, that memories aren't permanent immediately, but they become permanent over time. And so in the human world, we were able to start studying this in a prospective way in emergency departments. I'm talking to BJ Casey about the ABCD cohort. We started a cohort called the Aurora study that enrolled about 5,000 people over 20 emergency rooms with the idea of can we get very early biological data of people right after they've been traumatized and follow them up for a year to really understand the biological markers of long-term threat or fear disorders like post-traumatic stress disorder, whether you're haunted by this ongoing memory. And we were able to see very robustly that fearful, looking at fearful faces, which we know is an amygdala probe, was a very robust predictor, even a couple weeks after trauma, of PTSD 3 months and 12 months later. So this early sensor activation of this circuit was a strong predictor of later PTSD. And other measures of sympathetic activation and hyperarousal, such as galvanic skin response, right after trauma. So in this case, people would be in the emergency department getting their spine cleared, and you'd ask them what brought them into the ED, and that question and the electrodermal response was this very robust predictor of PTSD 6 or 12 months later. And this field is exciting now because we've been able over the last 20 years to start to bring in large-scale human genetics. So the Psychiatric Genomic Consortia now has over 1 million people worldwide, and this has been led by Kirsten Conan and Caroline Nevlegelt and myself and others. We now have over 100 loci that are very robustly associated with PTSD, and we're starting to be able to map those in the brain circuits. And of course, one of those that came up early on, and one of the jokes in the field is that it took schizophrenia 150 loci to get to the dopamine receptor, and and PTSD got to the CRH receptor in about the first 10. So there's something to this stress circuit that we've all been looking at. And as we look at this, there's a number of genes that are starting to come up that are related to glutamate synaptic plasticity and NMDA plasticity, GABAergic regulation, but also stress and HPA regulation. And with more recent work in postmortem brain, so we're able to start working with the Labor Institute and others to get large-scale data sets of PTSD, depression, and control brains to be able to do single-cell sequencing, multiomics, and really start to get at the underlying biology of both the human amygdala and the mouse or rodent amygdala, and are again seeing many interesting genes, including the CRH pathway and FKBP5 pathways, which are associated with, again, cortisol and stress, classic stress pathways. So a lot of the old theories from decades ago from the LeDoux and Fanzelo and Davis and others about how these systems are regulated are holding up in these disorders. And a lot of work over the years has shown, that we can mechanistically show that hyperactivating these systems increase fear responses, increase startle responses, increase freezing with threat conditioning, and deficits in fear extinction. And so where we've gone over the last decade or so is from the problem of plasticity alone is not always good. And so what we saw with the decycloserine story was it looked like if you enhanced plasticity for extinction, that was great if the person had good within-session extinction and safety learning. But if they had avoidance or bad within-session extinction, they actually, what looks like, probably had reconsolidation. So the old terms in psychology were, that patients would be resensitized to fear or versus habituated. But if you reactivate, that both of those processes can be LTP and NMDA dependent. And so the thought was that if you don't do a good enough extinction, you are likely reactivating reconsolidation. So a lot of what we're probably seeing with therapy is sort of a constant push-pull of are you reconsolidating and strengthening the memory, or are you weakening it or enhancing inhibition of the memory with extinction. And so the idea that we've been focused on, that a lot of labs have been focused on obviously, is can we separate the valence of positive versus negative learning? And Andrew, oops, I don't know what I did. Andre Sluthy, help, I went some way and it went away. Anyway, there are fear off and fear on neurons that project differentially. And the take-home message is that as a field, many folks, Kay Tai's been doing this, Tony Gawa's lab did some, I think many in the lab here have been doing it in different ways, can start to really identify the molecular identities of these different microcircuits so that we could really target plasticity ideally within, say, extinction-driven pathway versus a fear or threat-specific pathway. And in doing that, can we identify novel pharmacology? So this is one set of cells that are pyramidal neurons in the BLA, but as opposed to the traditional canonical pathway of coming from the lateral BLA to the central, these primarily project not to the central, but to the accumbens and the infralimbic, preferentially to the prelimbic. And when you activate them, you block the lateral amygdala flow through, and you importantly block fear or threat consolidation, enhance extinction consolidation. And if you identify receptors that are specific to these, you can start to come up with novel pharmacology, in this case a neurotensin receptor that specifically, it looks like, blocks fear or threat or enhances extinction in a valence-specific plasticity way. So with that, I think understanding the amygdala and understanding these threat pathways is starting to lead to a whole new pharmacology of amygdala-based targeting that we hope within the next decade can really lead us to much more precise precision treatments for interventions and preventions of fear, threat, and PTSD. So with that, thank you, Joe, for all you've done for the whole field, including the cousins from the Fanzelo and Davis lineages, and most importantly, for both just contributing to an amazing science, an amazing, probably I think one of the best stories in the history of understanding the brain, and that also is going to have very clear translational implications for patients. Thank you.
Great. So I just want to say what an honor it is to be here honoring Joe and to be with so many just amazing colleagues. It really does feel great to be up here and look out at all of you too. And I look forward to cocktails later, which I keep thinking I should have with my band on here. So I'm going to talk about just a few studies that were really driven in changing the way that I was thinking about my own research based on Joe's foundational discoveries. And so I'll be talking about threat responses from a developmental lens. And I'd just like to begin by saying the way that I think about development and younger ages when they perform tasks is not as deficits if they don't perform as well as adults, but more in terms of how they're adaptive and how has the brain evolved so that we can most effectively respond to the demands and challenges of that time. And I would say adolescence is a very demanding and challenging time as we learn to negotiate our world, in particular our social and emotional world, without the buffer and protection of our caregiver. I've been fascinated with this period, particularly in terms of how you get almost opposing characterizations of it with regard to fear and threat, particularly. So often adolescents are described as reckless and fearless. But it's also the case on the other side that we really focus in and talk about adolescence as being incredibly anxious and incredibly stressed. And of course, this is supported because we do see an increase in the anxiety and stress-related disorders during this time, and they've been increasing even before the pandemic. And so, in trying to think about threat responses during this period of adolescence, I've really built on the foundations of some of Joe's discoveries in adult animals and translating that to developing ones. And the animals that I'll be talking about today are humans and mice. And yes, that is my 13-year-old flipping me off. Okay. All right, so our early studies where where it was very important that I was picking up from Joe, let me just talk about the first ones. And that was using very simple stimuli, where over a lifetime we learn to predict that there may be danger or uncertain danger around us. So just these simple static images of a fearful face and trying to understand how different age groups respond to these when they're presented repeatedly. And so what we discovered early on, and other groups too, is that there was an inflection during adolescence with this heightened response to these cues of potential threat that we didn't see in younger ages or in the adults. And it wasn't simply the magnitude of activity, it was actually the pattern, the temporal pattern that was also changing in these young individuals. So let me just show you a really simplistic illustration of this. We would see bilateral activation of the amygdala with repeated presentation of these cues, but eventually nothing's happening. And so adults are like, get over yourself, and you see the activity revert to baseline with repeated presentations. And in adolescence, we see a similar pattern of activation. So bilateral activation of the amygdala with repeated presentations that goes on and on and on. And so what we were seeing more of in adolescence responses to threats was actually this more sustained response to this repeated exposure to threat cues. And I mention this because individuals who show this continued response also endorse increasing symptoms, more symptoms of anxiety. But there are problems with using simple stimuli like this, particularly when you're asking developmental questions, because as adults, we have more experience with facial expressions and fearful faces than, say, adolescents who have more experience than children with these cues, which moved us to thinking about taking a neutral cue and pairing it with something aversive, with the Pavlovian conditioning. And also because I think of development as adaptive and how the brain has evolved to really respond to challenges. Not only did I want to use this paradigm in humans, but also I wanted to collaborate much like Joe and Liz have. And so in collaboration with Francis Lee, we were also looking at the development in the mouse. And the two different conditioning paradigms that I'll just highlight today are cued and contextual threat. And the cued conditioning for this crowd, simply pairing the shock and tone a few times, then 24 hours later you can test for learning, but you can also just present the tone alone for extinction. And in the human, building on the classic work by Liz Phelps, using these yellow and blue squares that we pair with an aversive stimulus, while she used shock, we just used a really aversive sound since we're working with developmental populations. And then in this work, we wanted to see how well individuals across childhood, adolescence, and adults, how well they acquired the threat learning, and also we wanted to look at extinction. And so you can see that children, adolescents, and adults can easily associate something very aversive with a cue. It's like touching a hot stove. But the findings that were a bit surprising was that the adolescent had a difficulty once forming a threat memory, extinguishing that threat memory. And this is a finding that had been reported by Rick Richardson a year earlier. We were just waiting for our human data to see if it actually translated to the humans as well. This finding is a bit reminiscent of when I was showing you those fearful expressions and with repeated presentations, seeing that the adolescents were still showing this stickiness to it. So what about contextual threat? And this was work I'll be describing that was driven by Siobhan Patwell and Kevin Bath while in Francis's lab. And here, first, we wanted to look at acquisition of contextual threat. And we were very surprised to see that while younger animals and older animals, when put back in a conditioning chamber where they had been shocked the day before, when they were placed in it the next day, the adolescents look fearless. They showed very little evidence of this contextual threat response. We were scratching our heads about this, thinking, are they just not learning? Is it the case that they're stupid? I mean, I hate to say that, but often when we talk about adolescents, it's like, and we're, as parents, we go, you want to say, are you stupid? No. So anyway, it's not the case that they're stupid, and it's not the case that they're not learning. In fact, these same animals who, in this, what we're calling the equivalent of adolescence, when they were tested as adults, they actually show the fear response. So they're learning it and maintaining it, they're just not expressing it during this time. And so these findings from just very basic Pavlovian conditioning paradigms are helping us begin to inform us about basic processes that appear to explain sometimes a fearlessness and sometimes a fearfulness. And my question is, why would those opposing behaviors be adaptive for an adolescent? And I've speculated in a number of ways, as have others. We actually think the sustained vigilance, once you do detect a potential cue of threat, maintaining that long term during this period of adolescence where you're exploring, searching for new resources, you need to be very attentive to predators or other cues of potential threat for your own survival. And then in terms of the contextual fear, not showing that threat response, if when exploring, something bad happens, you wouldn't want that to prevent you from continuing to explore, because the whole purpose of adolescence is moving from dependence on the caregiver to relative independence. And so being able to secure your own resources, water, food, and also for the species, finding a mate as well. And then the reemergence, or the emergence expression of the contextual memory when the animal is reaching sexual maturity may actually help them in terms of thinking about where they're going to build their nest or home. Now, I've been talking about development as adaptive, but there are some where the changes that we're seeing in the brain based on their biology, genetics, or experiences might actually increase their risk during adolescent development. So a question we've had is, how may we attenuate threat-related responses in these adolescents? And there are a number of people in this audience who have been trying to do that in adults and in children and in clinical populations, but I just want to highlight one of Joe's studies that was carried out by Marie, and Carrie's already mentioned this, and that was of showing that if you extinguish a threat memory within the boundaries of reconsolidation, that you can get persistent attenuation of that fear memory. So using a simple retrieval extinction paradigm, where you retrieve or have a reminder cue, a delay, and then in that sweet spot, administer the extinction. And then you don't see the renewal that you would had you just done simple extinction. And then, of course, as Liz introduced today, the beautiful work of Daniela Schiller and Liz Phelps, translating this to the human using a similar paradigm and being able to show that using this reconsolidation update mechanism, that the conditioned stimulus that's manipulated in this condition shows a very similar response as to the cue that was never paired with an aversive stimulus, right? So really showing like the prevention and the return of this threat response. So what we wanted to do is to see, given that adolescents show diminished cued extinction, we wanted to see if we could use extinction during memory consolidation to attenuate that threat memory in adolescence. And so using very similar procedures across a 3-day experimental design, what we found is we replicated Daniela Schiller's results, but we also showed in those adolescents who have sticky fear, difficult to extinguish, they too showed reduction in that response. So this is just a teaser, and I wanted to highlight the special issue of Cerebral Cortex that's honoring Joe LeDoux, and in saying that the really interesting work that I wanted to discuss is really about the circuitry, because I'm really trying to get lay people and some of my colleagues to move away from saying adolescents do what they do because they have an immature prefrontal cortex. It's so much more complex from that and so dynamic. And then also in this paper, besides describing some of the circuitry changes that also explain the opposing threat responses I discussed, we also talk about the implications and, importantly, the limitations for optimizing treatments based on preclinical studies for thinking about treating anxious adolescents. So Joe, thank you. Thank you for inspiring so many people's work. Thank you for bringing us all together today. And really, I just want to thank everybody that put this event together. I can't imagine how much work and time it was. Thanks so much.
So I want to follow BJ and just say what an honor it is to be here, and particularly not just as a person who studies children, but as a person who's really primarily a clinician. I think it speaks a lot to your impact, Joe. I was really lucky to have spent the first 10 years of my training in New York, and really enjoyed interacting with basic scientists and spent a lot of time with basic scientists. And I'll talk a little bit about how my relationship with Joe developed in that context, but it's really been truly unique, and it has profoundly not just shaped me, but an entire generation of clinicians. And I'm going to speak a little bit about that, Joe's lessons specifically for clinicians. So I'll begin by talking a little bit about how I see those lessons, and then I'll end by talking about, much like BJ, how some of Joe's ideas really heavily influence the way we think about development in patients. Somebody who, yeah, there's something, a thing came up. So you'll see in the next one, it'll say, oh, you see it. I can't do that. There you go. All right. All right. So when I began my training, I was at Columbia, and I'm sure you remember this, Joe. I spent a lot of time with Joe. There would be a large group of us, and just like Carrie said, I very much remember the Davis camp and the LeDoux camp. And being in New York, we would spend a lot of time with Joe. And right from the beginning, I have to say it was wonderful to watch the films of Joe as early in his career, because you can really see the roots of somebody who cared very much about patients. And we would spend a lot of time with Joe talking about patients. And I like Carrie said, I read Joe's books. And I will say I was a lot younger then. This is probably 30 years ago. And I was very much a skeptic. And I don't know if you realize this, but I would always kind of be grumbling under my breath, you know, what the hell does this guy know about, you know, and I would do that a lot. And I don't know if you remember this, but we were at the ACMP. It wasn't 20 years ago. It was between 10 and 15 years ago. And Joe was getting off an elevator, and I was getting on the elevator, and it was just the two of us. And he kind of said hi to me in the quiet way that Joe often is. And I kind of grumbled, you know, something like that. And I have to say, I've never had an experience like this. Joe said, you know, what did you say? Really wanted to know what I said. And we started a conversation right there. And I just kind of let it go about, you know, how I thought aspects of his ideas were wrong and that he was leaving out this or that or whatever. And unlike most of the basic scientists that I have spoken with, Joe really embraced that. And he really wanted to know, well, how am I wrong? Tell me more. And it really began a very deep relationship that has grown over the last 15 years that really shows how much Joe cares and how much he wants to learn. It kind of culminated in this paper, but I will say in many ways, this paper just kind of describes the discussions that Joe and I had from around 2008 until when this was published in 2016. And the ideas really focus on how heterogeneous the response to threats are, and how we can break these into different components based on evolutionary principles and based on developmental principles. And it's something that's really shaped my work for the last 15 years. And Joe often, you can kind of sense when he started thinking this way, because he stopped talking about fear conditioning, and Joe and I started talking a lot about defensive reactions, and never used the word fear about that. And so a lot of the work that I do in young children looks at measures of physiology and attention in particular, and we find very similar relations between people who have clinical problems with anxiety and these measures of physiology and attention across age. And this is very different from what I call appraisal. I think this is what Joe now calls fear, the report of fear. And this kind of illustrates that idea developmentally, where early maturing systems that relate to attention and autonomic physiology, rapid implicit avoidance, these mature very early. They look very similar in children early in life who later develop anxiety, and these are very different from other systems, particularly late maturing ones. Like BJ, I'm very interested in adolescence, and these phenomena look very different in adolescents, children, and adults when we think about threat appraisal. So much of my work also is conducted developmentally, and very much like BJ was describing, we try to study kids over time, and we try to study how changes in these responses relate to clinical changes. as BJ also mentioned, it's very difficult for many reasons to study these responses in children and adolescents. And the approach that we take, largely related to the measurement complications, is we use multiple different measures in the same children. So that's one of the nice things about being able to work at the NIMH Intramural Research Program. We can see children many, many times. And here you see 7 different tasks that kids undergo that evoke threats. And we measure both these implicit responding and explicit responding on all these tasks, as is described in this recent paper. And you can see that a latent factor can be fit from the data that are showed here. And this relates quite powerfully after adolescence to a whole range of measures, including children's report of their anxiety on a given day or their report over the coming weeks. At the bottom left of the slide, that's one of the stimuli that we used in this study, and that's Frances. And Frances is from a movie that tells the story of this adolescent woman who, like BJ's son, was behaving in a not the best way and ends up have something bad ends up happening to her. Yeah, exactly. And so what we are doing now is we've taken this movie watching paradigm into the MRI scanner, and we describe this in the paper that acknowledges Joe in cerebral cortex. And the main measure that we examine here is we examine the across-time correlation between the heart rate of the child and the adolescents and variation in brain activity. And these are early days. This was the first study in 78 kids. And this just shows you the magnitude of the effects. And the main finding to take away here in the bottom right-hand corner is there's in kids with anxiety disorders, there is a greater connection during movie watching between activity in the subgenial anterior cingulate cortex and heart rate on a minute-to-minute basis in that region. And as we talk about in the paper, ultimately what I think has to happen to move the field forward is these many systems have to be put together and measured simultaneously to thinking about what are the ways in which systems function similarly across species, and things like defensive reactions and autonomic physiology to certain kinds of threats are likely to operate fairly similarly across species, whereas other more complex functions that draw on more recently evolved areas of association cortex, like appraisal or certain forms of memory, those are later developing and are likely to be unique in clinical disorders, particularly after adolescence. And that's really the focus that my group is adopting now. So I have to say I'm really humbled both to be here and to be able to have been associated with you, Joe, for as long as I have. I have to say when we met 10 or 15 years ago, Joe was at the peak of all his powers, and he very easily could have brushed me off and been angry or annoyed because I was critical, but it was humbling that you so deeply embraced me. And I have to say, writing the paper with Joe was one of the best experiences of my life. We would work together for hours and hours and hours, and he really wanted to think very deeply, particularly about the clinical issues. And I feel very grateful for that. And so once again, want to thank you for your influence on me and for all that you've done for the clinicians in the room and throughout the world. So with that, I'll thank you. See, you still make my collaborators list there, Joe, but I want to thank all the people who contribute to this work, and thank you for listening.
Fingers crossed, this is actually going to work. There you go.
That's good.
All right, good afternoon. I'm Huda Akil. I'm from the University of Michigan. I'm really honored to be here at this event. I'm very grateful to you, Joe, and to the organizing committee for including me. And I think I'm maybe a little bit of a weird choice because I am weird and I am a weird choice. And because I have not collaborated with Joe, I haven't trained Joe, I haven't been trained with Joe, I haven't argued with Joe in an elevator, I have not even had a drink with Joe, as far as I can tell, that I can remember. So you owe me a drink, and I would love someday to write a paper with him, but I haven't done that either. But I think maybe I am representing the old guard that also as maybe potentially the chairwoman of his fan club in neuroscience. So I am here really to acknowledge Joe's influence in making us talk and think about the emotional brain, both in English, in French, and other languages. And really, I see him as a founding father of affective neuroscience, or whatever you want to call it. And I want to talk a little bit, go back a little bit in time. This is a quote from him saying a little history goes a long way when he's talking about the study of consciousness that Mike has mentioned. But in my own personal history, I just want to briefly, very briefly, touch on how I became aware of Joe and his contribution. So I've always been interested in the brain biology of depression and anxiety disorders, like several before me. I've been interested in stress biology. I see it as the basic biology of affective and mood and affective disorders, the way cell biology is the biology of cancer. And that understanding it is really has to connect closely, like we have heard, with clinical science, clinical practice, and it has to be a bidirectional connection. And so my lab, in collaboration with that of my husband, Stan Watson, who is, by the way, from Louisiana. There is something about Louisiana and going out of your mind, I think, that kind of go together. But so we've been studying the stress system for many years, and we were interested in the neural circuitry of stress. And Stan's lab, in particular, was focused on, this is a very old work from the 1990s, where we were trying to figure out the circuitry of negative feedback, of the termination of the stress response, and showing that there is a relay station in how you turn off the stress response from the hippocampus through the bed nucleus of the stria terminalis. And one of the questions we had, sort of similar to the question you heard about, is extinction messed up? Is whether negative feedback, the termination of the stress response, was messed up in depression. So we knew here that the BNST, the amygdala, et cetera, were important in this termination. And with my colleague, she was in my lab at the time, Elizabeth Young, we wanted to study whether negative feedback and the termination of the stress response could be messed up in depressive disorders. And we found that that was, in fact, the case, that the stress response would not shut off properly, and that was correlated with hippocampal function, where we know the glucocorticoid receptor exists and have the circuitry that then goes out and shuts off the stress response. Part of the job was to come up with a good stress paradigm in humans. And I can't tell you how many people I have shoved in cold rooms or, tried weights and cardiovascular things and cognitive tests. And we could not get a reliable HPA axis response across a whole group to save our lives. Some people would give us a big one, but others wouldn't. And one day, our research nurse said, you know those kids who give a big stress response? They think we are judging them, that we are going to use the information somehow, and they get nervous. This is what we see in HPA axis response. And so I decided that we really need to study a social stressor. And we started using the Trier social stress test, which had already been developed. And lo and behold, you get a beautiful, beautiful stress response. And it's dysregulated in depression and anxiety and PTSD in different directions. So that then, as a basic scientist, told me I've been not necessarily studying the right behavioral paradigm in my animals. So I got interested in stress and hierarchies, in social hierarchies. And this is a study we did in Syrian hamsters. So now I'm from Syria, and those hamsters, like other Syrian males, are into dominance and subordination. And we did one of the earliest FOSS studies in them, Sarah Kalak-Walker in my lab. And we had a group that was unhandled, a group that was just handled, and then two males that got together and established the dominance hierarchy. And as you can see, the dominant and the subordinate animals, their brain lit up with the white stuff, which is FOSS activation. And sometimes it was hard to tell who was winning and who was losing until you drilled down. And we discovered independently that it was the amygdala where we could tell the losers from the winners. So if you looked at the amygdala of winners, there was not that much activation, but in losers, we saw a lot of activity. And the other area that stood out was the septal area, which was more activated in the winners and not so much in the losers. So we published this paper, and I got very interested in learning more about the amygdala because of that. I mean, I know, but I was a hippocampus and hypothalamus person, you know, and so amygdala, like, I need to learn about that. And this is really where I started, where all that beautiful work from Joseph LeDoux was coming along, and I started reading this lovely work about contributions to fear and stress, the stimulus evaluation, and emotional memory. And sort of this is how I became, and my lab became very aware of Joe's work. Now, what was interesting is that right around that time in the 90s, where all of this was happening, I happened to be on the council of the Society for Neuroscience. That arrow is me. And my head was all over, like, we really need a better science of all of this, like the basic science of affective disorders, mood disorders, and so on. And during the council meetings, we would have a time where we would select committee members, like who's going to go on the program committee and on this committee and on that committee. And people would just call names. And I would call name of people who were doing work on emotions. And nobody ever picked them. Everybody, like my colleagues, everybody wanted learning and memory, perception, motor behavior, developmental neurobiology. But I felt like we were, in affective neuroscience, sort of the stepchildren of neuroscience. And it was really helpful to me to know that there is this burgeoning field, these competing labs that we heard about, were just really gearing up in a big way and starting to have a big impact. And I realized it is coming. Affective neuroscience is coming. Thank you, Joe. And really, that was the moment. And I remember that Stan, my husband, was the chair of the program committee at that time. And I complained to him about this, and he said, there is no topic. There was no topic in the whole Society of Neuroscience meeting around affective neuroscience. Imagine that. Imagine that. So I said, we need to create a committee on committees and make sure it's better balanced. And there is something called the Committee on Committees in the Society for Neuroscience right now. So I started really paying attention to what Joe is saying and doing and thinking. I'm not specifically a fear or threat conditioning person, But we have a program to study the genetics and environmental factors and developmental factors relating to emotionality and temperament. And this is a meeting in Venice, Italy, where I heard Joe give a talk about how we shouldn't say fear, we should say threat. And the next day I was talking, I went back to my slides and X'd out fear and put threat in all my talks. And I was looking at the role of gene and environment in extinction learning, so it's relevant to what you've been hearing about. So Joe remained a very big influence to our lab and my thinking. And even though we don't really specifically work right on top of the same area, I've always appreciated not only his thinking, but his influence on the field. But one of the things that I want to underscore, again, from this greater distance, is his influence on the world beyond the world of scientists. Because, you know, especially I think as you age, like me, you realize that other than training amazing people and collaborating, it's a big privilege to be a scientist, but also to share what it means to be a scientist with the world. And around the topic of emotionality, there is no more important time and no more important people than people like Joe to talk about how to think about emotions. Because emotions are highly maligned. People are scared of them. They don't really necessarily understand them or embrace them. There is a lot of bias against them. And as we heard, like BJ talked about, their adaptive value and so on. And we really do need to find ways to talk about emotions differently. And I feel like Joe has done that not only by writing books, but by sharing himself. So in this work called The Neuroscience and Emotion, the Life, Work, and Music of Dr. Joe LeDoux, if you have not seen it, it's amazing. This is part of a review I've written. You can find it on Amazon. I felt like I wrote that he was a remarkable man, and that his journey is both unique and highly relatable. And to me, it's not only that he was a pioneer in tackling brain emotions at the time when few people confronted that, but how he brings his own life into it and how he connects it to who he is as a person, and how in doing so he has impacted the lives of so many. I feel like for example, the way he talks about music, his relationship to Cajun music, how it connects him with his son, the way it's helped him cope with tragedy, is extremely relatable. And that part of the message, not only about the content, but the meta message is that as scientists, our life informs us and our science informs our lives. And there is nobody better than Joe to have actually reflected that as sort of the mission beyond the science. He and I were in this kind of a weird project called, it was in a museum in Venice about the human brains funded by the Prada Foundation. And during the pandemic, it brought 36 of us. Here is Joe, here is me, you can tell. Behind the same background, they had these extensive conversations with us and recorded everything. And then they picked little things, and then they put us in this dark room. Joe went to it, but I didn't, so I have not seen it, where like a bunch of talking heads around certain topics, and you could hear us talking, I guess, about it. You know, I've never been to it, so I don't know. But they also put out this book about it, and the images, it's very artsy, right? So you could see these dark pages with the little images here. They have objects that we care about. I'm gonna zoom in it, and quotes from us. So Joe picked something called Cowboy Tales, so I was interested in the cowboy hat today. And then they had little outtakes as you were talking, and then quotes, and I'm like ending my talk with a bunch of quotes from Joe about this is the idea of your body producing behaviors that you don't feel you are in charge of is kind of disturbing. And so to resolve that dissonance, it creates what we do as people, is narrate our lives. And so these narrations that we constantly create are only a tiny part of our brain, our mind, and somehow they make sense of what is actually happening in the world. And I like this because I think a lot about beliefs. As somebody from the Middle East, I think a lot about how beliefs drive wars, how they drive life, how they, have just upended the world, and why people tell themselves these stories that make sense to them, but sometimes pit them against each other and create a lot of havoc. And I feel as an affective neuroscientist that I really need to understand beliefs. And I see this connecting very closely to the idea of emotions and cognitions and beliefs. I also, Joe talks about hallucinations and how you're more than your hallucinations, and I appreciate that. But also, he talks about suicide and how humans are the only animals to our knowledge who do that, and that we exist. It means we not only exist in our own past, but we anticipate the future, and that suicide is the decision to make that possible future now rather than later. It's kind of an interesting take on suicide. And the role of the prefrontal cortex, the price that we pay for being anxious. And finally, Joe talks in this book about those little outtakes, about the things that have affected his emotions, the loss of his son Jacob at the age of 17. And he says, so I think perhaps the music was so important, not only because of my connection with Jacob, but it is a kind of way of being in the brain where language fails. And then there is this very moving piece where he apparently is singing. I didn't hear the singing because I didn't go to the museum, but I'm sure you can hear it, where there is this lovely piece, that's a very moving piece that he wrote about his son and death and music. So I feel like I hope I have conveyed to you that there are many levels at which to admire Joe, not only as a scientist, but as a messenger for science and how you can embrace science in a way, and hard truth about being human in a way that is still courageous, has integrity, and gives us hope. So thank you, Joe.
By hitting the right.
Yeah, I think you can just press the space bar once we get you up here.
Oops. All right. Thank you very much. And it's a great honor and pleasure to be here. I'm another speaker who has not collaborated directly with Joe, but has been influenced by his work for a very long time. Like many people, of course, The Emotional Brain, when that book came out, was something that had a big impact on me. But I actually go back a little further, probably than most of you here with Joe, except for Mike Azana, of course, because nobody goes back that far. But I go back at least to the early 1980s, when Joe and Bill Hurst edited a very forward-looking book called Mind and Brain, Dialogues in Cognitive Neuroscience. As Liz Phelps knows, because we teach a cognitive neuroscience course together, every year I tell our students in the first lecture, the first book I'm aware of that had cognitive neuroscience in the title was Mike Gazzaniga's Handbook, 1984, unless anybody knows something before that. And this is the second one. And so Joe and Bill brought together psychologists and neurobiologists to talk about their perspectives on attention, perception, memory, and emotion. And that's the best I could do in taking a photograph of the table of contents. So I was the psychologist talking about memory and went back and forth with Mike Gabriel and his colleagues about our perspectives on the psychology and neurobiology of memory, and possibly more interesting, what we see here is some early writings from Joe on emotion, more than 10 years before The Emotional Brain, and he went back and forth with Ross Buck in a very interesting dialogue. So that goes back to about 1984, I think, was when that whole project got started. I didn't really know Joe at the time. I knew he had published a book with Gazaniga. That didn't quite satisfy my questions about him, and Morris Moscovich vouched for you. And then I was okay going ahead with this project. All right, but for today's talk, what I'm going to do is follow the lines of the article that I wrote with a couple of my graduate students for the special issue honoring Joe. And we talk about here work that we've been doing in our lab on what we call emotional future simulations that actually brings together interactions between some of our interest in emotion, obviously, Joe, the leader in emotion, and work on thinking about the future. And you already heard a little bit about some of Joe's thoughts on that. And he's expressed that in a variety of his books more recently. So there are really some interesting points of interaction here. So in our lab, over the past 2 decades, we've been thinking a lot about the relationship between remembering the past and imagining the future. And a portion of that work has focused on the emotional aspect of thinking ahead to the future. Emotional future simulations I'll define as constructed representations of positive and negative future experiences. These are typically personal, specific, episodic kinds of experiences. And I'll briefly discuss some fMRI evidence concerning neural correlates of emotional future simulations, talk a little bit about a cognitive manipulation that we've used that impacts emotional simulations of worrisome future events, and then briefly talk about some very recent work on individual differences in emotional future simulations related to narcissistic grandiosity. Okay, fMRI. This is a line of work that goes back to 2007, when in our lab we did a very simple experiment. A couple of other labs were doing similar work at the time, where we put people in the scanner, we gave them cue words, and we asked them either to remember a related past experience or imagine a future experience related to whatever that cue word might be. And compared to control conditions, what we saw was strikingly similar activity in the brain regions that I'm showing you here on the slide, that Donna Addis, Randy Buckner, and I referred to at the time as a core network, corresponds largely to the well-known default mode network, that's involved in remembering past events, imagining future events, and supporting related kinds of mental simulations. Key components of that are the medial prefrontal cortex, medial temporal lobe, posterior medial regions, including retrosplenial cortex and lateral parietal and temporal regions. These regions all showed strikingly similar increases, whether you remember the past or imagine the future. That was just based on our work and 1 or 2 other studies available at the time. Over the next few years, work in this area really took off, and by 2015, we were able to replicate that core network and meta-analysis led by Roland Benoit, then a postdoc with me, on the basis of a much larger sample of studies. Now, with respect to emotional future simulations, the region that really captured our attention was the ventromedial prefrontal cortex, because we thought that it might have a role in both functional and emotional aspects of episodic future simulations. So it's consistently activated when people simulate these future experiences and other hypothetical experiences. And in previous work, it had been linked with both the beneficial effects of prior knowledge and schemas on subsequent memory, and also, of course, as is well known, implicated in coding of emotional value. So we kind of pulled these 2 things together in a PNAS paper published about a decade ago, led by Roland, in which we scanned people, they were in the fMRI scanner, and they generated episodic future simulations of future events involving people and locations that they knew more or less about. So some, we had them prior to going to the scanner, they tell you, they give you a list of people and places. Some of these they really knew a lot about, some they knew very little about. And then what we'd ask them to do is take this person and that location, combining together, imagine a future event in which they would be carrying out some kind of action. And what was interesting here is that the medial prefrontal cortex showed what we called a familiarity-dependent coupling with content-specific regions. So the dorsal rostral prefrontal cortex was specifically activated when people were imagining, when participants were imagining people. Parahippocampal cortex, not surprisingly, when they were imagining locations. But the key finding on the knowledge side was that the medial PFC was showing a stronger coupling with them the more you knew about a person or a place. At the same time, it was coding for what we call the emergent affective quality of these simulated episodes. The more you anticipated this interaction being pleasant, the more activity you saw up in the VMPFC. So it was within the context of future simulation, a nice bringing together of these two lines of work on the VMPFC. In a follow-up later study, also with Roland and Philip Apollos, we took a slightly different approach. And here, individuals imagined meeting people who they either liked or disliked at initially neutral places. And one of the behavioral findings was that when you imagine meeting someone you really didn't like in a neutral place, subsequently change the affective value of these places, kind of a conditioning-like phenomenon that was assessed by liking ratings provided both before, what we call phase 1, and after episodic simulation. So just imagining interacting with people you liked or didn't like changed the value of these neutral places. Now, again, the VMPFC was showing relevant activity. So the VMPFC activation was reflecting the affective quality of the person, and maybe more interestingly, it was predicting change in the attitude toward the place, whether you liked it more or disliked it more, from phase 1 to phase 2. So it seemed to be intimately involved in representing these emotional aspects of thinking about this future event and people you would interact with. Okay, move on to some cognitive work. In our lab, over the past decade and more, we've been doing some work with a procedure that we call an episodic specificity induction. And in this particular study, we focused on worries about the future, again, in the theme of emotional future simulation. People worry about all kinds of things that might happen to them in the future, and we wondered whether getting them into a mode of detailed episodic retrieval could help them to effectively deal with these worrisome future problems. So the episodic specificity induction involves brief training in recalling episodic details from a recent experience. So you see a video, then we put you through an adapted version of the cognitive interview. You recall a lot of details about what you just saw. And what we're interested in is do these this detailed episodic retrieval mode, does this carry over to subsequent tasks that you perform? And what we have found is that it increases the number of episodic details in subsequent tasks that tap future simulation and some related processes. In this particular study, Harvard students first told us about things they're worried about, which were typically, how they're going to do on their exam and other things like that. And then they got the episodic specificity induction or a control induction, and then we gave them what we called a means-end problem-solving task, where they've got to generate steps to solve their particular problem that they're worried about. And what you can see here is that they generate more relevant steps towards solving their worrisome problem after they've just had this specificity induction compared to the control. Critically, we also saw effects of these episodic detailed future simulations on various measures of emotion. Anxiety, the likelihood of bad outcomes, difficulty to cope, all these things improved when they were approaching them with this having had this detailed episodic retrieval experience. We're then able to extend this work to older adults as well in a more recent paper. Finally, I'll say a few words about individual differences in emotional future simulations related to narcissistic grandiosity. Grandiose narcissism, as you may know, refers to a subjective sense of self-importance and superiority. It's a fundamental component of pathological narcissism relevant to recent political events in our country. Please do not let that get out of this room. That might be the end for me. And in a recent collaboration with Jill Hooley's lab at Harvard, led by her former graduate student Ellen Finch and my graduate student, now postdoc Sarah Kalinowski, we had individuals who varied in level of narcissism according to a standardized scale. They remembered past events and simulated future events in response to positive cues like imagine a future episode in which you're exhibiting skilled performance of some kind, or negative cues, remember a past event or imagine a future event in which you were or would be inadequate. And what you can see here, if you follow the blue lines, the positive lines, solid line for memory, dotted line for future simulation, what I'm showing you on the x-axis is the extent of grandiosity. It goes from less grandiose to more grandiose, and on the y-axis we've got the vividness of these memories and simulations. And so what you can see is that as you get more grandiose, the vividness of your positive memories and positive future simulations increases in a very similar way and decreases in a corresponding way for these negative future, past and future events. Looking at the plausibility of these positive future simulations, we found that with increased grandiosity, it seems much more plausible to these folks that all these good things are going to happen to them in their future, and much less plausible that these bad things are going to happen. Interestingly, that contrasts with a study we carried out about a decade ago with Stefan and his then graduate student Jade Wu, with looking at episodic future thinking in generalized anxiety disorder, where anxious individuals, anxious patients, you can see in the bar on the right, when they were imagining negative, neutral, and positive events and rating their plausibility, they showed the opposite to what I just showed you for the narcissistic group. They think it's more plausible that these negative things will happen to them in the future than controls, and there were no differences for neutral and positive. And that, of course, links to a whole range of work that's been done in this area on how changes in episodic or emotional future simulations are observed across a wide range of psychopathological conditions. We've heard about some of them already today. Amanda Brunette and I reviewed a bunch of these in an article published a few years ago. And this links, I think, nicely with Joe's fantastic book on anxiety, Anxious, published a few years ago, which I read with great interest and I think is closely related to a bunch of what I talked about. And also, what I've talked about today relates to another one of Joe's wonderful books, The Deep History of Ourselves, where he talks about some of his perspectives on mental time travel and future thinking and how memory and future thinking are related. So I see a lot of convergence between these different lines of research. Thank Joe for all of his amazing work over the years, and conclude with thank you to my lab collaborators, funding sources, and thanks for your attention. I can use,
Use the slides.
I can use this slide.
Thank you. About 30 years ago, an event occurred that initiated two very important associations in my life, both scientifically and personal. This was, well, the title is How a Science Paper Got to be Heavy Mental, and it will be explained in a few minutes. So this is a meeting that took place at NYU, and the associations are between Joe and myself. Joe is I would say, a great personal and scientific friend, colleague, and between me and NYU, but that's another story. And some of the people here are, in fact, in the audience, listed here in this meeting. And by the way, it's interesting to note what were the topics covered and the resolution of the topics. So LTP, well, I don't know if LTP is still a topic in many meetings. Behavioral strategies is general, aging and amnesia, emotion and memory, theoretical approaches and molecular mechanisms. Nowadays you can probably dissociate any of them into maybe 1,000 subtopics. So many of us have dedicated their scientific career in search of the engram. And the initial idea that memories are fixed in consolidation gave way to the dynamic view of a restless engram. So this was a restless engram. So the initial idea that consolidation leads to fixation of memory was replaced with an idea which was actually older than the one that said that traces are fixed in memory. It's just a matter of zeitgeist, and led to the idea that memories are actually restless and they change. They may change in retrieval, and this is a cartoon that shows that. And a new window of plasticity occurs when memories are retrieved, and this may lead to either extinction of the memory or reconsolidation of the memory. And in fact, several of the previous speakers already referred to this change, to this time window of reconsolidation. And of course, a major event that led to the huge increase of popularity of reconsolidation, which is now reconsolidates again, the popularity, but that's another story, was, and this is just where the restless engram title is taken from. This is a review that I wrote in 2012 that says that consolidations never end, and this is the more or less the zeitgeist now. And memory consolidation, I just gave here the plot of the publication citations over time, and as you, I'm not sure you see that, as you see here, till the year 2000, if you can't, you see that there were probably 1 or 2 papers per year that cite reconsolidation, mainly by Susan Sarah. And the paper that led to the change is the year 2000. Fear memories require protein synthesis in the amygdala for reconsolidation after retrieval, which is Karim Nader and Glenn Schaaf and Joe LeDoux. And how the story evolved is another issue that requires a different meeting. But you see that there is a huge increase over time, and with a peak around the 20s, there might be a slight decrease. As I said, the issue reconsolidate, and there are other issues that probably become more popular, but it underlies many of our studies in of memory. And over time, it became clear that reconsolidation requires certain boundary conditions. It doesn't occur every time. This was sort of an idea that was in doubt at the beginning, but it's clear that reconsolidation doesn't occur every time that memory is retrieved. There are certain boundary conditions, and a very important boundary condition that is actually the one that underlies some of the studies that are mentioned here. For example, the competition between extinction and reconsolidation in retrieval, and whether you can use this time window in order to abate an anxiety or PTSD, is the idea that the trace that controls behavior at a given point in time. There are many traces, multiple traces that they compete to control behavior at any given time, especially episodic traces, becomes more susceptible to reconsolidation, which is the one that seems stronger is actually more susceptible to change. And you can immediately imagine why it happens, because this trace is now more plastic. It's the one that dominates the behavior. And this is the paper that introduced the term trace dominance, which is the trace that controls the behavior at a given point in time. So this is about science. Now, given my love affair with the memory trace, I was excited to learn that a song has been devoted to it.
Left a trace in my memory, a permanent place in my synaptic sea. They say that time heals all pain. Time can erase your place in my brain.
So, given that, I was even, just a minute, don't be so happy. Given that, I was more thrilled to hear that in that same song, Joe refers to the dominant trace. So you can use that as an example of the combination of the beautiful combination of science and art in Joe's career. The love of music, the beautiful, very influential science, but it's also an example of poetic freedom. Because as opposed to scientific reality, it allows dominant traces to become more stable. Because he said we will never erase. You are the dominant trace. So the data don't show that, but it's not as simple. Life is complex, as we now see in both countries. Don't cite that. I need my visa. In the lab and in real life, maybe it works. That the dominant trace is the most susceptible one to change. But in real life, Joe, I wish you many more healthy, productive, and happy years. You are definitely left in me a memory trace that is dominant, but is very stable. Thank you.
That song, I don't know if you remember, we were at Japonica on Union University Place, you and Richard Morris, and we were talking about the dominant choice. And Richard said, I challenge you to write a song about that.
I knew that dominant story, but I thought that time would not allow me to tell it. And yes, thank you very much. I'm very proud and happy to be here. Thanks.
So we have a little change in today's program. Nicole Rust can't be with us here today, but has shared some words in her stead video.
So we have a few videos, actually. A number of people have sent us some tributes, some scientific videos in place of their talks. So we're going to do a few of them now. We'll play some things during the break, and we'll have other videos in different slots as we go through the program tomorrow. So we're going to start out with a video that Nene Humphrey made. She was the artist in residence in Joe's laboratory.
Made a map of your mind. I've charted my course. I'm sailing deep inside. I got the winds of force. Got the heat of your heart to keep me from the cold. Got the currents of will to take me to your soul. I'm on a journey that I've got to take. It's a trip that I knew I'd make. An excursion of need and desire. It's a voyage fueled by fire. Made a map of your mind. I've charted my course.
Whoops, sorry. Can I bring it up all the way? Good. Yeah, bring it up full. Yeah, and then can we just play it from here? Okay, thanks.
Hi, Nicole Rust here. I am so honored to participate in the celebration of Joe, and I'm so sorry I can't be there with you in person. I've hurt my foot. I overlapped with Joe as a graduate student at NYU. I was there from the years around 1999 to 2005, but I didn't work with Joe at the time. Fast forward a few decades, I'm now a professor at Penn, and I've had the privilege to work with Joe on a paper titled The Tricky Business of Defining Brain Functions, which was published in Trends in Neurosciences in 2023, it was January, along with a workshop that followed soon after. And I want to emphasize my gratitude to Joe for three things. The first is for his books. Joe, as the author, was a huge inspiration to me when I was thinking about writing my own first book. And in addition, his book Anxious was part of what inspired me to pivot my entire research program to study affective science, specifically mood. In that book, he laid out not only what we know about emotion and what we don't, but also why studying emotion is so incredibly difficult. And that clarity is really what inspired me to want to dive in and help contribute. The second thing I want to thank Joe for is his support. My conversations with Joe and his support have played such an important role for me in helping me make this pivot, including securing a pivot fellowship from the Simons Foundation. But most of all, what I really want to thank Joe for is setting us all on a path that I believe is the right one to arrive at an impactful understanding of the brain and mind of the type that we'll ultimately need to treat brain and mental illness. I was excited to work with Joe on The Tricky Business because what he's been saying is so incredibly important. Words matter, and as brain researchers, we aren't just here to collect facts. We are here to lay the foundations that will be required to ultimately treat brain and mental illness. We need to think very hard about what words do and do not mean, like fear. And the subjective experiences of emotion is so central to that meaningful understanding. It's around this that I'll be shaping my own research program in affective science, and no doubt many others are too. So thank you, Joe. Congratulations on a tremendous career. If I can accomplish even half of what you've accomplished, I will be very, very proud. Congrats to you.
Okay.
Hey, Joe. I just want to thank you for the lives you've changed, for your inspiration, for your knowledge and generosity, and your research and your love. And I know you're retiring, but I doubt you'll retire. I think you'll probably still be giving those things out in the next phase of this journey. So John has agreed to join me and play this song for you. I think you'll recognize it. Mind over matter, it's something I'm trying to do. It's just a little physics that keeps me apart from you. Mind over matter, something I'm trying to do. Break down space and time and be together with you. I'm trying to will you here and find you wherever you are. Locate your body, your soul, pull you in from afar. Are you still in my time, or have you slipped away? Have you gone forwards or back, living in another day? Mind over matter, it's something I'm trying to do. I guess it's only physics that keeps me apart from you. Mind over matter, it's something I'm trying to do. Break down space and time, be together with you. Be together with you. Be together with you.
Okay, so that closes our first session. We have a coffee break coming up at 3:05, and Joe, I don't think coffee is available. It's not set up yet. It's not early, right?
What time is it?
So it takes a couple minutes to walk down the hallway to 233.
2:33.
It could be set up.
Okay, so we'll run ahead and check and see if 2:33 is set up yet, but maybe people can hang out in here for a few minutes, and then you can make your way over for coffee, and we're going to resume again at 3:35. Okay. That was Roseanne Cash, yes.
Hi, everyone. Welcome back to the second session of the day, and we will start with Greg Kirk.
Okay, thank you, Daniela, and thanks to Liz, the two Liz's, for organizing this meeting, and glad to be here for Joe and to talk about the gifts I've received from Joe. So these are the four gifts I got: an intellectual home, career guidance, professional support, and a role model. So I postdoced with Joe. So I'll go through each of these. My graduate work was on place cells in SUNY Downstate. My mentor was the late Bob Muller, a wonderful mentor. But I invited Joe to be on my thesis committee, and he courageously went across the river to Brooklyn for my thesis defense. But after my PhD, I really wanted to make my science more meaningful to me and how to do that. So I had an interest in developing countries, and I was traveling in Latin America. And I got a Fulbright grant to establish the first neuroscience lab in Honduras in Central America. So I went down to Honduras. And the idea was not to study place cells in Honduras, but they were struggling with malnutrition, so to use a rat model of malnutrition and study its effects on the CNS, and to what extent could malnutrition be reversed with feeding, et cetera. So we trained a group of, and there was medical students. There's no graduate school there, but there's medical students, and we did some work in malnutrition. And these are the papers we got from that year in Honduras. And it was very encouraging because it showed me, yes, neuroscience can be socially relevant and can address society's problems. So that was a great year, but then the question is, what do I do now? So I was back from Honduras visiting New York, and I was walking down around NYU, and I turned this corner right here, and I slammed right into Joe LeDoux and his wife Nancy. We actually butt heads, we slammed into each other. And after recovering from that shock, Joe said, "Greg, what are you doing after Honduras?" And I said, "Joe, I have no idea." And he said, "Well, how about moving to fear and recording from the lateral amygdala?" And so that was the intellectual home that Joe provided me, because if I was going to go back to Latin America, again, I just couldn't bring place cells with me. There has to be something more relevant, more clinical, more translational. And so the fear field made a lot of sense for me. So again, Joe's model, the direct projection from the thalamus to the lateral amygdala, and then we needed to use units to test this. And the question is, would the units learn at the latency of the thalamic input? And of course, the answer is yes, they did. They learned at 10 milliseconds, which is the latency of the thalamic input, and they learned faster than cells in the cortex, the tone shock associations. This is good support for Joe's model. So after my postdoc, then there was the question what to do. Do I return to Latin America, or do I stay in the US? So like any other postdoc, I went on the job market and interviews in various places and startup grants and various things. And I got an offer from a very small medical school called Ponce School of Medicine in the south of Puerto Rico. And there I'd be the only neuroscientist. There's no other neuroscientist there. And the startup was $20,000. Now, that was the most they'd ever offered to a person, is $20,000, right? But it was just so small, and everything was so small. And I kind of had my doubts, and I went into Joe's office, and I said, "Joe, I just don't know whether I should take this Ponce job or continue for the job search in another year." Now, what do you think any mentor would say to a rising star postdoc? No, you should not go to Ponce for 20,000 startup. You can get a much better job. But this is what Joe said to me: "Greg, you should take that Ponce job. This is what you're all about." Right? And it just hit me that I had kind of forgotten my sense of meaning. What am I about? And Joe had not, thank God, he had not forgotten what I'm about, and he reminded me at a very key point, because I could have made a big mistake. So down I went to this very small island, Puerto Rico, to start a lab. And I'm also reminded of another great thinker, Carl Jung, who said, "The least of things with a meaning is worth more in life than the greatest of things without it." So armed with that, down to Puerto Rico I went. That's the career guidance from Joe. And here is Ponce School of Medicine, a small medical school, but had everything we needed. We bought this folding table at Kmart for the lab, and just managed to make things work the best we could. And I decided to focus on extinction. Again, no one was really looking at it much at that time, and I thought, well, this is clinically potentially significant. Let's look at extinction. And the first paper on prefrontal cortex and extinction came from Joe's lab. This is the work of Liz Romansky, working with Mariah Morgan, her thesis, both of whom are here. And they found that lesions of the prefrontal cortex increased the number of days it took the rat to extinguish, the tone shock association, suggesting that prefrontal cortex is somehow involved. So I said to Joe, I would like to focus my lab in Puerto Rico on extinction. And he said the following: "Okay, to give you a head start, my lab will delay research on extinction for 5 years." Incredible. I did not ask for this. This was his idea, and it was not going to be a collaboration or co-authorship. No. just saying, we will hold off on extinction so you can get that head start that you need. So that's the professional support, really profound. And it had to do, I think, Joe, with your vision or image of what I would do there, or the importance of that in some way. But it really was the key thing, because there's no way that this little lab in Ponce with $20,000 could compete with the LeDoux lab at that time. So we did work, found out that prefrontal lesions, in fact, don't impair extinction within a session, but repairs the recall of extinction the following day. And then unit recording showed that cells signal this extinction memory at that time, and it increased the interest in extinction and prefrontal control of fear. And then other experiments showing that you can activate the prelimbic, you decrease fear. If you inactivate the infralimbic, you increase or impair extinction. So that these opposite effects that these subregions have on the expression of fear, which is very important, led to these circuits of prefrontal control of extinction, which then were used and explored in humans by Liz Phelps and Mohammed Balad, who was my first student in Puerto Rico, to identify homologs in the human brain, anterior cingulate cortex and ventral medial prefrontal cortex for infralimbic. Then we started optogenetics in Puerto Rico. This is the work of Fabricio Del Monte and Calvin Quinones, and they actually were the first time they actually were able to turn off a pathway by silencing an input. So they were very thrilled. And then we shifted to avoidance. By inserting a platform in the chamber, the rats could jump on the platform to avoid getting shocked. This platform-mediated avoidance, and Christian Bravo was one who developed this. So we've done a lot of work with that. And then this is our last paper from Puerto Rico showing the circuits controlling the expression of the avoidance, and then after extinction, infralimbic and insular cortex and lateral orbital inputs can modulate that expression. After extinction, do I avoid or do I not? And we think the VLPFC in the human brain may be a homolog of this AILO input to modulate the expression of avoidance. This is the work of Fredison Martinez when he was a postdoc. So this was 20 years of the Fear Lab in Puerto Rico, and a lot of people, old former students, came in to celebrate, and Joe LeDoux called in and sang a song for us at this celebration. So these are first for Puerto Rico, first paper in all these various journals to capacity building. And then my former trainees, which are now, they have their own labs in different places, including the United States and Mexico. So they're all doing their thing. And fortunately, two decided to come back to Puerto Rico and set up their labs there. And one, Christian Bravo, who developed the platform mediated, he now inherited my lab and it's now his lab, switched to mice, and is doing lots of things with molecular techniques to study avoidance. So my job in Puerto Rico was done, so I moved to the Philippines, to Manila, to really try and spur neuroscience there, which really doesn't exist at this point. So the idea there was not to have my own lab, but really to help young PIs develop labs in neuroscience. Now they're involved in other specialties, so to convert people into neuroscience, get them involved in neuroscience, start journal clubs, a seminar series, the whole thing. So fairly easy to do, a lot of low-hanging fruit. And the idea is to work with, and particularly this woman here, Rohani Navarro. She really is the one I'm working more closely with, her lab, and looking at addiction. Addiction, a big problem in the Philippines, and looking at glue sniffing or toluene, which is the active component of those drugs, and trying to develop a rat model of this addiction that we can help rats overcome withdrawal. And just a little bit of data showing that rats that have a history of toluene exposure show social deficits. So normal rats would spend more time with a novel rat compared to a familiar rat, but rats with this toluene history don't show that preference. This is the first description of social deficits with this drug. And we're looking at treatments that could revert this, low-cost treatments, exercise and antioxidants like N-acetylcysteine. And we just found out this week that our first paper was accepted in psychopharmacology from Manila. Here's the first poster at SFN from the Philippines in 2023, presenting this work. The MD, PhD students presenting their work, and then other colleagues and staff. Very exciting. Reminds me of the Honduras days. So all these gifts that I received from Joe really made all this work possible. That's really my main message, that, that this whole journey really was started with that push and that reminder and that support from Joseph LeDoux. Now, last, the role model. So I think Joe is a role model for us in many ways, but in this way, I'm really talking about music. So Joe, using music in his work and has really been inspirational. And I did not start a rock band. I have no albums, but I do play the violin. And I've been playing with a pianist in Manila, Chad Gilhang. And the idea is to do like home concerts by Zoom and just sort of incorporate that in. So I'm going to, in Joe's tradition, which he sometimes plays music during his talks, I'm going to play some from our concert last week in a little bit of, Liz mentioned Mozart, we're going to go to Mozart.
Will you send us the Zoom link?
If you ask me carefully for it, yes. Okay, very good. Gracias, Joe. Thank you, Joe.
Hi, I'm Liz Romansky, and I was actually Joe's first graduate student. We started up at Cornell Medical College. And this title here, I was trying to think of what I could say that would really encompass all the things that Joe has done scientifically. And of course, there's no one way to do that. But one of the things that I think he really, to me, has meant is he really provided this gateway for all of us for studying the emotion, providing tractable models, new terminology and ways of thinking about things. And I think that's been, really his greatest achievement. So I think we've heard a bit about his beginnings, his humble beginnings growing up in Louisiana. I had no idea he was, living in a van down by the river in Manhattan while he was doing his early postdoctoral studies. He didn't move up that much when he moved to his first faculty position at Cornell University Medical College, where I met him. This was on the Upper East Side before it got all built up. And we were in a very small lab, just with a few personnel, and me as the only graduate student. And this was in the basement of a very, very old building called the Kips Bay Building. And it had this maze of hallways and labs, and it was kind of a spooky, dirty place down there. I brought a friend by. In fact, I married that friend later. Not because of that, but, and he took one look at it. He goes, wow, have you seen the movie Altered States? It was filmed in Cornell. And I was like, no. So, of course, we had to look this up and found out that, in fact, it was filmed partly in the basement of the Payne Whitney Psychiatric Building, which was just 2 blocks away. So if you watch that movie, you see William Hurt, of course, ranting and raving quite a bit. But he has one rant that was kind of interesting. And he's the lead character, Dr. Carl Jessup, a psychologist who's delving into the origins of consciousness. Does that sound familiar, Joe? But here's the quote, he says, there's a physiological pathway to our evolutionary consciousness, and I'm telling you, it's in the goddamn limbic system. So I learned a lot about the limbic system, mostly from Joe, not from Altered States at all, really, and about so much more. And in his laboratory at that time, between about 1986 and 1993, his laboratory performed a number of really beautiful studies on the anatomical connections, neurophysiological responses, and behavioral studies on these circuits that included the amygdala. And it was wonderful to be a part of all that work. In those studies, he and others showed that there's this very important monosynaptic pathway from the posterior thalamus to the lateral nucleus. And that pathway, in fact, provides both auditory and this aversive somatosensory information. It converges in the lateral nucleus right there during this whole aversive classical conditioning process. See, I didn't say fear conditioning, right? During this aversive classical conditioning, these things are converging in those neurons in the lateral nucleus of the amygdala. In addition, we showed that lesions of just that one small part, that one small nucleus in the amygdala can disrupt tone-evoked aversive Pavlovian condition responses. So very important work. And for me, one of the things that really stuck with me throughout my career and throughout my future career was this idea of the importance of convergence and integration. And in my work in my own lab now, where I study the ventrolateral prefrontal cortex, we're very interested in how face and voice information come together and are integrated in the ventrolateral prefrontal cortex during social communication. And it was really looking at convergence and recording from these amygdala neurons and seeing those amazing multisensory responses that kind of brought that on. Another story, or lesson I should say, that I took home from Joe's lab was looking at the way he evaluated data and experiments. Anybody who knows Joe know that he really embodies humility and integrity. And that really comes into play when you're dealing with science, of course. there was one particular experiment that we did during my thesis where we lesioned, or I should say I lesioned, the thalamo-amygdala pathway. This very important pathway showed synaptic plasticity, direct input to the amygdala, and nothing happened. It did not abolish the conditioned responses that we thought it would. Brought it to Joe, showed him the data. He made me go do it again. So after performing it all again, same results, we were a little surprised for about 1 minute. And then Joe pivoted. We redesigned the experiments to include lesions of that cortical amygdala input. And those combined lesions together, the thalamo-amygdala pathway and the cortico-amygdala pathway, did, in fact, abolish the conditioned responses. And I was really impressed with the way he pivoted so quickly to redesign those experiments and how accepting he was of a complete disruption of a hypothesis that we had. And that was a real lesson to me as a scientist. We published those papers in the Journal of Neuroscience, and I'll never forget this paper because the title has kind of haunted me. The title of the paper that he gave it was Equipotentiality. Now, when you start off a paper with a word that Carl Lashley used, you're setting yourself up for criticism, and people had a lot of interesting things to say to me about that. Thanks for that. Well, I left the lab in about 1992, and it was a sad time for me to leave. We'd been very productive. I was able to publish 10 papers with Joe, 5 of my own and 5 collaborative studies, and I really enjoyed working with so many different people. But I kind of felt I was leaving just as the party was starting, because from that point forward, people just, gradually filled into the lab, and the lab exploded with people and fantastic studies, published in Science and Nature and the Journal of Neuroscience and Neuron. Joe's books started coming out, his beautiful writing taking form, and most importantly, his passion for music really got to be public. And it was wonderful to see that, and especially loving when they formed the group The Amygdaloids and came out with their first album. So it's been a wonderful journey to watch, and I'm so happy for Joe. I've learned so many things from him, and I try to incorporate that, of course, in my research and also in my teaching. And I'm charged with teaching the medical students and the graduate students about the limbic system. So in the first slide that I show them, I tell them there is no limbic system. It's just a concept. This is something that we've often talked about. But in the last slide, I show them this. I show them this slide of the excerpt of lyrics from the song It's All in a Nut, and I tell them that one of the most famous amygdala researchers, Joe LeDoux, in fact, has a rock band, and they are just floored. So thank you so much, Joe, for all the things that you've done for all of us, but especially the things that you've done for me. Oh. Don't worry, I'll keep playing it anyway.
I'll use the preview function, I think.
All right, go ahead.
That's not what I want. Uh-oh. All right, so unfortunately I'm a philosopher, the only philosopher today, which goes to show you that, Joe talks to everyone. So I'm going to present some of my research that's influenced by Joe's work. So a few months ago I saw Joe give this talk that was entitled My Rant, and it's going to be very much inspired by Joe's rant. It's my version of Joe's rant. And it's going to be about non-human animal consciousness or non-human animal sentience. So I'm sure you've seen all those headlines that keep popping, like further evidence that crabs and other crustaceans feel pain. There's growing evidence that insects feel pain just like us. Recently, there was the New York Declaration on Animal Consciousness, where so that prompted headlines such as, a group of prominent scientists believes fruit flies, fish, and mollusks might experience pain and pleasure. Recently, there was a big review of the evidence of sentience in cephalopod mollusks and decapod crustaceans, which led to an amendment to the animal welfare bill in the UK. Okay. So, what is this evidence that keeps growing? How would we know whether crabs, for example, among other animals, have feelings? Legitimate questions. All right. So the main method for answering that question is the so-called markers methodology. The idea is that you're going to determine which animals are conscious or sentient by determining which animals can perform functions that we think are associated with consciousness. So first you find functions that require consciousness, then you verify whether the animal can perform that function, then you say conscious or not conscious. All right. That's the methodology. All right. But now the question is, which functions require consciousness? How would we know which functions require consciousness? So here is a list of, I went through the whole literature. Here is the list of all functions that require consciousness. All right, are you ready? Trace conditioning, integrating information across sensory modalities, self-recognition in the mirror, flexible planning, instrumental learning, responding to analgesics, making motivational trade-offs, the ability to engage in play, episodic memory, flexible self-protection, source memory, remembering which places are linked to noxious stimuli, rapid reversal learning, self-administering analgesics, metacognition, and so on and so forth. So many functions seem to be associated with consciousness or seem to require consciousness. If you find any of that in a non-human animal, the current literature will say that's evidence for consciousness. Okay. Now, you might wonder, where is the list coming from? Where is the list coming from? Why these functions instead of any other function? Well, I've been in this consciousness business for some time, and I can tell you there's no evidence for those in the empirical literature. It's just intuition. For example, for self-administering analgesics, we just have the intuition that a non-conscious animal wouldn't go around popping painkillers. Okay, it's just an intuition we have. It was never tested. All right. For some other functions, we do have empirical evidence, or so we think. So I'm now going to argue that we have no reason to accept anything like the list. Our intuitions are driven by fallacious reasoning, and the empirical evidence is mostly due, I think, to experimental confounds. So I'm going to argue, following Joe, that the current evidence is deeply, deeply unreliable. All right, two fallacies that distort our intuitions about the functional roles of consciousness in our lives. The first one is something Joe has been talking extensively about, which is the fallacy of concluding that just because a conscious state correlates with the performance of a function, the conscious state is relevant for that function. So take this example. You're walking in the woods and you just see this snake. You might freeze and your heart rate will increase, right? And it's natural to think that you do that, or that happens to you, because you feel afraid, okay? Because you feel afraid, because you have the subjective experience of fear. But Joe would say, that's wrong. Behavioral freezing and your heart rate increase are instead triggered by a non-conscious threat detection mechanism. So Joe writes, although scientists are taught from day one not to confuse correlation and causation, they too can mistake the intuitive for the factual. An example near and dear to my heart is the notion that the feeling of fear is what causes us to freeze and flee in the presence of danger. This bit of folk wisdom does not reflect the way things work in the brain, even though it is the accepted view of researchers and lay people. Right. So that's the consciousness-only fallacy. The fallacy of concluding that just because a state correlates with a function, the conscious state is relevant for that function. Second fallacy, the consciousness-first fallacy. Let me illustrate it with a simple example. Can you tell me whether those two are the same? Right. So as you're rotating the figures in your mind, you have a conscious experience, of mental imagery, of mentally rotating the figures. So just assume that your conscious state is relevant for mental rotation. that you do perform the function by having the conscious state. The question is, does that mean that the function requires consciousness? Just because your conscious state is relevant for performing the function, does that mean that you wouldn't be able to do that, to perform that function without consciousness? So there's emerging evidence of unconscious mental imagery in people with aphantasia, in subjects with aphantasia, namely people who report no mental imagery whatsoever. Okay? I'm not going to argue for it, just trust me. Actually, we have a paper coming out with Ned and Hakwan Lau arguing for this. These people perform totally normally. They have perfectly normal lives, and they perform normally on mental rotation tasks, for example. Right? So just because a conscious state is relevant for a function does not imply that the function requires consciousness. You could have an equivalent of it that doesn't require consciousness. Okay. So the upshot is that if it seems just intuitively obvious to you that an animal wouldn't be able to perform some function, pick any function. If it were not conscious, think twice, Joe says. So Joe wrote, judging what an animal is feeling on the basis of what we might feel in a comparable situation is fine as a way of interacting with our pets, but it is not the way to do science. That's a great line. Just as physicists do not rely on intuition alone to understand the universe or biologists' life, we should not rely solely on intuition when it comes to the science of mind. Okay. So the lesson I draw from Joe is look at the evidence. What does the empirical evidence tell us? Okay. As for the empirical evidence that the function actually requires consciousness. Now we are going to say, but Matthias, don't we have good empirical evidence that many functions do require consciousness? And I say, no. 99% of the relevant studies are severely confounded. All right, so let me take just one example which was used in this report. They say that instrumental learning seems to have a particularly strong link to sentience, and that's based on this study. All right, so instrumental learning, you all know about this here. The goal of the study is to associate a symmetric stimulus with pressing a button with getting a reward. What they do is they create two conditions. One condition, you're conscious of the stimulus because this stimulus is presented for a long time. Other condition, it's sandwiched between two visual masks, okay? So you don't perceive it consciously. Now, after this, you decide to press a button or not. If you press the button and the stimulus was symmetric, you get a reward. Otherwise, you're punished. You lose money. Okay. After this, they do a discrimination decision to make sure that you didn't consciously see the stimulus. And the idea is, if you didn't consciously see it, you should be at chance. All right. So, conscious conditioning, we all know this. It works. It would be incredible if it did not work. But there's no evidence of instrumental conditioning in the unconscious condition. So the conclusion is, there's evidence that instrumental conditioning requires consciousness. And so now, if we find it in non-human animals, well, that's good evidence that they're conscious. That's the idea. But wait, there's no evidence that the participants in this experiment unconsciously see the stimulus in the unconscious condition. In the unconscious condition, the masks might be so strong as to erase all visual signals. So the study is not really comparing conscious versus unconscious vision. Unconscious vision like in blindsight patients, for example, where the patients would still be able to discriminate. What the study is doing is really comparing conscious vision versus no vision, versus nothing, because the signal is killed. There's nothing. So it just shows that you can't learn based on what you don't see. Not even unconsciously. So there's no evidence that instrumental conditioning requires consciousness. There's just evidence that learning based on visual cues requires visually seeing the cues, which is trivial. Right. So I think this generalizes to nearly all experiments studying the functions associated with consciousness. Unconscious perception in those studies is almost always achieved by decreasing perceptual signals to the point that perceptual signals become non-functional. In order to make it unconscious, you have to make it non-functional because otherwise people see it all the time and it's not unconscious. And if you think about it this way, almost all results showing that unconscious states are non-functional compared to conscious states, which are all-powerful, are totally unsurprising, because after all, it's by making them non-functional that you've made them unconscious. Okay? So again, we have no reason to accept the list. Our intuitions following Joe, I think, are biased, and I think the empirical evidence, while we are making progress, is not quite there yet. So I think we should be careful about headlines. All right, so that's the end of my rant. And I thank Joe, thank you so much for interacting with me and having all those discussions when I was just a postdoc in philosophy at NYU. You're really a great source of inspiration. Thank you so much.
Cut off somehow. Let's see. So, then we'll do this one. No, that's not. That one? Yeah, that should do. Yeah. Beautiful. Thank you. Oh, wow.
Well, hi, everybody. Thanks so much for including me in this amazing celebration of this amazing scientist and amazing person. My name is Sheena Jocelyn, and I'm a scientist at the Hospital for Sick Children in Toronto. And I'm the proud mom of a 15-year-old girl. And I realized a few years ago that in order for me to still be able to have a conversation with her, that I better learn a little bit about Taylor Swift. So Joe, I hope you like Taylor Swift, because this is a Taylor Swift theme. So this is called Joe LeDoux, The Eras Tour. So this is my daughter, Charlotte. She is 15, and last weekend she went to see Taylor Swift with her friends. There they are walking in. And Taylor's actually performing this weekend in Vancouver. It's the end of this very long Eras tour. So this is Taylor Swift, and she goes through like her different eras, her different albums, her different personas, and what she's interested in. And I think the same thing sort of happens for Joe. So this is how I figure his eras are. So he sort of started off, we're a bit funny there, but you can, this is Mac to PC to Mac, but studying emotion, Joe. We have amygdala tracing, Joe. We have the molecules of threat conditioning, Joe. We have, of course, reconsolidation, Joe. We have the threat and fear avoidance, Joe. We have the author, Joe, and we have the rock star, Joe. So those are the sort of eras that I put in for Joe. And this is a very famous poster of Taylor Swift, and she is the most famous rock star in our house. And this is the same one for Joe LeDoux. He is the second most famous rock star in our house. My daughter tells me there's an app, and she took me to the app, and you can make this. So anyone can download their own poster. So of all these amazing eras from Joe, I'm going to take you back in time to the era where I first met him. So the year was 1997. Slides were blue background with usually Comic Sans. And here I am, fresh-faced young postdoc, and everyone was just sort of having fun getting started with PowerPoint. So I was doing a postdoc with Mike Davis, and we were collaborating with Eric Nestler, and I was at Yale. Luckily, people don't do that so much anymore in their talks, but it was fun to, go back and do that. And I was looking at what happens when we overexpress this transcription factor called CREB, details aren't important, in the amygdala. And I love this vision of the eyes popping out, seeing a snake, so I needed to include this here. And we wanted to say, what does that happen on at the time we called fear memory? So I was in Mike Davis's lab, and Carrie's already said that we're sort of cousins on the sort of evolutionary family tree of the amygdala. So I wanted to overexpress this in the lateral amygdala and see what happened. And we were in Mike Davis's lab, so we studied something called fear potentiated startle. And this was in a rat, so we would pair a light with a shock for the conditioning phase, and then we would test the rat with the noise and see if the rat jumped more when the light was on, showing fear-potentiated startle. And as you recall, back in the 1990s, you couldn't give a talk without a Far Side comic slide. So here's the startle reflex, and this is showing fear-potentiated startle. So these are the results that we got from that experiment. So LTM, long-term memory, this is a wonderful Excel graph. It's so beautiful. So what we saw was that we had very low levels of fear-potentiated startle, so a very small decrease, a very small change between what happened when we played the light with the startle noise versus the startle alone in our PBS, in our control viral expression. But we had this huge increase when we overexpressed Krebs in the lateral amygdala, and we didn't see this when we expressed a non-functional form of Krebs. And we were really, really excited about these findings, and we ended up publishing it in a paper in the Journal of Neuroscience in 2011. And we're very excited about that. But one thing that we noticed, and of course all the reviewers pointed out to us, and if any of you are in the audience, you can raise your hand now, was that we got this really lovely enhancement of fear-potentiated startle, even though we infected a really small percentage of cells in the lateral nucleus of the amygdala. So we had about a pretty sparse infection, about 10 to 15% when I went through and counted all these things. And we couldn't understand, why is it that we see this lovely increase in this, fear or threat memory? Why is it that we saw that even though we infected a really small number of cells in the lateral amygdala? And then we started thinking, well, how could we explain this? And then we thought, well, how do we think an experience is represented in a relevant brain region, such as the lateral amygdala, at the sort of ensemble or circuit level? Do we think it's a dense code where every cell would be involved? Or at the sort of other end, do we think it's like a grandmother cell, this sort of Jerry Lettvin concept where all this information would be stored in one cell? And of course, we think it's, probably going to be in a sparse code. And I've lost my PowerPoint. Sorry. Where'd we go? Oh, there it is. There we go. We found it. Am I conscious? Yes, I am. Okay, next. It's going to be a sparse code. That could be, we could try that as maybe one of the tests. Okay. So here's what we thought. Maybe we saw this large increase in this memory because these neurons that we've infected with our Krebs vector are somehow more likely to be recruited or allocated into this engram, supporting this engram or memory trace that's supporting this conditioned threat memory. And many of you have probably heard of the concept of an engram. It was defined by Richard Semon, and I learned about it from reading Dan Schachter's beautiful book. And Dan, I'm not going to show that wonderful old picture of you, but I usually show it every talk, and I'm sorry. Apologies. So an engram, Richard Semon, the scientist, defined it as the enduring, though primary, latent modification in the irritable substance produced by a stimulus. So it's essentially a memory trace. But the language is like really, really opaque and kind of archaic. The irritable substance, I'm presuming that's the brain. I'm not quite sure. So in this sort of Taylor Swift tradition, I went back and I redefined engram. So this is Sheena's version. So Taylor Swift went back and re-recorded all of her songs so that she would own the rights to them. So I'm going to try and own the rights to this. And I'm going to give my own definition of engram. It's cut off here, but it's the ensembles of neurons that are active during an experience that somehow become important in the memory of that experience. So I went back and redefined that so people would understand at least what I'm talking about when I refer to the engram. So we go back to Carl Lashley and his search for the engram, and I'm going to redefine that. So I wrote a paper way back in 2010 where I said continuing the search for the engram, and I was going to examine the mechanisms of fear-threat memories. So now I'm going to change from startle reflex to something that is really easy to do, inspired by the beautiful work that's been coming out of Joseph LeDoux's lab, where we just pair a tone with an aversive shock, and then we look at the percentage of time that mice spend freezing as an index of their memory, and of course we look in the lateral amygdala. So what we thought, oh my goodness, I'm all mixed up with these slides, but I will walk you through it. We found that, so here's the lateral nucleus of the amygdala. Each of these is a really large excitatory pyramidal neuron, and when we inject our Krebs virus, we infect the sparse population of neurons. Now, how do we know that these neurons are important in this memory for this threat conditioning? Well, we're going to look at the neurons that are active when we ask the mouse to recall this memory. And what we found was that, we're going to define that as our engram neuron, and what we found is that those principal neurons that we've overexpressed Krebs in seem to be preferentially biased for allocation, supporting this sort of threat memory. And the very first time I presented these findings publicly was at the Molecular and Cellular Society meeting, Molecular and Cellular Cognition Society meeting that occurs before SFN, and this was in 2007. And there I am getting ready to give a talk, and Joe LeDoux was one of the first ones in the audience giving me a ton of support, and I cannot thank him enough for that. I also chatted with him at the very first Gordon conference on the amygdala. There's Joe, front row, almost center, which maybe we can sort of front row, almost center. Maybe that's sort of where you like to be. I'm not sure, but that sort of seemed fitting to me, that you are right up there, but you don't want to be the total center. So he's always been a huge supporter of my work. I cannot thank him enough for the amazing questions and conversations we've had over the years about how I interpret this, how I interpret that. And it's really been fun to sort of catch up with him over the years at different conferences. So that was the very first thing that I was super proud about in my career, was finding this overexpression of Krebs in the lateral nucleus of the amygdala increased this threat memory. And over the years, we have looked at why these cells overexpressing Krebs are more likely to be allocated to this engram supporting threat memory. And what we found was that Krebs is a transcription factor. It does a ton of things, but one of the key things it does here is that it increases the excitability of neurons that it's infected, and they go on to become neurons that are important in this engram. So these are the many papers that we've published since that time. And so what we really wanted to do then was to say, okay, what we think happens is that neurons compete against one another for recruitment or allocation into this engram. And we think that the neurons that win this competition are the ones that go on to hold this threat memory. But how can we possibly study this? It's all very correlational. So if we think that these neurons compete against each other, and this winning neuron in the middle wins this competition, gets to hold this memory because this neuron is more excitable at the time of the training event than the others, the key experiment, of course, to do would be to target just that neuron after the mouse has learned something and see what happens to the memory. And in a control experiment, we would do the same thing to a control group, kill the same number or target the same number of neurons that we don't think are important in the memory, and see what happens. And this is exactly the experiment that we did later on this, in this sort of experimental vein. So we used a genetic trick that allowed us to kill neurons after they'd been infected with either our Krebs vector or our just GFP expressing vector. So this is the experiment that we did. We first trained and tested mice before there'd been any killing. So this is our Krebs group, good memory. This is our GFP group, also good memory. And then we went in and we killed just these cells that we've infected in the two different groups. And lo and behold, boom, there's killing. What we see is that there's a decrease in memory when we kill the cells that were expressing Krebs at the time of training. And when we kill the same number of cells in our control group, but cells that weren't really important in the memory, we see no deficit at all. And this paper came out in 2009, but right before it came out, the postdoc who was the first author on this, Jin-Hee Han, gave this talk at MCCS, again, Molecular and Cellular Cognition Society, in 2008, and his big request to me was, can you introduce me to Joe LeDoux? He's my hero. And sure enough, we got a lovely photo with him. So this paper was actually published on March 9th, 2009. And this is Taylor Swift saying all too well. She remembers something all too well. And there's a 10-minute version of her singing this song. So I say that I remember this date all too well, to the date one of our biggest papers came out from our lab. And how do I remember that? Well, here I am. March 9th, 2009. I'm very pregnant. I'm across the street at another hospital where I'm giving birth to my daughter, Jin-hee, who had no problem getting his picture taken with the amazing Joe LeDoux, however, felt very uncomfortable talking to the press about his paper. So here I am on my laptop doing a press interview while I am in labor. Sure enough, the nurses came and took that away from me. I was wheeled off, and one small emergency C-section later, the next day, the Toronto Star, my hometown paper, said science a step closer to the spotless mind, where they talked about this work. I'm very excited about that. They said Sheena Jocelyn, a sick kid scientist, politely, I am Canadian, declined an interview request from the Star because she had just gone into labor. So here is a picture of Charlotte at her very first Amygdala Gordon conference. And here she is later, now she's pretty grown up in this picture, at this wonderful conference in Florida. She also wanted her picture taken with Joe LeDoux. And here she is hamming it up with the lovely statues in Florence. So I'm going to say that one of the real highlights of my career has been interacting with Joe and the amazing scientists that have come in and from his lab, and just really reading about the sort of depth with which that they think about things. They don't just do an experiment, they really think very deeply about it. So you can imagine how thrilled I was when I got to finally collaborate with one of my true role models, and we published this paper along with Eric a few years ago, 2013. So it wasn't all that long ago. Wow, 2013, seems like just yesterday. And so there's been many, many eras of Joe LeDoux, and I've been thrilled to witness many of them, sometimes close up, sometimes from afar. And for the special issue of Cerebral Cortex, I wrote a piece about Joe LeDoux and role models, and my concluding statement was, for all these reasons I listed, Joe LeDoux continues to be one of my scientific role models. Some might argue that the idea of a role model should be relegated to business speak, self-help books, or that the concept is adorably retro, having been replaced by social media influencers and Nepo baby celebrities whose rapid fall from grace is both expected and disappointing. But I still try and emulate the behaviors of my role model, Joe LeDoux, and one of my other role models, Sir Charles Barkley, because I've always wanted to dunk a basketball. But that is another story. So with that, I just want to show this picture because I see that Kareem is in the audience, and this is one of my favorite pictures. I remember this was one of the SFNs where he and Glenn were talking all about reconsolidation, and you can see me here, and I'm thinking of going back to that hair color. Anyway, I just want to say thank you so much, Joe. I'm thrilled to have witnessed your many eras, and I can't wait to see what the next one will be. Thank you.
So we have a little bit of a change in the plan. Christina Alberini unfortunately wasn't able to make it. Instead, we will show you two videos. The first one is from Hakwan Lau. It's coming.
Hello, everyone. My name is Hakwan Lau, and I'm sorry for not being able to be there. And but I'm very honored to be invited to have a little presence here virtually. It's an honor to be able to be here to celebrate Joe's career. I remember when did I first met Joe? It was a kind of interesting occasion, I think, when we were playing music together on the same stage, but in different groups. In a Lower East Side bar. So when I was living in New York, and I remember, everyone knows who Joe is, but I was just too scared to talk to him, and I was just playing music, and at some point he came and shook my hand, and I thought that he just thought I was the random guy who played guitar in Richard Brown's group, and I just went home, and then I told people I met Joe LeDoux and blah, but I didn't really talk to him. But surprisingly, in a few months, or maybe more than a year, he actually emailed me and asked, why don't we have lunch? I was passing by New York again, and I thought that he was to complain about the musical setup or something. But turns out he wanted to talk about consciousness, which was really shocking, because the field of consciousness, as some of you know, isn't exactly a very respectable field. It's pretty chaotic and often borders on between the scientific and the not so scientific. And my own reputation in the field is maybe even worse. If I'm generous to myself, I would say my reputation is mixed, really I'm just kind of the black sheep in that field. But Joe was very interested in the issue. I remember he came with very well prepared questions, and he read my papers and asked me, like, how do I think of certain matters? And that was really very refreshing and just very different. You know, in our field, I think many senior authors would just say whatever they want. In part, it may be their problem, but I think it's also the case that the data is really so bad that it allows all kinds of interpretations. So very few people really care to argue, especially with people who are nobody like myself. And so I was quite pleased with myself. I thought maybe I earned a reputation for being a critical thinker or just very opinionated. But turns out he does it with all sort of people. He also, consult Richard Brown on all sort of philosophical stuff, and he openly talks about this, and he acknowledges where the ideas come from, where he benefited from discussions. So that's really, I think, something that to me, that's what when I think of Joe, this is the first thing that came to mind, is this level of humility and very fair kind of leadership. And that's very admirable and almost shocking. It's really very rare in our field. And then since then, one thing led to another. We started co-authoring some papers. And I must say, I often feel completely out of my depth. It's a great honor to be able to co-author with him. But at the same time, I often feel the topics were, because he works on emotion and I mostly work on basic, simple perception. The two fields are related, but I often feel out of my depth. But another shocking thing was he would often say the same. He also feel that he's only going into doing this because of having me as a co-author. And of course, he was being very generous about that. And I learned a lot more than anyone could in this situation. So yeah, this is what I would think of him. In our field, it's very rare to have this kind of leadership of someone who has the stature and influence and years and decades of experience, starting from his work on split brain patients, and yet not just like barging in and just pushing for his own ideas. Of course, everyone has their own ideas, but I think his way of doing it is very fair and very self-critical and very humble and very much trying to take into account opinions of people who often don't even have a voice, but he would just seek them out and see what they mean and try to incorporate the ideas and wrestle with the issues. So yeah, this is maybe all I should say here. There's a lot more I can say, but maybe I should save them for later, because I do think that I don't believe him for a minute that this is the end. I don't think he would actually retire. I think he would just continue to think and to lead the field. When he's not making music, he would probably write more books and make more impact. So I'm sure there will be more chance that I can interact. So I hope that my absence is excusable. Thank you.
And the next from Steve Fleming.
Hi Joe, it's Steve Fleming here in sunny London. I hope you're having a fantastic celebration. I'm sorry I can't be there to join you all in person. So it's hard to squeeze all my thoughts on what has been an amazing and prolific career into just a couple of minutes, but I thought I would highlight a few areas in which you've been a key influence on me and my thinking. So I first discovered our shared interest in consciousness science when you started collaborating with our mutual friends Richard Brown and Hak-Wan Lau on refining higher order theories of consciousness. Now, I'm not sure if higher order theories have been talked about or not yet, but in a nutshell, a higher order theory says that what makes a mental state conscious is that it is monitored or targeted by a meta representation. And that can explain why I'm aware of seeing the computer screen in front of me right now. Now, higher order theories have long been popular in philosophy and notably advocated by your fellow New Yorker, David Rosenthal. But it's only recently that they've begun to gain traction in neuroscience. And the obvious way of cashing out a higher order theory in neural terms is to say there's a brain system involved in monitoring sensory processing. And that's usually specified vaguely as, well, the prefrontal cortex. But you've described beautifully in your books and articles that that's just much too simplistic. I think you're a fan of the promise of higher order theories, but you're reminding us that it can't be as simple as one brain region or circuit monitoring another. Instead, you've urged a more detailed focus on the neuroanatomy and how this functional anatomy differs in its details, particularly in the frontal lobe between humans and other animals. Now, this was always music to my ears because I was approaching this subject from research on the contributions of prefrontal areas to metacognition in humans. And I wanted to know, how does this rich functional anatomy map onto higher order theories? So nowadays, whenever I think of these theories in consciousness science, I always try to remind myself of the LeDoux lesson. We can't simply adopt a one-to-one mapping between philosophical ideas and brain mechanisms. Instead, understanding the messy reality of the biology and how it evolved is crucial. Another major impact on consciousness science has been your urging to ensure that we seek out experimental measures that can't be explained by unconscious processes. So this is reasonably straightforward in humans, as we can usually trust what people say about their experiences. But even then, there's challenges and potential dissociations, as you've shown in your pioneering work on split brain patients. And it's even more difficult in animal models, where behaviors may be driven by the kind of action control that we think in humans can occur unconsciously. Now, this perspective is often unpopular because people like to give animals the benefit of the doubt when it comes to consciousness. But scientifically, it's vital, as you've pointed out. In particular, if we don't have good behavioral tests of conscious processes, then pharmacological and circuit interventions aimed at changing conscious experience and psychiatric symptoms in humans might go on to fail after expensive preclinical trials. And I was lucky to watch you engage in this kind of vital integrative neuroscience firsthand when I was a postdoc at NYU. And you were in the middle of a productive and long-standing collaboration with Liz Phelps's group. And in the future, it's clear that developing new treatments is going to need exactly that kind of work. Animal and human neuroscientists working side by side and taking consciousness both seriously and skeptically. So it's been an absolute pleasure to get to know you over the past few years. And as someone who once dabbled incompetently in the New York music scene as a postdoc, I've always admired your love of life outside the lab and your passion for music and your easy ability to bring people together, just like you're doing right now. I'm also hugely grateful for the personal support you've given me and others in consciousness science. So I always look forward to crossing paths with you and having a drink at conferences and meetings, and I hope there's going to be many more of these to come in the future. So cheers, Joe. Have a wonderful day.
Hello, I'm Eric Klan. I'm at CNS at NYU.
I won't go into all the details, but Joe is the reason I came to NYU 18 years ago. And his work has influenced me even before I got to NYU, and I thought it would be fun to give you a little story of our many collaborations together. I published more papers with Joe than any of my other colleagues at NYU. And I actually consider him one of the godfathers of the field that I'm actually best known for working in, and that is on regulation of protein synthesis, especially in the context of memory consolidation. So I often start my talks with this slide. And it's funny because all the molecular people that end up in this field, they've copied this slide and they use it, but they don't even actually know where the data comes from, right? So we know from studies from Flexner and Agronoff in the 60s and Squire in the 70s that protein synthesis is required for memory, but most of those studies were done with memory systems that take many, many trials to do the memory. They inject the protein synthesis inhibitors at different times. Sometimes they're IP or it's ICV, so you don't know where in the brain it's acting, et cetera. And so, Joe in his system, along with Glenn Schaaf, was really the one who defined and demonstrated for the first time that it was memory consolidation that required protein synthesis, and he defined, of course, the nuclei in the amygdala that were responsible for this. And, Joe's known for the amygdala, but, he also showed this in the hippocampus, which is where I was working at the time, right? So he really is, the basis of much of the work that we do and many others in the field is based on this. And so when I came to NYU, not surprisingly, I collaborated with Joe. I was very fortunate to co-mentor a graduate student, Carrie-Anna Collinsage, who's actually here, which is great. You've seen some of these papers already from both Sheena and others. But I would draw your attention to this paper, which really started a really interesting journey. This is Carrie-Anna and Chuck Hofer, a former postdoc in the lab. I won't go into the details. They show that a particular type of translation is required for memory consolidation in the lateral amygdala. That was followed up by another paper by Linnea Ostroff, who's also here in the audience, which is great, where she was following this up. She was doing beautiful EM work showing how inducing threat memory would cause redistribution of polyribosomes to dendritic spine heads and that it required EIF4E. And it was in those studies that Linnea was doing, she was doing immuno-EM and looking to see if these translation factors moved from various parts of the neuron to others, that she found that, that these translation factors and then subsequently ribosomes are not only present in dendrites, but they're also in axon terminals, right? So it had been speculated for a long time that there might be axonal protein synthesis, and it had been shown in invertebrates and had been shown during development, but it had never really been shown in an adult neuron. And so that led to Linnea's second paper, which she started in Joe's lab and finished in my lab, where she used some fancy molecular techniques, and she really demonstrated for the first time that there were learning-induced changes in translation locally in cortical axons in the amygdala. Right, the first evidence really that local translation might be involved in memory. And so that's, I think, and I'm going to come back to that paper because it illustrates how Joe's work always has an impact on our work. So there was another collaboration that we did with Joe. This is this paper from Priyana Shrestha, who's a former postdoc in my lab. And Priyana had developed these fancy chemogenetic mouse models where we could have very tight temporal control and also cell type specific inhibition of protein synthesis. And we used this, of course, and demonstrated that it actually worked in the lateral amygdala and excitatory neurons. So that's not that surprising based on all we know about plasticity and memory consolidation in the amygdala. And so I was really excited about these tools, and Priyana came to me and said, okay, where are we going to focus next? And she said, I want to work in the central lateral amygdala. I'm like, okay, because I'm not, I came to the amygdala from the hippocampus. I barely knew the lateral amygdala, and now I'm moving to another nuclei. But
of course, Joe had already demonstrated that protein synthesis in the central-lateral amygdala was also required for memory consolidation. And this is a really interesting nuclei because it's primarily made of inhibitory neurons. And Priyana showed really nicely, even though Joe's not an author on the paper, that these inhibitory neurons are loci for translation in both threat conditioning and safety conditioning. And so, and I think I have these out of order. I'll come back to that. And so what she showed was that if you inhibit protein synthesis in these somatostatin-expressing neurons, it would block the consolidation of the conditioned threat response, whereas if you block protein synthesis in PKC delta neurons, it would block the conditioned safety response. And we think this is due to oxytocin receptor signaling. And Rodrigo, who's also here, owes us a paper where he showed that oxytocin is actually required, so we still have work to do, Joe. And so I want to come back to this paper by Linnea. So, of course, we would like to know if this is causal, right? So I'm often introduced at meetings or in universities as being somebody that's demonstrated that local translation is required for memory consolidation. And that's just not true. There's no evidence at all. I wish I had shown it. It's one of the things I hope to achieve before we finish. And so I'm always I'm fascinated, the idea that you have to be careful about the words you use is really important. And I never even used the word local until Linnea published that study in any of our studies. So anyway, this prompted us to develop even better tools that we have better control of translation to see if we can determine whether local translation is required for memory consolidation. So we have an optogenetic tool now, and this is how we did the experiments. We based it on Linnea's experiments where we express our optical inhibitor in the excitatory projections from the cortex to the amygdala, shine the light in the amygdala, and there's a hint of an effect, but it's not convincing. And so the first time I talked about this was at a symposium at SFN, and Joe happened to be in the audience, and afterward he came up and said, really great talk, but you should probably look at the thalamic nuclei. And I was, okay, of course Joe has done something along these lines that I probably forgot. I read at the time, but forgot. And of course, in this paper, he basically hypothesizes that presynaptic protein synthesis at the thalamic input to the amygdala is actually required for the consolidation of memory. And now I'm fortunate enough to have a graduate student who's studying this, and she's already demonstrated that those neurons, actually there's an increase in protein synthesis when you train the animals. And I wouldn't be surprised at all if we're able finally to demonstrate that local protein synthesis, at least at this input, is required for memory consolidation. So Joe is not only, goes all the way from consciousness to the nitty-gritty of the molecular biology of how proteins are made. And I think that impact is so unusual in the field, and it's really amazing. And I'm just going to close one of my envies of Joe, which are many, is that he's has this now music career also. My wife and I are audiophiles. We listen to music nonstop. We once calculated we've been anywhere from 500 to 1,000 shows, mainly rock and blues and Americana, but also other types of music, classical. Amy's a classical trained pianist. And so at some of these shows when I go, if I'm close enough with my iPhone, I take pictures. And now I have this great wall where I've taken some pretty nice photos. And here's one of my favorites. And this is, for those of you who don't know, this is Patti Smith, who's one of our favorite artists. I think we've seen her at least 20 times, maybe more. And you'll notice this guy over here, this is Lenny Kaye. He's the lead guitar player, and he's really the guy that holds the band together and does really the music. Patti Smith's more of a lyricist and poet, but Lenny Kaye is. So imagine I'm sitting, and I have guitars, but I'm so terrible I could never play at all, let alone play in front of people. So I'm sitting there one morning having coffee, and I'm reading and getting these notices, and I see, what? You got to be shitting me. Joe LeDoux's playing with Lenny Kaye at a museum? I mean, come on. Is this possible? Who is this person? And so, Joe, this is, Patti Smith's referred to as the godmother of punk. So, Joe, the godfather of protein synthesis in the amygdala. And, Joe, you really rock. Thanks.
So actually, if we're lucky, Lenny Kaye might show up tomorrow. So that concludes our session 2 of the day. You're all welcome to a reception in the same room of the coffee. That's down the hall, room 233. And please come back at 6. We have a special Amygdaloids reunion for you, an acoustic gig.
Apparently there are lots of Facebook posts that are going around about this wonderful event. I've heard from the Netherlands that somebody keeps seeing you and they're like so jealous that they're not here.
Yeah.
So I'm just posting another one to make them even more jealous. Okay. So.
All right. I think we're pretty close. Okay, so we are well, first of all, first things first.
Is everybody over 18?
All right, the amygdaloids. So, the original amygdaloids, Colin and me. Then we had a bass player, and the bass player rotated quite a bit over the years. At one point, the bass player became Amanda Thorpe, great friend from London. She moved back to London and was replaced by Colin Dempsey. And I get invited to give lectures in different places, and they always, not always, but I'd say 80% of the time, they say, "Can you bring the amygdaloids?" And I said, well, not really, but I can bring Colin. Oh, I didn't introduce David Humphrey here. David is a visual artist in New York and a great musician, and he sits in with us quite a bit. So they say, "Can you bring the amygdaloids?" I said, "I'm bringing Colin." I didn't? I said, yes. We were the original. Tyler Vokes and Daniela, we were the original amygdaloids. All right, so this is kind of an amygdaloids reunion kind of thing. So we're going to play some of our heavy metal songs. We're going to start with one that, it's sort of, I think it probably reflects the two greatest philosophers in the world ever in life. Rene Descartes, of course, and John Lennon. So if you can figure out what the John Lennon part is, let me know afterwards. I think so I am, feel so I must be. Sometimes I don't know it's to be me. I see your eyes staring at me, sharp as knives. What do they see? You can't see inside of me. That's a place only I can be. My soul has passion, it's trying to feel, searching for desire. Nothing seems real. My will is weak, not very free, nothing to get hung about, that's just a deal. You can't see, oh, you can't see
inside of me, inside of me. That's a place only I can be. You can't see, oh, you can't see inside of me, inside of me. That's the place only I can be. Think so I am, I think so I am. I feel so I must be, so I must be. Sometimes I don't know, I don't know. It's to be me. I think so I am, I think so I am. I feel so I must be, so I must be. Sometimes I don't know, no, I don't know. It's to be me. You can't see, oh, you can't see inside of me, inside of me. That's the place only I can be, only I can be, only I can be, only I can be.
All right, thank you. So, anybody got the John Lennon line? Yeah, you don't know?
It's not a question to get hung about.
Exactly. Nothing to get hung about. That was a Lennon, key Lennon song. Okay. You know, the amygdaloid started, just me and Colin kind of playing guitar together and...
Tyler, not Colin.
Tyler, man. Okay, so Tyler and I would get together. We found each other because we both wrote books about two lake people. And we got together and found out we both had Stratocaster guitars. So we started kind of just playing together. And we noticed that a couple of the songs we were playing had some kind of mind thing, like Mother's Little Helper, Mind Brain, 19th Nervous Breakdown, and Manic Depression. So, you know, I said, look, I can write some song like this and try it anyway. So we started out as a kind of like having a good time. Daniela joined us for a gig, and we went from having a good time to, it was kind of a joke at first, you know, the amygdaloids and heavy mental was our kind of music, and the joke became an obsession. So here we are, still obsessed. All right, we're going to play another song. This is called My Mind's Eye. About, you know, the first song was kind of about consciousness. This is about how your unconscious brain, mind, can sometimes play tricks on you.
If you look into my mind's eye, you would see the way that I cry deep inside in my mind's eye. We had love, it was good. You were there, you understood everything, yes, everything. Can't always trust what you think you know, can't trust what you believe. Sometimes your mind just puts on a show, it simply deceives. My mind's eye, you were mine, my mind's eye was blind. If you look into my mind's eye, you would see the way that I cry deep inside, in my mind's eye. Can't always trust what you think you know. Can't trust what you believe. Sometimes your mind just puts on a show. It simply deceives. You were mine, but my mind's eye was blind. My mind's eye was blind. My mind's eye was blind. Oh yeah, my mind's eye was blind.
All right. So, earlier today I mentioned people who had been in the lab a long time, and one person who's been in the lab a long time is Mian Wu. And Mian would like to do a karaoke version of the song Mind Over Matter that you saw Roseanne Cash sing. Not to get any pressure on no. Come on.
Oh my God. This is the boldest thing I never did in my life. And, you know, I used to be a hidden person in the lab. For many years, probably nobody heard me publicly speak anything. But until Joe started to rethinking about emotional memory and fear. So I got on myself and so rethink about my life. And incredible to hear so many people get touched by Joe's research, and not only in our field, but beyond. And that's incredible. I graduated from medical school. I'm particularly impressed by those physicians think about how Joe's theory applied to human. And this is remarkable. You know, I have to see, for Joe, I'm the second longest LeDoux member after Claudia Farb, and which is such a sweet lady. Now we stay the loyalist for Joe LeDoux, and for Joe, I said, okay, I can do everything, but I hope I will not scare you guys away. Joe's music will continue. This is not a karaoke machine. I did not rehearsal, but I just like, you know, Mind Over Matter. You already warmed up this morning. Look at the lyrics, and I put some lab slides go through it. And I think most of us would resonate some of the emotion from our heart. Yeah, I try to do my best. Okay, I wish if you can sing along with me, that would be great. Okay, let's try it. Mind over matter. There's something I'm trying to do. It's just a little physics that keep me apart from you. Mind over matter. There's something I'm trying to do. Break down space and time and be together with you. I'm trying to hear, find you wherever you are. Locate your body, soul, pull you in from afar. Are you still in my time? Or have you slipped away. No one go by, even in another day. I'm sorry, Joe asked me to, thank you. Joe asked me, don't be nervous, look at the ceiling, and pretend yourself. But I have to look at the screen because I couldn't remember the lyrics. Oh, yeah, you may be different continent on the sea, in the air, fly free, or in a place heavenly. Oh, there you are now. Have you found your mind to find? No amount of space and time can keep you from being mine. Sorry about the lyrics. Thank you. You know, it's incredible to follow Joe. He's such, to me, he's not only, you know, I stopped to follow idols, but Joe is truly beautiful human being in my sense, right? No matter, there's something I'm trying to do. It's just a little physics that keep me apart from you. No matter, there's something I'm trying to do. Break down space and time, and get with you. Be together with you. Be together with you. Be together with you. Thank you. Now back to Joe's stage. Thank you, Joe.
Wait, hang on just a second. I'm going to play a couple of songs that I've been working on, just little snippets of them. Can you pull up? Let's see what we have there. This is a song called Too Much.
There's too much fear and hate. Too much fear and hate.
All right, we kind of lost the important part. It's all right. Just hear a little bit if we can. If not, we'll move on. Yeah. He knows what it is. It's Amanda singing.
Life's roads are rough. You can't always steer straight. The times are tough. There's too much fear and hate. Too much fear and hate. Too much fear and hate. Oh yeah. So much fear and hate. Right on. So much fear and hate. Yes, you got it. Feels so good, but sometimes it brings pain. When that happens, don't try to explain. It's just because love is a many splintered thing.
Okay, those are just a couple of things we're working on. Thank you. All right, we're going to bring the band back up for the last song of the evening. It's a song called When the Night is Dark. It's about someone who's afraid of the daylight. No? Oh, okay. Matt, sorry. Yeah, Matt, my mistake. There we go.
Got us for a bit. By the way, all those songs that Joe does, they are on Spotify, and you can buy them as albums, and you can like stream them and do all kinds of things in your labs whilst you're wanting to be inspired. And maybe if enough of you do that, then he might get like a little bit of royalty money.
Hardly any. Okay.
0.00001 cents or something.
Map of your mind. Map of your mind.
Made a map of your mind. I've charted my course. I'm sailing deep inside. Got the winds of force. Got the heat of your heart to keep me from the cold. Got the currents of will to take me to your soul. On a journey that I've got to take. It's a trip that I knew I'd make. fuel by fire. Made a map of your mind. I've charted my course, sailing deep inside. I've got the winds of force, got the heat of your heart to keep me from the cold, got the currents of will to take me to your soul. I'm just hoping my sails will be true, that they'll take me straight to you. Let the winds blow strong, strong but sweet. Soon I land, our minds will meet. Got the heat of your heart to keep me from the cold. Got the currents of will to take me to your soul. Made a map of your mind, take me to your soul. Made a map of your mind, take me to your soul, to your soul, to your soul, to your soul, to your soul.
Thank you. So, I mentioned the Herzog documentary that's showing at Film Forum starting Friday. That's one of the songs that's in it, as is the next one, When the Night is Dark. Okay, so this is about a guy or somebody who's afraid of the daylight instead of the dark. Got a good fear or threat.
You have an Irishman in the band?
All right.
When the night is dark, I lose my fear. When the night is dark, never shed a tear. Then the daylight comes, it scares me away. When the daylight comes, it's when I have to fade. When the night is dark, when the night is dark, I feel so free, feel so free. When the night is dark, when the night is dark, I feel at peace. When the daylight comes, and the daylight comes, it scares me away, it scares me away. When the daylight comes, when the daylight comes, when I have to fade. dark, when the night is dark, I begin to thrive. When the daylight comes, and the daylight comes, it scares me away. It scares me away. When the daylight comes, it's when I have to fade. When the daylight comes, when the daylight comes, it scares me away. It scares me away. When the daylight comes, when I have to fade.
Thank you. See you tomorrow. Good night.
So one more thing, Joe. So we have a little gift for you. So super small is me and Daniela, and we're the rhythm section of the Amygdaloids. And Daniela asked me to, with the two of us together, to write you a song. So we wrote the song for you it's a super small song called Something to Forget. And this is a super small, it's going to be on our next album for you. And basically, we've never played this before, and all of a sudden I got really nervous. That beard did not work at all. So it was we kind of write what Daniela calls songs are disjointed imagery that tell stories. And this is basically, I had an image of you living upstate where you do in Fossadale, and just thinking back on, you're growing up as a kid and all that kind of stuff. So a lot of these lyrics are kind of a little all over the place, but they came from conversations we've had over the years, going on tour and that kind of thing. By the side of Route 17, and headlights are fading so slowly away. Now I find myself 1,000 miles away. At least I've something to forget. No remorse and no deadbeats and the countdowns to the map of my mind lay the orange shade of the Cajun prairie town. So long since I saw you, the good and the bad. The memories change over time, but my feelings they never have. At least I've something to forget. No remorse and no regrets. Cajun music, boudin and beer. When Liverpool sings, I'm in love. KEU and FM radio. DJ's good, not bad for a rodeo song. At least I've something to forget. No remorse and no regrets.
I said I wasn't going to cry, but that made me cry.
Okay, it's 1 o'clock. Let's get started. Before we start our program for today, we want to take a moment to acknowledge that this is not just a celebration in honor of Joe's amazing career, it's also a celebration of his birthday. Joe is turning 75 today, and in honor of that, we got him a red velvet cake from a fancy Brooklyn bakery. We'll serve this during the break, but Joe, we have to do a little happy birthday, and there are some candles for you to blow out. So if you could come forward, that would be great.
So,
happy birthday to you. Happy birthday to you. Happy birthday, dear Joe. Happy birthday to you.
Okay. All right, we're going to start the first session, and our first speaker leading off is Tony Mobshin. Each of you, for the rest of the session, just come on up and introduce yourself and let us know who you are.
Well, thanks. So I have no slides and no voice, which is a good combination to fill 10 minutes with. And as Liz says, I'm Tony Mobshin. I have been Joe's department chair for much of the last 40-odd years. There was a moment about 50 years ago when we almost worked together. And that's when Joe was with Mike Azanaga. And he was actually briefly interested in vision, which is what I do. And he and Mike came over and discussed doing some experiments that needed to be done on binocular vision in split brain patients, which was a concern of Joe's at the time. They were good experiments, but we never actually did them for reasons I no longer recall. Maybe Joe will remember, but the thing with the way that and the whole enterprise foundered under its own weight, and time passed, and Joe disappeared. And this was when I was in the psychology department, where I was when I joined NYU. And some years later, we started the Center for Neuroscience, and we started hiring faculty. Now, because I do vision, it was obvious that we were going to hire vision people because that was the relevant faculty specialty for the Center for Neuroscience. There were a few others around who had different views. Some of them are still here. Ted reminded me this morning of this, but we had brought in Bob Shapley from Rockefeller, and Lynn Kurpis and Mike Hawken had arrived, and there was a general perception, which I did everything I could to foster, that this was going to be a center for visual science, with neuroscience being an incidental sidebar. Not everyone agreed with that, and so I remember in particular having some disputatious conversations with a colleague of ours from the biology department, who Joe will remember, Gary Aston Jones, who's well known for work on noradrenergic systems and other things. And Gary was one of the members of the center early who agitated for other specialties to be represented. Anyway, time passed, and Gary actually disappeared. He went off to, I forget where he went first, Jefferson Medical College, somewhere. Anyway, he visited a number of places. But around that same time, we recruited our first two non-vision people, one of whom was Joe, the other was Chris Leonard. I still remember that Joe seduced us very carefully by taking a good look at the audience that he was going to be giving his job talk to and choosing a title which was provocative and evocative for us. The title was this. I was thinking of Paul when I saw this title, reminding myself. It's called The Neural Computation of Stimulus Value, right? So how could one resist such a title? You know? And of course that was the framing he used to talk about it. So Joe, who was I think then at Cornell, came and joined us and spent, as you guys have already heard, the next 4+ decades, or 4 decades, I guess it's not 4+ in CNS, but 3+ decades working with us. A couple of years later, I was at the neuroscience meeting, and I had a drink with Gary, and he looked at me in the eye and he said, you know, you just did that to spite me. then you would never have hired Joe, right, had I not left. Right? And I said, well, you can, however you want to frame it, you can do that. Anyway, so I don't, unfortunately, have any really juicy stories about Joe, nor do I have slides showing him in embarrassing positions. That was not for lack of looking around through my archives, but it was simply a representation of the fact that it's extremely difficult to search images if you look for LeDoux embarrassing pictures. Right? Nothing comes up, or very little comes up. But Joe has been an extraordinary colleague. I did remind myself while looking to see when the last time he worked on split brain patients was, that he has an astounding number of publications and citations and H indices and things of that sort. Puts the rest of us to shame. But he has been, the founder and root of what was really the second great piece of the Center for Neuroscience, which is the piece that studies learning, memory, and emotion. And of course, many people in this room and elsewhere followed him in through that door that he opened, and I think the entire enterprise has been much richer for it. It's been a privilege to be a part of the universe in which he inhabits. It is, I realized as I went looking back that Joe is one of those faculty who manages, by coating with Teflon, to sort of slide through life without actually ever really getting hammered by the duties of a faculty member. So I look around and I can say with safety that Joe is one of the faculty who has spent the longest in CNS and has managed to do the fewest numbers of jobs. And it's not because he doesn't do them when he's asked. It's just that because you think, oh, well, we're not going to ask Joe to do that because ... whatever reason it is you want. So there are those who serve, and there are those who watch, and there are those who do other things. And Joe, of course, made us all famous by becoming who he is and becoming the public intellectual that he is. And I think we have always been happy to salute him for that, as I'm sure all of you guys have in the room who have worked with him and learned from him. So I don't have anything more to add. I'm going to use what remains of my voice to offer my thanks to Joe for hanging out with us for all these years, and to express my pleasure and my honor at having been part of that enterprise. So thank you.
Thank you, Tony, for this great introduction. So my name is Rodrigo Triana del Rio. I'm the last postdoc at the LeDoux Lab, and I'm going to tell some stories about the last batch of scientists and sprinkle with some science, fun science that we did. I would like to start with a story about the first time I had a disagreement with Joe. This was about Mexican foods. So when I came to the lab in 2019, I was very pleased to know that the favorite food for celebration was Mexican food. And then Joe invited us to his beautiful flat in Williamsburg, and over some margaritas, he said, hey, Rodrigo, come and get some chimichangas on the table. And then I came and I saw and I said, these are not chimichangas. These are actually called picadas. And then Joe didn't know that I'm from this region in Mexico called Veracruz. You can see the map here, over there in the Mexican Gulf. And it's where these appetizers are coming from. So we went on a bit back and forth on the terminology. And what I can say is that Joe is somebody that is always open for discussion, open for conversation, and open to change his mind as well, right? We don't talk about fear conditioning anymore. So as a person, as a leader, as a mentor, Joe has touched our lives as somebody that cares about people, that cares about our well-being. He was very important for us during COVID. We faced some challenges far from our families. Joe is somebody that cares about us, that cares about being with our family, having a good time. I got my no self, no fear from the amygdaloids. So that is as a person. There are some people from this last batch, like Javi, maybe Christiana is over there, and of course with Claudia and Mian, we had a lot of fun. And I'm going to talk a little bit about the science that we did. Of course, with Joe, we were interested about disentangling these circuits on how to cope with traumatic memories. And my main question was about how it's possible that some populations, when faced with traumatic memories, are overwhelmed by this fear. And in red rodents, we can see they are freezing, they cannot move, they reactivate these threat memories, and they cannot do anything with them. Whereas other populations, we see in patients or in rodents, they are proactively coping and proactively avoiding the threats. And we want to know why some people or populations would work on one way or in the other. And Joe, you know, he has a very bright scientific intuition. He told me, Rodrigo, you need to explore plasticity. This is like the central point. So we started doing some experiments, some behavioral experiments, doing this signal active avoidance paradigm that many of you are familiar with, in which rodents, they are conditioned to an auditory tone that is going to predict the food shock, right? But in this test, animals, they have to jump to the safe side, and then they're going to learn to proactively avoid the threat. We were following some functional connectivity studies from Rob Sears, Chris Kane, that show that the connections from the basal amygdala to the nucleus accumbens core, that in humans is the ventral striatum, are important for this kind of behaviors, proactive threat coping or signal active avoidance. But my question was more about the acquisition of the behavior and how we can modulate the circuit with different pharmacology, for example, cannabinoid pharmacology. We used some viruses, chemogenetic viruses, optogenetics, to inhibit and stimulate this neuronal projection from the basal amygdala to specifically the core area of the nucleus accumbens. And by doing chemogenetic inhibition during the test, I don't know if you can see my cursor, we inhibited the acquisition of the behavior. So animals, they didn't learn to jump, they didn't learn to avoid the threat. Nevertheless, when we had the chemogenetic inhibition, but at the same time, we infused a cannabinoid receptor agonist to stimulate the cannabinoid-dependent plasticity in the nucleus accumbens, animals, they were able to learn. And then we would ask, thanks to Mian, shout out to Mian, that has been so supportive for so many years in the lab, we decided to go through some slice physiology on something that we had never explored before in the LeDoux lab, that is long-term depression, and specifically cannabinoid-dependent long-term depression in the nucleus accumbens. And following the same techniques that we did in behavior to inhibit the projection with chemogenetics, we stimulated in the nucleus accumbens to see if this, you can see in dark blue, kind of purple, the normal LTD or long-term depression. But what happens when we block the projection from the amygdala to the nucleus accumbens? In slices, we impair LTD. And then if we stimulate the cannabinoid receptor, the CB1 receptors in this area, we mimic LTD even in absence of electrical stimulation, showcasing this relevant role of cannabinoids to induce plasticity in an area so relevant for active avoidance. And we could block this as well with cannabinoid receptor antagonists, as we did with the behavior. Then shout out to Claudia. Thank you very much for Claudia, that is helping, I'm sure, many of you as well during so many years with her magic on getting the most beautiful imaging, going through electron microscopy, confocal microscopy. And then with Claudia, we were able to disentangle even more the circuits where these receptors are located in this neurocircuit framework of the amygdala and the nucleus accumbens, looking at the specific cellular populations like the parvalbumin interneurons, like any other interneuron in the network. And then having this knowledge on how the target engagement of cannabinoid receptors could help to modulate networks for traumatic memories, I was inspired to make the change and go on the more clinical side. And now I'm working on the medical center, working with Esther Blessing, that is over there. And then we started to follow this question on how to cope with traumatic memories using the target engagement of cannabinoids. And now, as part of the psychiatry cannabidiol research at the medical center, we are working on clinical trials to explore on a large scale how cannabinoid treatments can modulate the threat circuits and can help to alleviate symptoms related to the PTSD disorder. And on a different level, we want to explore how these targets, they are relevant for neuropsychiatric symptoms in Alzheimer's disease, and these related to physiological markers like sleep, temperature, and Alzheimer's disease biomarkers, and how we can put all these together. And I hope Joyce is proud of how his very similar and pioneering ideas about traumatic memories, about threat conditioning, can touch so many lives when we bring them to the clinics. And then one example of this is how helping elderly populations on experiencing, on treating traumatic memories. And to close my story, I'm talking about Veracruz, my region. Next year, I will go to visit my grandma that is turning 100 years. So I hope that all the research that I do will help people like her. And on a final note, I wanted to have like a musical note, but I'm terrible at this. I suck at anything musical. But last night, I asked my sister that dance. She's my sister. She dances folk Mexican music. And this is a snippet of folk dance called The Witch, La Bruja, in which the singer has a glass full of water. And I'm going to play it for you. All right, so this is just to finish. Next time you go to Mexico, you go to Veracruz instead of going to Cancun. And thank you very much, Joe, for the opportunity of meeting you and being part of your team, and all this last batch of scientists that you have inspired so profoundly. We wish you the best in this new chapter in your life. Thank you very much.
Okay, great. So, tough act to follow. So I'm Megan Peters. I'm a faculty member in the Department of Cognitive Sciences at UC Irvine, all the way on the other side of the country. And so today I'm going to talk a little bit about, I think, why I'm here, and my gratitude to Joe for inspiring both the work that I'm doing right now and that I plan to continue doing for a long time, and also for inspiring the kind of scientist that I want to be in terms of his commitment to his ideas and his ideals. Okay, so why am I here? I don't work at NYU. I have never officially worked with Joe. But when you get invited to speak at something like this, you absolutely show up, especially for someone like Joe, who has inspired so much of my work on subjective experience. So not just the fear circuitry, but the idea of fear being a subjective experience that we have. And I've always been, as far back as I can really remember in my academic career, been interested in studying subjective experience. And so when I went to graduate school, I thought that the way to get at the subjective experiences that we have of our sights, our smells, the sounds that we have, that we experience in the world, would be to just study perception and to get at how the brain is able to engage in perceptual decisions and understand the world around it. So I'm a West Coast girl, and so I stayed on the West Coast for graduate school, and I ended up doing my PhD at UCLA with LeDoux Shams, studying multisensory integration, how the brain knits together different sensory signals into this coherent, conscious, subjective experience. We didn't quite get all the way to the subjective experience part, unsurprisingly. And so I was very fortunate then, as I ended my PhD, that I was approached by Hak-Wan Lau, who was moving to UCLA from the New York area. And he said, do you want to come be my postdoc? And we'll work on subjective experience and metacognition. And I said, great, where do I sign? So I then worked on metacognition with Hak-Wan, and we heard from Hak-Wan yesterday about his deep relationship with Joe and with also the New York music scene and New York in general. And so what I got out of working with Hak-Wan was not just a great scientific mentor, of course, but a connection to all the folks here in New York who are really taking subjective experience seriously. And several of them are going to be speaking directly after me, and some of them were hanging around here or are hanging around here. And this includes, of course, Joe. And so in addition to being a giant in the field, the founder of the field of fear and anxiety and what does the amygdala do, what is the fear circuit, Joe is also a pioneer in understanding the differences between the activation of a circuit in the brain and the experience that goes along with the activation of that circuit. And I don't think it's going to be a surprise to anyone in this room that subjective experience is not synonymous with activations in neural circuitry. And this is a really important thing for us to drive home, right? So when I show you this red apple, we can measure all the activations in your brain, the retina, the LGN, V1, V4, blah, blah, blah. But, how is it that these are accompanied by the subjective experience of something as simple as red? Maybe the PFC is involved, I don't know. But, we don't really know how this works. And the difference between the brain activations and the subjective experience that accompanies them is something that we all, I think, know how important this is. But it's not something that when you go into the broader neuroscience field or the artificial intelligence field is often talked about, at least not as much as it should. So it's precisely this separation that we need to make people aware of. And so when I got the chance this last year to contribute to the so-called Mysteries Day at the end of NeuroMatch's new Neuro AI course, where we covered consciousness and neuroethics. And by the way, for those of you who don't know, NeuroMatch it's not a university, it's an online summer school that we founded in 2020, and we've got a huge reach. And so this was a fantastic opportunity for us to educate people about the differences between subjective experience and activations of brains or activations of nodes in a neural network. ChatGPT does not have experiences. So, of course, when I got the opportunity to lead this Mysteries Day, one of the first people I called was Joe. And I said, hey, Joe, would you be willing to contribute just a short little video snippet, which I won't play for you today, but I would encourage you to go and have a look, a short video snippet on the differences between the behavioral signatures of fear and the neural activations that drive those behavioral signatures and the feeling of fear, the subjective experience of anxiety, of freaking out, because they're not the same thing. And of course, Joe, wrote back basically immediately and said, yes, absolutely, and sent me a lovely video. And so I'll just show you a screenshot here. And this video now and this course has been seen by just over 500 students in this summer alone, and will continue to grow and to influence folks. And so thank you, Joe, for contributing to teaching the rest of the world about the differences between subjective experience and neural activity in such an interactive and accessible way. So, fear is not synonymous with activation in neural circuitry. And so if we look at the neural circuits that are involved, of course, like I'm not a emotional neuroscientist, so well, maybe I'm an emotional neuroscientist, but I don't study emotion, and so I'm not going to go into the neural circuitry here. So I go and do things like I ask neurosynth, where is the area of the brain that is associated with fear that creates the experience of fear, and you get something that looks like this, which is some blobs in the amygdala. And then you can do something even lazier, which is what all the kids are doing these days, which is just to ask AI, how does the brain create the experience of fear? And you get this nice list of, there's a fear circuit activation, and there's a role for the thalamus, and the amygdala does something, and so does the hippocampus, and so on. And of course, we have a list that looks like this, to the extent that this is correct or not, in large part thanks to Joe. So we know the answer to this. But you'll notice that there's like kind of this throwaway thing that the AI does, which is like, oh, the prefrontal cortex evaluates the threat, and then somehow the insula integrates sensory and emotional information and creates the feeling. And I don't know that that is really a satisfying answer here. How is it that this part works? How does that work? What is the difference between the activations of neural circuitry and the subjective experience? And this explanatory gap is big, and it's unsatisfying, and the consequences of glossing over it can be really dire, as Joe has really pushed on, that we shouldn't say you're not experiencing fear just because your brain isn't making the right pattern. If you say that you're scared, I should believe you. I should believe you when you report on your subjective experiences, even if your brain's not doing the right thing. So the way that this has inspired me to work in my own work is to change the question a little bit from how is it that subjective experiences are created, because I don't know how we're going to get there. But instead, I'm trying to build quantitative ways to characterize that subjective experience, to not just say, how do you feel, and write it down, but to understand the relationships between the subjective experiences that we report and the underlying neural circuitry and the behavior that goes along with it, but isn't the subjective experience itself. We cannot choose just behavioral capacity to index subjective experience. So blindsight, which I think we all know is a thing, the idea that you can perform adequately on some visual task and yet report no visual experiences whatsoever. Behavioral capacity is not the same as subjective experience. Behavioral performance, any neural activity, again, not the same thing. So we have to ask people. And we can't ask rats, but we can ask people. And so we're going to use introspection and self-report. And there are a lot of challenges that go along with this. So self-report and introspection is unreliable in general. We know that from Nisbet and Wilson, we can see that subjects are sometimes unaware of the existence of a stimulus that importantly influenced their response. So the response is there, but the awareness is not. It's an unreliable indicator of cognitive and neural processing in general. Blindsight is a thing. We also have cases where people say, I'm really subjectively feeling like I'm doing super great on this task, and they're just absolutely garbage at the task. So it's also not objectively verifiable. If you tell me that you're feeling fear, if you tell me that you're seeing a Gabor patch at a very certain specific intensity level, I just have to take you at your word. But that's what we're dealing with here. And importantly, then, subjective experience also co-varies, typically with a lot of non-subjective experience stuff, like capacity to actually perform some task on the basis of an external input. So my approach has been to make these strengths, not weaknesses, and to basically do psychophysics on introspection. And what that looks like, it's part of a broader research program that's been called for in philosophy and neuroscience recently. And this is my contribution now to Joe's special issue in cerebral cortex, is to talk about the ways in which we can quantitatively characterize subjective experience so that we can then start to get over that explanatory gap maybe a little bit more. And so what this looks like is, to take, typically we would do psychophysics on the relationship between stimulus and the internal representation of that stimulus, the type 1 kind of decision or estimate. But I want to do psychophysics on the metacognitive evaluation of that. On the relationship between the experience that you're having of doing a task so that I can control for all of the stuff that isn't the subjective experience part. Okay. All right, quick kind of shameless plug here is that we're building kind of methodologies to do this. We have a little toolbox if this sounds kind of cool to you. But the punchline is that what we're going to get out of this is a way to understand the functions associated with and facilitated by subjective experience, including their neural correlates, so that maybe we can start to bridge that gap between the brain and the mind. And this work is supported by a project that Joe helped create. And so at a workshop back before the pandemic here in New York, I was fortunate to be in the same room with Joe where we were talking about exactly these kinds of questions and how we could design tasks and psychophysical approaches to get at them. And Joe, you were very inspiring in that workshop, and it was my great pleasure to get to know you there. So a lot of this work that I've just told you about now is really, I think, inspired by Joe. So this is, thank you, Joe, for helping me take one more step along this ladder so that, eventually maybe I will get to the answer of how is it that these things are accompanied by experiences. And the last thing that I'll say then before closing is that Joe is not only scientifically inspiring to me, he's also inspiring as a scientist. And so I've also had the great pleasure and honor of getting to interact with Joe through an organization called the Association for the Scientific Study of Consciousness, where Joe is the now past president. And so therefore, I, as a board member there, had the opportunity to get to talk with him about running organizations that are about the things that I have just delivered this talk about, subjective experiences. And it was through those interactions and through that time period that I also got to work with Joe on another project where it was very clear to me that Joe really sticks to his convictions about a great many things. And this is the project. This created a huge amount of uproar, but it was Joe was such a rock as we were doing this very kind of ballsy thing. And I'm very grateful to Joe for your mentorship and for your stability and for your enthusiasm in bringing this to fruition. So that's it. Gratitude, just like fear and just like any other subjective experience, is not synonymous with activation of neural circuitry. Neurosynth has no idea what the neural circuits of gratitude are. But you should believe me when I tell you that I am experiencing gratitude right now. So this is just a marker then, too, that I carry this around here. Thank you for giving me your book, Joe, and I remember very keenly being in your office and feeling just so in awe of being able to be in your presence and get to learn from you. So thank you very much.
I'm not.
Sorry, I can just close this. If I close this, I'll grab yours.
Sorry.
No, it's okay. This is you, right?
Oh, good.
Yeah.
Thank you very much. I'm sorry, I need help from somebody. How do I get this?
Yeah, no problem. It's right there.
I don't do Macs.
And just hit this arrow right here, it advance, okay?
Terrific.
And your notes, if you have any, right here.
Sorry, I'm David Rosenthal. It's been a huge pleasure to interact with Joe over a number of years now, having to do with our overlapping interests and overlapping theories about consciousness, and that's what I'll talk about today. Joe's game-changing work on neurological survival circuits drove an important wedge between conscious emotions and behavior that's independent of conscious mentality. As Joe has put it, behavior is not, as we commonly suppose, primarily a tool of the mind. This is a crucial result. And Joe adopts what's come to be called a higher-order theory of mental states being conscious, which nicely accommodates unconscious function. As Joe notes, a state is conscious on a higher-order theory if an individual is aware in a suitable way of being in that state. That reflects the common sense observation that no state of which one is not subjectively aware is a conscious state. Joe describes his higher-order theory as a modified version. It involves hierarchical layers of neural processing, which I won't be talking about, but it also sees consciousness, as he puts it, through the lens of memory, and it's the connection with memory that I want to focus on this afternoon. Joe adapts Endel Tulving's taxonomy of memory types. He adapts it. It's not exactly the way Tulving had it, I think. And Joe urges that those memory types correspond to different types of higher order machinery that that result in various types of mental states being conscious. This is a very exciting and inviting proposal, but I want to suggest that there is a possible tension between the way Joe does appeal to Tulving's taxonomy of memory and a higher order theory. So, some about a higher order theory first. A theory of consciousness can help isolate various neural correlates, the so-called neural correlates of consciousness. Higher order theories, of course, as is well known, point to frontal regions. But consciousness itself is a psychological phenomenon. A state's being conscious is a psychological property of that state. So the ultimate test for any theory of consciousness, I think, must be whether it explains what it takes for a state to become conscious, and explains that in terms that are distinctively psychological. Just as we need to know how the physical makeup of objects results in observable macroscopic properties, so we also need to explain how the psychological property of a state's being conscious comes about. And to explain that, we have to begin by being clear about exactly what it is for a state to be conscious, again, of course, in psychological terms. Consciousness is mental appearance. Consciousness consists in the way one's mental life subjectively appears to one. A state's being conscious is simply its appearing subjectively to one. Each higher order state is an awareness of oneself as being in a first order mental state. So a suitable higher order awareness actually constitutes the subjective appearance of one's being in some mental state. And higher order theories explain that in psychological terms. They explain a first order state's being conscious by appeal to a higher order state's making one aware of that state. What then are the first order states that one is aware of being in? Suppose, for example, one experiences conscious fear. What is the first order state that one is aware of being in? It's of course not the activation of survival circuits, though that may trigger a first order mental state. The higher order awareness makes it subjectively appear to one that one is in a first order mental state, a state with particular mental properties. The first order state, then, is whatever state exhibits roughly those mental properties. And we know that the activation of survival circuits does not do that. It does not exhibit those mental properties. So we must first fix the various mental properties of first order states that can become conscious, and then we can slot in the relevant properties into the content of a higher order state that makes one aware of being in that first order state. I'll skip those two. The higher order states never exhibit the properties of the first order mental states. Higher-order states do not replicate or repeat the first-order states. A higher-order states having first-order metal properties would do nothing to explain why a first-order state that has those properties is conscious. The higher-order state, then, is simply a generic subjective awareness of one's being in some first-order metal state, whose content, then, is in each case filled out by the metal properties of the relevant first-order state. And that's all there is to a higher-order state, because that's all that's needed to explain, in psychological terms, why the higher-order state constitutes a first-order state's being conscious. So there is only one type of higher-order state, no matter what type of first-order state is conscious. By contrast, on Joe's appeal to memory, each of Tulving's 3 types of memory delivers a different type of higher-order state, which in turn figures in making a different type of first-order state conscious. So in this useful summary, in an article of Joe's with Hak-Wan Lau, there are 3 types of higher-order state: anoetic memory higher-order states for first-order sensory states, noetic memory higher-order states for first-order semantic states, and autonoetic higher-order states for first-order states of episodic memory. But the very same type of higher-order state, I have been arguing, results in both sensory and semantic states being conscious, and indeed mental states of any other type. Also, all higher-order awarenesses are, in effect, what I think Tulving would call autonoetic, since all higher-order awarenesses have their content involving the self. Each higher-order awareness makes one aware of oneself as being in some first-order state. And I think that this is crucial, because if one were aware of some first-order state but not aware of it as a state of oneself, it wouldn't be a state that one subjectively appears to be in, so it would not be a conscious state at all. Still, the neural mechanisms that subserve Tulving's 3 types of memory may very well figure, as Joe urges, in the way various higher-order states come about. But even if those neural mechanisms do involve those types of memory, that would not result, I think, in distinct types of higher-order state. To recapitulate, because consciousness is itself a psychological phenomenon, no theory is acceptable unless it explains, in psychological terms, how some mental state ends up being conscious. The goal of explaining just how conscious states differ from unconscious mental states dictates what type of psychological machinery is required. And a single type of generic higher-order state, with the relevant first-order mental properties slotted in, is all that's needed. Neural mechanisms can then explain why such higher-order psychological states occur. And that may at bottom be exactly what Joe has in mind. I will be eager to learn from Joe either way. Joe's penetrating and remarkably prolific work has been making highly important and fruitful advances in the way we understand and explain consciousness. I look forward to much more exciting work from Joe about all of these topics in the years to come. Thank you very much.
All right, so hello, my name is Richard Brown. It's a great privilege and honor to be here. I've been having so much fun. All the talks have been inspiring, humbling, and just really a real reflection, I think, of Joe's contribution. So it's been great to be a part of this. So my connection with Joe is a little bit roundabout. So I'm going to set some context first before diving into some of the more penetrating remarks that I have. So I first came to New York as a plucky young graduate student back in 2003. This is my then girlfriend, Jennifer, who now is my wife of 15 years. We just had our 15-year anniversary. So I came to study consciousness. I was interested in neuroscience. And I fell in with a group of graduate students who also played music. We cheekily called ourselves the neural correlates of David Chalmers, or NCDC for short. And we would get together, semi-regularly in various basements and rehearsal studios across Brooklyn and Manhattan to play our music, as the case may be. In 2007, 1 year before I defended my dissertation, I actually was lucky enough to land a job at LaGuardia Community College, where I still teach to this day. And I thought to myself, well, I didn't get out of New York, but I landed a job in New York, but it's a great place to be a fly on the wall to observe these titans and to have a front row seat for the battle of consciousness. And I thought to myself, this is a great place to be. Now, in my spare time, I still had people over to my home for the various jam sessions. So here's Hak-Wan Lau playing the guitar, and a couple of our compatriots. This is, I think, coming from 2009 or something at that point. And I eventually started realizing that there was enough critical mass that perhaps we should try to get out of the various basements and bedrooms that we were frequenting and take our show into the big stage, so to speak. So I coined the term the New York Consciousness Collective, which was supposed to represent a vast group of people who were interested in consciousness living in New York City, but who also played music. And I contacted lots of local venues around New York City, one of them being the Parkside Lounge in Manhattan, and they let us host a jam session there every Monday night for about three and a half years, almost 4 years actually. So here's a bunch of philosophers. That's me on the drums, look, wearing, I think, pretty much the same tie. Well, no, it's a different tie. Okay, good. So we would play there every Monday night, and eventually, out of this jam session where it was just all comers were welcome, anyone who could play an instrument could show up and just take the stage. Well, we eventually started forming some groups. And one of those groups involved Hak-Wan and Steve Fleming. You can't see the picture very well in this lighting. Trust me, it's a fantastic image, though, of Steve Fleming and Hak-Wan and myself performing. This was actually when Hak-Wan was leaving New York back in many, many years ago. Anyway, so the point was I started thinking to myself, it might be nice to not organize a jam session, but to actually try to organize some kind of performance with bands, because we were starting to form more localized groups. It was at that point that someone said to me, you know, there's a neuroscientist at NYU named Joe LeDoux who has a band called the Amygdaloids. I was unaware of that, but I immediately thought to myself, yeah, it would be a good idea to email this gentleman. And so out of the blue, I emailed Joe and I introduced myself. I said, would you like to come and be a part of this thing which I'm trying to organize, which at the time was a going away party for Dave over here, Dave Chalmers. He was visiting NYU at that point. They were trying to entice him to come to New York City, which they eventually did. And of course, Joe, without knowing me and without batting an eye, responded that sounded like a great idea. And thus was born what I call the Qualia Fest, which was a musical event showcasing the musical stylings of various philosophers and neuroscientists. So this is from the first Qualia Fest that we had so long ago. And true to Joe's nature, very soon after that, I saw this article come out in Trends in Cognitive Science, The Flip Side, Scientists Who Rock. And I thought, gee, that's interesting. And Joe was talking about his relation to music and especially the way he thinks it makes him better at everything else he does, which I agree, by the way. And I was reading this article, and lo and behold, I came across this passage here where he mentioned me by my name and said, Richard Brown, this philosopher. And I was like, well, that's the first time anyone's referred to me as a philosopher, so that's nice. But also, it was the first time I saw my name in a journal like Trends in Cognitive Science, which I had been reading, obviously, for a very long time, and it was kind of a big moment moment for me to see my name in that journal mentioned by someone who I respected as a neuroscientist and a musician. And this is emblematic, I think, of the way Joe approaches these things. He's inclusive, and he wants to give a leg up to all of those people who are around him. And so I found myself to be the beneficiary of that. Now, we had several more of these Qualia Fest. So here's a picture from the one that we held at the Bowery Electric. And as I was, this was in 2012, so I think this is the last one that I organized before I had kids and moved out of the city myself and settled down out on Long Island where things are much quieter. But lo and behold, I was approached by a reporter for the New York Times, Ariel Kamener. I think that's how you say her name. And she said, I'm writing a story about the Qualia Fest in the New York Times. And I said, how did you find out about this thing that we're doing over there? And she said, well, Joe LeDoux told me this is a place where scientists and philosophers get together and talk. And in fact, that's what it says down here. I don't know if you can see. It says philosophers and neuroscientists communicate at a yearly music festival. And so this is, again, a way in which I see Joe trying to include everyone and give them a leg up, pointing the finger, bringing attention to all those who are around them. And myself, being a lowly community college professor, I was quite humbled and honored that someone would think to mention that what we were doing in the New York Times. So that was kind of a big moment. We had several other interactions after that, but then there came this time in 2015 where he came out to the CUNY Cognitive Science Colloquium, which was at that point organized by David Rosenthal. And he gave his talk there. And I convinced him to come out afterwards and to play music at the local rehearsal studio that we go to. And so he became one of only 3 people in the entire history of that speaker series to give a 2-hour talk, followed by 1 hour of discussion at the local pub, and then an all-night jam session after that. So that's a very distinct and small discerning club of people who have that level of enthusiasm and engagement with their craft. So that night was a very inspirational night for me. We were out all night arguing, talking about consciousness. His book, Anxious, was just about to come out, and so he was thinking about these issues, and we're just having a great time talking about this stuff. And then lo and behold, I got this email a couple of days later, which said, oh, hey, Richard, I was thinking about maybe asking you to write a paper with me, but I think maybe we disagree too much, so maybe it's not a good idea. And I thought to myself, something's gone wrong here, because as philosophers, we like to play the devil's advocate. And I wasn't necessarily disagreeing, but I was trying to figure out what your position was. And we worked things out. Everything turned out all right. And so we wrote this paper together, Higher Order Theory of Emotional Consciousness. And we've had a couple of others that have come out as well, especially this one in Trends in Cognitive Science with Hak-Wan Lau and Joe. Still some of my proudest academic achievements to this day. And working with Joe was a tremendous honor. Anybody who knows him knows that he has a deep desire to really understand these issues. He's not superficial. He doesn't want to just toss something out, but it took 3 years basically of intense discussions, emails, meetings, drunken arguments, you name it, before we could hash out something that we thought of as presentable. And so having said all that then, by way of which I come to understand our relationship, I want to say that as a philosopher, we pay homage to those that we respect and admire by disagreeing with them. So now the Love Fest will end, and I'll make a few comments about why I think that some of the things that he says could be improved in slight ways. So I want to make kind of 3 points, and I'm going to go through these quickly. This is part of the contribution that I made to the special issue of cerebral cortex. So one, I want to ask, what is the target for a theory of consciousness? Second, I want to ask, what is it that's higher order about this pre-conscious content of the present moment mental model that Joe talks about in his multi-state hierarchical view? And then finally, I want to ask the question about whether the prefrontal cortex is necessary for phenomenal consciousness. So my own particular view on these things is that phenomenal consciousness is the appropriate target for a scientific theory of consciousness. What we want is a scientific theory of phenomenal consciousness, which is hopefully familiar from the literature. This is the notion that there's something that it's like from our subjective point of view when we encounter the world. And sometimes people talk as though this is a spooky, extra-scientific, non-physicalistic part of the world. And there are people who have that view. Maybe some of them them are in this room currently. But it's not foisted on us by the concept of phenomenal consciousness in and of itself. I myself consider myself a physicalist. I think phenomenal consciousness can be explained in purely naturalistic physical terms, and I don't think that there's any reason to back away from that claim and to say that that's not what we're trying to explain. So in his more recent work, Joe has said things like maybe this is more akin to the notion of a soul and science can do without it. And I want to push back against that way of thinking about phenomenal consciousness in the strongest possible terms. This is what we want from a theory of consciousness, and if we turn away from it, then I don't really think we're discussing consciousness in the sense that matters any further. So how could something like this be a component of the physical world? Well, one popular strategy is to connect it with representations of the right sort, popular in neuroscience and popular in philosophy. Not to say that it's correct, but it certainly is possible that it could be correct. And my own work has largely centered on trying to give a representational account of phenomenal consciousness from the higher order perspective. This is something that I call the higher order representation of a representation view, largely inspired by the work of David Rosenthal and other higher order theorists. I was a student of his, taking classes of his at the Graduate Center. Seeing those slides with the yellow font was bringing some flashbacks to me a moment ago. So my view is that what we want is a representational theory of phenomenal consciousness, but just from the higher order perspective. And so instead of thinking of the higher order state as making some other state conscious, I think that just as with other representational theories, the state which is conscious is the higher order state itself. So I have a book that's coming out on this in early next year, Consciousness as Representing One's Mind. I don't recommend that you read it, but I just wanted to say that it is coming out. And I try to sort through a lot of these details in that book. So that's very exciting. It's kind of a huge task. So very quickly, what do we mean when we talk about these different kinds of representations? Well, first order representations, as I understand them, are mental states which represent the world. They represent things that are external to the mind, whereas higher order states, as we typically use them in philosophy, are mental states that represent one's own mind. So that this distinction is over what the content is. And there's a kind of tendency in the literature to naively try to map these, as David was saying, psychological constructs to the neurological level by sort of saying, okay, so the higher order states, maybe they're in the prefrontal cortex, and the first order states, perhaps they're in the sensory cortices. So maybe if you're seeing a red apple, maybe the red's in V4 and the appleness is is somewhere in the temporal lobe. You could ask Megan, she was talking about that earlier. So I think that that's not really the right way to think about these kind of states. And one of the things that I was struck by Joe's own work was that he seemed to be suggesting to me a view where the first order state, the state of which we are aware, which is the representation of the world, would itself be found in the prefrontal cortex in an area that was anatomically higher order. And I thought that that was a very interesting sort of view. And that's the way I approach things in the papers that we wrote. But what I've noticed is that recently he seems to have backed away a little bit from that way of thinking. He has sometimes referred to his view as a ho-ho horror view, which is a terrible pun that's due to Matthias, and I wish people had more self-control about these kinds of puns, but it's difficult when you're in this line of work. So I understand the impulse. However, the ho-ho is supposed to stand for higher order theory, because Joe has often talked about this state in the prefrontal cortex as another higher order state, which gets re-represented by a still further higher order state. And we used to argue about this a lot. I have many, many email exchanges, and luckily no video recordings of drunken rants between us. But there's a lot of time that we spent trying to figure out why is this state higher order on his view. And I don't think we really made any progress on this until I read his recent book, The Four Realms of Existence. And then it kind of clicked to me. I was like, okay, so this is where our disagreement really is. And the reason being is that the way that Joe defines what a mental state is in terms of whether there's a mental model. So if there's a mental model, then the state is a mental state, and thus the activity in various parts of the brain at what he calls the neurobiological level are not themselves truly mental states. And if this state that's in the prefrontal cortex is re-representing neurobiological level phenomena, then it doesn't strictly count as a higher order state in the philosophical sense because it's not representing a mental state. It's representing something in the brain, but it's not representing something in the mind, according to the way I understand Joe's position to be. So it does seem to me that the way I first interpreted his view is the correct way to interpret it, namely that the higher order state, which he calls the higher order state, is really just the first order state. It's just in an anatomically higher order area of the brain, which is somewhat surprising that you would find a representation of the world perhaps in the prefrontal cortex. All right, so in the last couple of seconds here, I just wanted to say something about the prefrontal cortex because there's a growing tendency in the literature to identify higher order theories with theories that are committed to the prefrontal cortex being importantly involved in consciousness. And this seems to me to be a mistake. So there are reasons for looking to the prefrontal cortex, that is for sure, but it seems to me that it depends on how you're thinking of this kind of inner representation, this representation of one's own mind. So if one thinks of it as a kind of self-consciousness, then one should expect that kind of activity in areas related to processing about the self, perhaps parietal cortex areas surrounding that area. If one thinks of it as a kind of metacognition, then perhaps one would expect to find it in places like the dorsolateral prefrontal cortex or these other areas. I tend to think that Joe thinks there's anatomical reasons for looking at the prefrontal cortex, a kind of all roads lead there sort of view on which the most abstract higher order representations would be found in something like that. But it doesn't seem that any of these things are a direct result of higher order theorizing itself, as instead they are a result of theorists who have a particular interpretation of what this kind of inner awareness is going to amount to. So I view these as empirical conjectures about where we might find these things, but I don't view them as commitments of the theory itself. And I think we're a long way from being able to say whether or not a bee, a crab, a dog, or an octopus can have the kinds of higher order states that the theory like this proposes. My own view is that I think that's an empirical possibility, but we wouldn't be able to answer that kind of question until we knew more about what we were looking for. And that really is the challenge, I feel, of where we're at right now, is trying to say, well, how can we get this beyond this naive kind of hierarchical view in the brain and thinking lower order and higher order mean the same thing in both neuroscience and philosophy. So I'll end my discussion there. There's a lot more that could be said, but I want to end by saying thank you, Joe. You have been a tremendous inspiration to me, someone who I value interacting with, working with, someone who talks to philosophers, neuroscientists, anyone at all, takes them seriously, tries to promote their work, and make them feel as though they've made an important contribution to the discussion. And that is a rare quality, which I wish we found more often. So thank you.
Cake. I'll just ask if you have presentations later in the afternoon, if you could come up and we'll load them on here. If you sent it to me within the last half hour, I did not get it. I don't have Wi-Fi here.
All right, let's get started. Hope you all enjoyed some red velvet cake in honor of Joe. So this is the second session of the afternoon. Again, we're not going to introduce speakers, so just please come up, look at the order right now, know who you're following, and please come up when it's your time. And we're going to start with Stefan Hoffman.
Thank you. Thanks for coming back. Tough to compete against the cake outside. So I wonder what this sugar rush is going to do to you while sitting here. So it's, I guess, dopamine moderated listening to what I'm doing here. So you know, when you put in a neurotree, Joseph LeDoux. This is it's impossible to put it on one screen, by the way. This tree is way too big. But there, you see on the right here, I circled Joe LeDoux at NYU, and then obviously Mike Gazzaniga is in the left here. And you actually, I'm gonna zoom in. So zooming in, you actually are 1, 2, 3 degrees of separation from Ivan Pavlov. It was interesting little tidbit. Joe, you've been enormously influential. And I think this is also not taking into account the very fact that this is obviously primarily focusing on neuroscientists, but you have had an enormous impact in clinical psychology. So my name is Stefan Hoffman. I forgot to say that. I'm a clinical scientist, and I study therapy. I develop treatment approaches for anxiety disorders, and I'm always sort of at the fringes of my field, trying to figure out how to improve therapies using neuroscientific approaches. And obviously, you can't get around Joe LeDoux, and he's been sort of a larger-than-life figure for clinical scientists, certainly. I got to know Joe. Actually, we were Facebook friends. And at one point, when he was, I believe, writing his book "Anxious," he had a few paragraphs on CBT, I believe, in psychotherapy. Is this correct? Can you take a look at that? And so I did, and we started communicating over these issues in quite some detail. And just as typical as in a Joe LeDoux manner, he kind of asked really uncomfortable but really important questions that really got me going back to old literature and new literature and trying to back up all these issues that he doesn't take simple explanations for an answer, but rather he really is drilling in, he's digging deep. And it's not only the drilling deep part that I appreciate, but also the big vision that he has. That is, he's not staying in this little field, but he's bridging way beyond his field into philosophy, as we heard, into even clinical science. And over the years, we've become very good friends. He and I, we did even workshops together. Remember the one in Italy? I think we had like a two-part workshop talking to the amygdala and listening to the amygdala. I'm not sure. I think I did the talking, you did the listening. I'm not good, but it was a fun time. And I even had a chance to connect two of my big heroes in science, Aaron T. Beck, who developed cognitive therapy, and Joe. So we arranged for a get-together in Tim's apartment when he was still alive. He passed away at the age of 100, just about, I think, 2 years ago. And Joe then also visited me in Würzburg and doing a sabbatical. And then over the years, we established very close relationships. He introduced me then also to Liz Phelps, who became a very close friend and collaborator. And we had a grant together, the McDonnell grant, that we did this collaborative work, really exciting stuff. And then in more recent years, we wrote papers together with Hakwan and Matthias on the philosophy of consciousness and subjective experience. So what we clinicians learn from Joe is far-reaching, and I just want to highlight a few things. I think most of my colleagues would agree that we have to be careful with our language. I think they got it. Fear and anxiety-related circuitries in humans are complex, and it's not the amygdala being a fear center, but rather the amygdala being a threat center and the emotion that is closely tied to the subjective experience of mental operations. So in Joe's words, also when we've been talking about in these various workshops, he once stated that, I quote you frequently, there's a lot of animals in humans, but not a lot of human in animals. We have to be careful when we make inferences from animal work onto humans. We can do this on some level, on the threat level, on the less evolved evolutionary level to some extent, but when we talk about emotional experiences like anxiety, anticipating things that is lying in the future, which is going to be a topic that I will be discussing, or even the subjective quality of an emotional experience, then we have to be very careful because we are no animals. We are not bats, as Thomas Nagel once said. Or what it's like to be a bat, and it's a little difficult to say what it's like to be a bat. But I'm a human, I can see what's from a human perspective, but no matter how often I ask my dog, he hasn't responded well to my questions. So there are also many similarities, however, between animals and humans, but there are important differences we need to be aware of. And there are also many similarities, and this is now the quintessence of my talk. There are many similarities between individuals, but there are also important differences between individuals, individual humans. I think we haven't paid enough attention on these individual differences, but these are exactly the kind of things that we as clinical psychologists are very interested in. What makes, not only what makes us similar, but what makes us unique, what are the differences between individuals, and can we identify perhaps subgroups of individuals that belong together more than other subgroups on a neurological level, on a neurobiological level. This is the topic of my presentation that will be in the cerebral cortex, which I've done with my brilliant students at the University of Marburg. I'm not sure if you're familiar with this picture. You should be familiar with this. This is something that we, that Joe and Vincent and Matthias and Hakwan and I put together that sort of captures this idea of we heard already about the higher order theory, that it requires beyond simple threat circuitry to capture the emotional experience. And just as there are similar circuitries within an emotional experience, there might also be different circuitries that distinguish one person from another. So to study now this generality and individual differences of within person models concerning this suppression of imagined threat and to identify subgroups of similar connectivity patterns. That is the aim of our study. That was the aim of our study, and we wanted to determine what are those relevant subgroups and are they distinguishable on a subjective level of anxiety? That's sort of the important detail, obviously. You can probably always identify subgroups, but they have to have also some sort of connection to their subjective experience. We hypothesized that there might be such subgroups, and it would have quite important implications, because we might have then biotypes that might respond differentially to therapies and maybe differentially to different forms of therapies that might actually then, that's sort of one step toward personalization of therapy. In particular, I'm here focusing on psychotherapy particularly. So we used an experiment that was done by Benoit. You've heard of him already before. And he used an no-imagine task. The paradigm is as follows. So the experimental protocol spans 2 consecutive days. I think Benoit had a third one, but it's minor modification. The first day, participants then generated and rated 18 personal fear-provoking scenarios. For example, people then might describe sitting in the kitchen table in the evening when their partner announces they want a divorce. Something that sounds awfully familiar. And then they could elaborate on how emotions like anger, sadness emerge and how tears start to well up in their eyes. And for each scenario, then they provided a reminder word such as divorce as an obvious cue, and a code word representing a typical aspect of that imagination, such as glass of water or something. And then they rated these scenarios on 5-point Likert scales on various dimensions. And on a second day, then MRI measures were then conducted. And before scanning, then people underwent a brief preparatory training phase, and then outside the scanner to familiarize themselves with the task. And then they repeatedly imagined this event associated with the green reminder words that were followed. So I'm gonna, in the interest of time, I can go back to that, but I think I'll skip that and also this one and go right into the subgroup analysis, because that's the sort of the juicy part. So we used GIMI, which some of you are probably familiar with that, who've done a connectivity analysis. GIMI is a group iterative multiple model estimation, and GIMI does require sort of a prior selection of directional mapping between ROIs that you identify, and as in the case which I'm not going to go through that because of time issues, similar to the dynamical causal modeling, but there's different important advantages of GIME as compared to the DCM. GIME employs a unified structural equation models to establish first group-level connections that are applicable to the majority, to most individuals, generalizable connections between regions of interest, and then identifies clusters of individuals with similar connectivity patterns by creating this kind of personalized models within a person process. This is called SGIME. So this is on the the result of this group iterative model multiple model estimation. In panel A, you see black lines that indicate group level effects. Thin lines indicate individual level effects, and green indicate subgroup level effects. And the line width corresponds to the proportion of individuals having the effect. The dashed lines are lagged, so you can also do not just sectional analysis, but lagged, and the solid lines indicate contemporaneous connections. In panel B, you see these blue arrows. They indicate group level effects. The green arrows indicate subgroup effects. And then on group level, we could reveal contemporaneous connections between the conditions and the right and left posterior cingulate cortex and the right PCC and FMPFC. And then lag group level connections emerge between the left and the right PCC. So when we did this, the result of the GIME was eventually these two graphs. So we identified these two subgroups. In subgroup A, it showed a lagged connection from the right posterior cingulate cortex to the left posterior cingulate cortex, and in contrast, subgroup B exhibited a contemporaneous connection from the left PCC to the ventral medial prefrontal cortex, as well as connection from that condition to the left PCC. When you now look at how these subgroups differ in anxiety, subjective anxiety, in fact, they did. The p-value unfortunately didn't survive multiple corrections. But we got a pretty healthy effect size of 0.58. But again, there's the p level on this direct comparison is significant, but it did not survive Bonferroni. So now there is something there. I think there's a signal that we could follow up on. It's highly. It could point to important biotypes that might inform treatment selection. So what we did is to examine individual differences in functional connectivity. We used GIME in its subgrouping function, SGIME, to subcategorize 54 participants. We got two fairly equal subgroups, one about 4% of this group that showed additional lag connection from the right to the left PCC and less brain connectivity, and a trend toward higher anxiety compared to the other subgroup, which showed an additional connection from left PCC to the ventral medial prefrontal cortex. So the obvious question is, are these biotypes, do they have a value in terms of treatment selection? Can we, putting them through a scan initially, and then that would be obviously exciting to see one group responding better or worse to treatment, or even tailoring the treatment specifically to them. So we have more work to do. Thanks, Joe. Thank you very much for your attention. Next. So I think.
My name is Marco Tamieto. It's a real pleasure to be here to pay tribute to Joe LeDoux. Louder, right. It's okay, right? Okay. So it's a real pleasure to be here and pay tribute to Joe LeDoux, the scientist, the musician, and the friend. And I'm familiar with all these three dimensions to a different degree in my experiences with Joe. So just as a way to start it, this paper appeared almost 10 years ago, Antiques, under the title, What is the Most Interesting Part of a Brain? And here you see the most interesting part of a brain. It's a little bit of a joke. Actually, you could say what's the most popular part of a brain. And in red are the most popular part of a brain. And here on the right side, the most popular labels and topics associated to those parts. And, well, author says that regions in emotional processing return stellar performances. And I mean, to put facts in perspective, consider that some 40 years ago, when Joe started his seminal studies, emotion or fear were barely legitimate topics in neuroscience. And with a few exceptions, the amygdala was a small structure deep down into the temporal lobes nobody was really paying attention to. So Joe's work on the neural circuits of threat essentially determined to a great extent what is represented in this picture. And if you want to put it more formally, his studies provided a paradigm shift in contemporary neuroscience and paved the way to establish a new field that is now referred to affective neuroscience, even though I know that the word probably Joe doesn't like it too much, but that's at least to understand each other. But let's go back to the beginning of the story, at least my story of my connection with Joe without Joe knowing of me being present in this world, and dates back to when I was an undergraduate student. I was very interested in two topics within neuroscience. One was consciousness, or the breakdown of consciousness that happens when you have a lesion to the brain as a neuropsychologist. And I was attending Eduardo Bizia, who was a statuesque figure in neuropsychology, as a founding of neuropsychologists, and he pointed to me to read this book by Larry Weisskranz, Consciousness Lost and Found. And then the other topic I was very interested in was emotion, and he recommended to read this book, now here translated in 1996, I think, The Emotional Brain by Joe. And that's the way it started, so I wanted to combine the two things, and I wanted to study blind sight, which, as you know, is the ability of patients who have a lesion to the primary visual cortex to process the content of visual stimuli and to respond to those stimuli, even though they are not consciously aware of what is projected in the blind field affected by the lesion. And I mean, the combination of the two topics is very well represented in this picture that many speakers yesterday and today have presented. So I mean, I had the proverbial beautiful idea to try to test whether a blindsight patient actually can process some emotional stimuli, and was except that had already been done in 1999. This was the first behavioral study by Beat de Gelder and Larry Weisskranz documenting a phenomenon now known as affective blind sight, which is, I mean, the possibility for these patients to process non-consciously the emotional stimuli. So what's left to me? I mean, we kept going from that moment without publishing so far one single paper with Joe, but still having his influence on our own work. I started this long-lasting collaboration with Beat de Gelder, who is here, talk after, and Larry Weisskranz, he's no longer with us, and testing blindsight patients and trying to characterize different aspects of the way they process emotional information and the way they react to it. And I think, I mean, just to summarize, I mean, two things that I'm particularly fond of. One is that we were able to document that the non-conscious processing of facial or bodily expression actually go beyond the visual system and induce in the unaware observer some of the changes in physiological arousal as measured here in pupil dilation or in facial mimicry that are that occur in the healthy observers when they are exposed to emotional signals in the face or in the bodies of others, like emotional contagion. And then obviously, I mean, the low road that Joe reported and described for the first time in the auditory system of rodents, we try to characterize within the limits of tractography, of DTI, the presence of those fibers connecting the superior colliculus, the pulvinar, and the amygdala in humans. And we were able, almost, 12 years ago, to report that those fibers do exist in the human brain. Not only, but they are transformed to some extent. They undergo some plastic changes, and their number is increased in the V1 damaged hemisphere of blind-side patients, so showing some plasticity and so adaptation of the brain, even for lesions occurring late in life and for structure there are of old, ancient evolutionary origin. And these were the small movie clip documenting in GUI that the fibers in green, which are those in the left hemisphere where he has the damage, are significantly higher in number than those in his right intact hemisphere. And luckily, some years ago, the group of Bob Rafal and Andrew Bell in Oxford approached the same topic in a group of non-human primates and a group of macaques, and were able to report the same connections, so that finally, and then there are a couple of years ago, a study by Elret in Frontiers reported with track tracing this connection between these structures. But this is the scientific part. And then in 2018, the friendship arrived when I had the chance and the pleasure to invite Joe to University of Turin for a sabbatical, for a visiting professorship, and also to receive his honorary degree in the Neuroscience Institute. And that was the flyer at the time, which was fantastic and included some of the speakers present today. You might see some of those at the Academy. And but before, the night before, there was the amygdaloid concert. There was a plaque concert, I think. And no, there's an unplugged concert. And I remember we were in a relatively small club in the center of the city, and it was crowded. There were colleagues and people lining up out in the street trying to sneak in, and they just rocked the house down. And so, it's just to close circles through the high roads of science, through the low roads of music, through the multiple roads of friendship, and to thank Joe for all the inspiration and contributions and supports they gave us as a community, as effective neuroscientists or neuroscientists dealing with emotions and dealing with consciousness. And I have just one more thing to say, that here is a summary of recent and last recent book that have been translated in Italian. So, Joe, it's just too late to quit. Please keep going. Thank you.
Start from there. My poor ability with Mac. I'm sorry, but I didn't grow, so I'm still short. No, that's okay, don't worry. It's joking. That's fine. don't worry. That's maybe the only joke I made make today. Sorry about that. Okay, so I am Valérie Doyer, and I'm leading right now a team that's called, in France, in Paris, Paris-Saclay, that's called Memory, Emotion, and Time. And actually, it actually tells pretty much the path of my life, going through Joe in the emotion part. It started in 1999, long time ago, when I contacted Joe, and because I was working on the hippocampus, LTP, LTD, associations, that type of stuff. And then I wanted to move on to something else, part of the association, which was part of the emotion, motivation, that type of stuff. And so I contacted Joe, and he said, sure, come in. And so I ended up to go into his lab in '99, stayed there for 3 years. And then since then, I couldn't quit completely, so I had to go back and forth. And thanks to a lot of little grants that we collected to do that, and grew up then to an LIA, which is a lot of acronyms. Forget those things, because there's an even more funny one after. But that actually we connected with other groups. That's thanks also to Joe. Regina Sullivan arrived in New York, and so we connected with her, and Anne-Marie Molly, my colleague in France. And then we end up to grew even more with emotion and time. With the PUF, remember Joe was like, what is that PUF? And the PUF is actually Partner University Fund. That gives a lot of money, actually, to help collaborations and transatlantic Franco-American exchange, cultural exchange, with a lot of peers and students and postdocs and all that back and forth. And so we enlarged the questions to the time with two experts here that were actually on the French side, Sylvie Droit-Volet, and on the left side, Bruce L. Brown, who's there, and who ended up to be my partner, actually. So we got, as I was saying, back and forth and meetings and supported by the PUF. And one was in France, and the arrow point to Joe here. And then and another big conference that we also made in NYU, where Joe was talking and playing music. And because there was time flies with music, and therefore that was the connections between the two. Okay, emotion and time, so time flies with music, but what happens when you are afraid? So there are two ways to think about that, which is timing of a threatening cue or effect of fear on timing of a neutral cue. That's more like the science-based things. And how do you study that? Well, and then thinking of that, well, you think about the association, and then you learn and you read from Pavlov, but also to Gallistel and Balsam, that, well, time is actually maybe at the core of the association. That's a critical, that makes the association somehow. Okay, so how do we do that? And thanks to Joe, I hate to be on the web. I end up to be on YouTube. Well, because we published that paper, and for some people who may remember that lab, now it's closed, we made that paper that showed that there was the change in the CS-US interval would actually trigger reconsolidation in an amygdala-dependent manner. And that was a big hit, not only because of it was kind of saying, oh, reconsolidation, woo, how do you get that? But what it says also is actually that the animal had encoded the time. And so then I went on to that, and we recorded field potentials in different structures, kind of amygdala-centered structures, like with amygdala, with prefrontal cortex, striatum, and then look at the coherence activity between those of the North oscillation between these structures. And what we saw was actually that you do have in a frequency band, time frequency analysis here, an increase in coherence between those pairs of structures that are actually time-locked to the expectancy of the US arrival. So there is a temporal expectancy of the CS-US, of the US arrival, even though there is no US the next day. And it goes in those pair structures. As you can see here, 30 seconds, 10 seconds. When you do a CS minus in the differential field conditioning, CS minus, nothing. Even though they freeze. So they freeze, no temporal pattern of freezing, but the brain knows the actual coherence of activity in that network. We didn't record broader network. That may be true for the whole brain, actually, but at least this network. And what we saw also is that that actually from the early beginning of the learning here, where after only 10 trials you have that, actually if you continue training the animal for a lot, then the temporal expectancy ends up to peak at the time of the US arrival, and you can see that behaviorally. I'm not going to go through how we do that, but actually then the coherence within the same frequency band centered around that and gets actually correlated to the behavior. So from an initial asymmetry, we go to transform into a symmetry centered around the US time. And I forgot to mention in the slide before is that the CS-US interval is actually learned in even a single trial. That actually was demonstrated by Davis in 1989, but we also showed that through the reconsolidation paradigm. Okay, so you time the CS-US interval. How do you know whether your timing is actually accurate? You may have a distortion. And in fact, when you look at in a Pavlovian task, whether when a tone is predicting a shot, another tone is predicting food, and you look at the temporal expectancy, you see that actually when they're expecting the shock, it's shifted to the left here, meaning they overestimate the time at which the shock is arriving, which actually, when you think about it, is a good idea. It's better be prepared one rather than too late to receive a shock. However, if you give a fear cue, a cue that has been associated with shock, while the animal is timing something else, a neutral cue, well, actually the disruption is in the opposite direction in the rodents. So those are rodents. I forgot to say, yeah, indeed, that is also depends on the amygdala. That's other people have demonstrated that, and also from the prefrontal cortex. Back to that ordinary. So in humans, so that task is a bisection task. So whether you train the subject to associate a specific duration of a neutral cue with one response, a long duration with another response, and then you're going to ask the subject to estimate the duration of a face that express some fear or anger or whatever. And so here what you can see is that with anger, actually, there's a shift to the left. So there's an overestimation of time when the face is expressing anger compared to a neutral face. And the same, actually, when you shock the subject, when you tell the subject that this cue is going to be followed with, or there will be some shock given in the middle, and here also you get the shift to the left. So in other words, there is a distortion of time perception while under the emotional state in humans. I can say emotion in humans, right? So so you have this kind of big network, and you say, okay, well, how do you know what's functioning at the basic, really, and what do you need to have the whole network to be ongoing to have this distortion of emotion? So the thing is that the amygdala is one of the structures that's functionally mature very early, while the prefrontal cortex is, as everybody knows, is the latest one. Well, in fact, when you look at this, whether the rat pups actually encode the CS-US interval, yes, it does. So before the prefrontal cortex or this whole structure is mature, the amygdala is functional, and the animal is actually learning that CS-US interval. And what's interesting in humans, well, actually, the timing of the hungry face, what we saw here in the adults, is also, do you have the same shift in children of 5 years old, for example? That was Sylvie Droit-Vollet's demonstration. And, but what is interesting is that when you have a timing of an expression of a shame, which is actually a secondary emotion in humans, you have a shift to the right, which has been interpreted in the timing literature as attention mechanism. You don't have the same effect in children. So in other words, there's a kind of switch as there is a maturation of the network, the global network, there is a progressively an effect of the emotion on the temporal perception. And so, well, I'm not going to go through that, but it's just like we have been discussing a little bit with Sylvie Roy-Volet about how these things, and I think it's really to point actually that it's important to have maybe this, try to go through that more in-depth analysis of developmental and maturation of network that would help us to understand at which level or where you're going to enter into this fear, emotion, distortion of time, and things like that. And well, I cannot say more than just that. It's just like I've been through Joe, really collaborating with a lot of people in his lab. That's why I keep going back and forth for so many years too. And it took me, it was such a pleasure to talk to everybody, to collaborate with everybody, to have Joe, open to any options, possibilities. I mean, said, time, what is that? What's the need of that? And so, and he's been welcoming to ideas and discussing, and I've been collaborating with a lot of people through him. And that's just, I would like to thank you a lot with that. Without you, I guess, I would not be where I am at this point, really. Thanks a lot.
When you reach my age, you see that apple is not nice. Skill.
You think? When you reach your age?
When you reach my age, I think.
You think?
Yeah.
I think so.
Yeah.
We're going to.
I see. I'm.
What's this? Yeah, here you go. Which one? This one?
Raquel, yeah.
Floor is yours.
Thank you. Dear all, good afternoon. My name is Raquel Martinez. I'm a researcher at Hospital Sírio-Libanês, and the hospital is located in the heart of the city of São Paulo. And I want to start my talking saying that it's an honor to be here and say thank you for all the people who have organized this meeting. I know it was a huge effort, and it's great that you work hard and made this meeting a reality. Thank you so much. So I would like to start bringing you guys to my journey, and I want to bring you back to 2009 when I received a postdoc grant to work with Joe. And of course, we all know about Joe's career, how brilliant he is, and how he was inspiring people around the world. And of course, I was one of those students that want to learn with him. I want to learn more and more. I'm always looking for more. And it's exactly what happened with me. So I joined the lab, and it was very nice to be there. So one thing that I learned, it's about science. I learned a lot about science with Joe. But one thing that I want to tell today in this talk is that I learned how to be a leader. So this is the goal of my talk, to tell you guys what I learned about how I should be, how I should take care of the students, how I should organize everything, and how I should be a leader. So I was very welcome in the lab, and we used to have our lab meetings on Fridays, and we decided to give an upgrade in the lab meeting. We decided to change to Fancy Fridays lab meeting. So everybody would come very well dressed up, and we'll be discussing data. Joe was there guiding us, sharing data, sharing results. Everybody was in an atmosphere of very enthusiastic. And it was so nice to learn with Joe. And it was so nice the way that he was delineating projects and new experience and new stuffs. So it was amazing to be part of there. And of course, I had the opportunity to celebrate Joe's 60th birthday. And today, to celebrate his birthday again. So, Joe, happy birthday, officially now. And, oh, sorry. And during this time, I learned another skill from Joe. Joe has this incredible ability of bringing people together. So it was so nice to see past members and present members in the same environment. And it was so nice to learn with them. And this just happened because of Joe, because this powerful skill that he has. And we had several events. We have several things nice. And one of the nicest was our fun picnic at Governor's Island. And it was so fun to be there, everybody together, having a relaxing time, discussing, enjoying good food, yummy food, playing volleyball. And in that time, I figured out another powerful thing from Joe. He has this amazing ability of bringing people and making them feel like a family. This is a super skill he has. And of course, we had amazing happy hours along the time. And we were watching Joe playing the amygdaloids. We went to several places. And it was very nice to see that science and art, they need to be together. They are meant to be together. And Joe taught me that. And of course, time passed by, and it was time for me to leave. And I found the email that I sent inviting all the CNS members to join my goodbye party. But of course, it was a dancing party, not a regular goodbye party, of course. And it was sad, but I had to leave. So then I was in Brazil. I started my lab. I work with neurobiology of emotions. So I had my babies. I had 2 girls, my twins. And of course, I told you, I'm always looking for adventures. So I started medical school. I got into medical school. So now I'm the 5th year of medical school. Next year will be my last year of medical school. And I kept running the lab. I kept my students. I kept publishing. And of course, I was doing my training in medical school. So it was not the end. I was meeting people at conference. I was meeting people around. And last year, we had this unique opportunity to meet a lot of people at John's honor ceremony. And it was so nice to be there. It was so great to be how Joe has inspired so many people around the world. And of course, everybody, oh, I love this Glenn picture here. So it was so nice to see all together. And another thing that I learned from Joe as being a leader is the power of gratitude. He has received a lot of awards along his career. We know this. But he was emailing us and saying thank you for us for all the things that he had got. So this is the kind of person that he is. He's always very grateful. Oops. And this is my message to Joe. Joe, you had a profound impact on my life, not being just a mentor, but a friend. Oops. Think I can, oh, did I change anything? No? And a source of inspiration. Thank you for your guidance, kindness, and incredible legacy you leave behind. Oh, sorry. Can I just go to the next slide? I told you, Valerie, I'm not a high-tech person. Should I? Oh, thank you. And I'm sorry. And I'm incredibly thankful for the meaning experience and connections that have come from being part of Joe's lab. And to finish my talk, I brought this imagery that is exactly what represents Joe's for me. Joe has this incredible power of bringing people around the world together, and not just bringing them, but bringing them and feel that you are very welcome. So this is the way I feel, and this is how a lot of us feel. So thank you, Joe. Thank you, Joe's Lab, and everybody that's here for hearing me.
All right. Okay. Hello, everyone. I'm so excited to be here and that I get to talk to you all about Joe because we've heard from everyone what an amazing impact he's had on so many lives. And it's no different from mine. And I feel like it's really being back with a family here. So thank you all for being here. And thank you for the organizers. It's a wonderful job. So before I start talking about, my ride of coming into the LeDoux Lab, which started nearly 25 years ago, I thought I would share with you the movies I watched on the plane from London Heathrow coming to JFK. The first movie I watched on the plane was Twister. And this guy, he reminds me a little of Joe. Do you guys see the resemblance here? This guy's hunting twisters, right? Hunting storms. And he says, you don't face your fears, you ride them. I think, Joe, that's really what you've been doing. The second movie I watched was Blink Twice, where I again got a quote from Fear out of the movie. And that was, fear is just a trick your mind plays on you. The only way to overcome it is to blink twice. I don't know if that's going to work, but we'll see. Okay, so how did I get to know Joe? How did I end up at NYU and at CNS more specifically? So it was 1999. I was in my undergraduate, and I was thinking of becoming a psychiatrist at that point, studying in medical school. And I had read Joe's book, The Emotional Brain, which I think has made an impact on many of us. And I had created this fantasy. I think we had some talk before about like kind of future and fantasy, maybe grandiose thinking. Maybe that's what I had. Grandiose thinking that I was going to work in the lab of the man who had written this fabulous book called The Emotional Brain. So what do? You email him, right? Because he's on the other side of the Atlantic Ocean. At the time I was in Utrecht, in the Netherlands. And I had to email him a lot of times, and I was not getting a reply. Liking The Emotional Brain a lot was not really doing the trick. And at that time, I went on a big trip with my family, sort of like a last big trip with my parents, my sister, to South Africa, Botswana, and Zimbabwe. And I was traveling around, but the time was coming that I had to start this internship in, hopefully, according to my wishes, in Joe's lab in New York in January. So the time was ticking, and I said to my dad, well, this is getting really frustrating. This guy, Joe LeDoux, he's not really emailing me back. And sometimes you have to listen to your dad. And my dad said, well, I've got a really good trick for you. There's two things that you have to write in the email. One is you need to write something really unexpected to catch his attention. And the second thing is, which I don't think you've mentioned yet, you should tell him that you got the Erasmus funding so that you are fully funded. So I write to Joe again, and this time I write, hi, Joe, hi from Harare, Zimbabwe. I would really like to start working in your lab, and also I have Erasmus funding. Joe got back within 5 minutes. And he wrote back, hi from Paris, and gave me a date in January to meet him and a time to meet him in his office at NYU Center for Neuroscience, 4 Washington Place. So there it was. I bought a one-way ticket to New York because I'm an optimist. So that day, I got into we've gone into the new millennium. It's now 2000. And I have a meeting with Joe. I, of course, have never met him before. I don't know what kind of guy he really is. And I come into his office on the corner there. Many of you have been to this office. And I come in and I say, oh, hi. And I thought, well, he will have things arranged of how this is all going to work out. But he says, oh, I don't really know how to do this. Am I supposed to interview you or something? And or test you if you actually know stuff about neuroscience. So he grabs the Candle, Schwartz, and Jessel book, opens it up on a random page of NMDA receptors, and asks me to explain. Apparently, I didn't disappoint, because moments later he said, "Okay, yeah, you can come work in the lab, and I'll introduce you to Glenn Schaaf, because he really needs someone to work with him on some interesting stuff. And also, he's in a race together with Kareem to get this amazing paper finished on reconsolidation." So many of my early days in the lab were late night staining, slicing of brains, and all of that to help get this paper published. And my first proudest moment was that I was thanked in the acknowledgements of fear memories require protein synthesis in the amygdala for reconsolidation after retrieval, because we all know what a seminal paper this is. What I didn't know was all the amazing things that were ahead of me. Everything that was going to happen just because I made that amazing choice of joining the Joseph LeDoux lab. And one thing was that I was going to meet my now husband, John. Here we are in Washington Square Park in 2000, before the first kiss. And a friend of my sister's, who is a photographer, actually took this picture with like a long lens, sort of what the paparazzi use. So I didn't know this picture was being taken, and she sent it much later to me. We used it for our wedding invite as well. So at this time, of the year being 2000, and being really in the golden days of the LeDoux lab. So many people that were here, we had, at that time, I think Joe had the biggest lab that it ever was. It was like 20 plus people. Amazing discussions around, bagels and pizza, no fancy Fridays, but, we had a good time. And another thing that used to happen, illegal stuff happened, maybe you weren't aware of, was that many people used to go and have a smoke on the rooftop of CNS. You had to press a door and an emergency exit and then climb up to the roof. So I thought I was really missing out on this because I wasn't a smoker. And I thought, well, what I really need to do is pick up smoking, because that seems like a really wise investment. Because obviously I'm missing out on a really good stuff. Interesting science, interesting talks on the roof. So I picked up some Lucky Strikes and decided to start smoking 2 cigarettes a day on the rooftop. Kareem was there, many others were there, and of course John was there. And that's really how I got to know John, and that's actually how I think that we got together on those long conversations on the roof. So many cool things followed on from there. We did some science. And as I said, Joe had linked me with Glenn. And Glenn was my postdoc supervisor, and he was the best supervisor anyone could wish for. Because anyone who knows Glenn knows that he does things with such precision and integrity that he's really one of those great guys to really learn from. And what he wanted me to study was auditory fear conditioning, but looking at the role of the auditory thalamus. So that's what we set out to do and did for several years. And actually, a couple of years after that, after I started my PhD, because I hadn't put that part in the study, because I was a student, of course, working in Joe's lab, had to go back to finish my studies in the Netherlands. And then I got a really nice email from Glenn that he said, oh, I need a research assistant. Can you come work for us? And I thought, well, that's my dream come true. Because at that point, I already thought, like, what I really want to do is a PhD, and a research assistant would be really ideal for that. So I started my work and started working on this auditory thalamus. So I had a bit of a head start to my PhD, because I had already, of course, learned all these skills, and I knew how to do this. And yeah, I applied for the PhD programs in New York, got into several of them, and chose to stay at NYU, because there's no place better, really, right, than CNS to do your PhD. So you guys might remember this guy, Winston. Every morning coming into CNS, I would have a little chat with him. Great guy, always really fun to chat with him. And then my time as a PhD student started, but it didn't go exactly as most PhDs would go, because very early in my PhD, I got pregnant. That wasn't really in the planning. And being, we've heard of people, being even in labor and writing corrections and things like that. But yeah, having two kids during your PhD was challenging, but actually it wasn't that difficult because I had so much support. And at that time, I had switched my PhD that I'd started in Joe's lab, looking at fear conditioning in rats, and specifically auditory fear conditioning, to Liz's lab, who became my second, actually primary supervisor at that point, because I wanted to do auditory fear conditioning in humans, and I was going to set that up as we had just recently acquired fMRI at the Center for Neuroscience. So that was an amazing opportunity, and Liz was just as an amazing supervisor as Joe. So I was very lucky to have such amazing people watching over me, supporting me, and people that I could really, really learn from, and all the other people that were around at the time. So here you can see some pictures of how it was to, that was our apartment on 2nd Avenue and 3rd Street. We had a 7th-floor walk-off with 100 steps, because that's really handy if you have 2 little babies. And this, I had to put a picture in of my dad, because he was the one who encouraged me so much to email Joe again. It's on our rooftop on 2nd Avenue. And then this is a picture from yesterday, and then you can really see, how it comes that all of us coming together gives such a warm and fuzzy feeling, even though, of course, it's all about the science. We've had so many good times all together, so many outings, so many drinks, listening to the amygdaloids. It has been a really wild ride. Also, amongst that time when I was still pregnant, we got married, John and I, in New York, because that's what you do when you have unexpected pregnancy. And we gave also a proper party a year later with our 4-month-old daughter at the wedding. So that's Yesha. She's 19 now, studying herself at UCL. And in the meantime, when this picture was happening, so Glenn, my postdoc supervisor, was at the wedding, and he's like, oh, I got some really good news for you. Our paper got accepted on auditory fear conditioning, the thing that my career kind of started with working on. And he's like, and guess what? It got accepted without corrections. And he said, well, that is really, really good. We can go celebrate that. So I had that to celebrate on top of the wedding, dancing on the platanos around a tree. Life couldn't be much better. So here are some images of that paper. And we were basically trying to show the presynaptic involvement of the auditory thalamus. And the way I even learned for this paper, because Glenn was never happy that we had enough evidence, so I even had to learn how to do LTP as well. And finally, we finished it off by also showing that with these MAP kinase inhibitors, we could also block long-term potentiation when we were stimulating the auditory thalamus inputs to the amygdala. And I was really happy to continue auditory fear conditioning in Liz's lab. So there I got to record actually sounds, make sure they were outside the range of the scanner. And eventually we got a paper out as well on auditory fear conditioning in humans. And then it was time for me to wrap things up in New York. Even though I had 2 kids, that was actually my greatest accomplishments. Well, not having the 2 kids, of course, was good, but also that I still got to finish my, my PhD on time. And this is me giving my defense talk. Some of you that are sitting here were at that defense. Actually, doing a defense here in the US is quite scary compared to how it goes in the UK, because you have such a big team that's like, I remember, of external examiners, etc., that are also present. And one of them was Bruce McEwen, who has now since passed away. But I was thinking, how lucky am I that I have all these amazing and intelligent people, you know, reading my thesis. And my thesis, I think, was one of the first ones that had the word translational in it. So, okay. So this is it, wrapping up. I'm now working on OCD. This is Liz's cake for me, and here you can see some of my latest work, some partying that happened along the way, dancing with Kareem, meeting Joe at ACNP, going to Japan and doing karaoke, and these were just yesterday, and we're all warm and fuzzy again with beers in the German beer garden last night, and it's so great to be back and be with you all. Thank you so much, and thank you, Joe.
Just a short 10-minute break because we're running a tiny bit behind. So if everyone could, come back quickly, that'd be great. Just get some coffee and come join us again for our final session.
Okay. So as everyone's getting seated, I'll just start in the interest of time. So just a little plug. You've been hearing about a special issue, and this is something that Daniela Schiller and Linnea Ostroff and I organized. Go to Cerebral Cortex. Check out. There's a bunch of amazing articles about Joe, about people's work, about Joe's work. I really encourage you to take a look at it. Yeah. So my name's Josh Johansson. I have a lab at the Riken Center for Brain Science in Japan, and I was in Joe's lab from 2007 to 2011. And I was looking for a catchy title. I'll try not to cry, but we'll see how things go. But Joe's work, his scientific work, has really been sort of the soundtrack to my life from the very beginning. As a young neuroscientist coming up in the late 1990s, his studies on mapping out circuits, I think, were really ahead of their time. In the age of circuits now, he really led the way in understanding mammalian nervous system and the circuits that mediate defensive learning and behavior. This is this great annual reviews paper that he wrote. I guess I'll just make one edit, Joe, just so we're all comfortable and feeling good about this. But the rest of it's great, of course. So I did my PhD with Tad Blair, who was a postdoc in Joe's lab and couldn't make it today. And I first met Joe at a Pavlovian Society meeting, and he came up to me, and the first thing he said was, "I'm looking for an in vivo electrophysiologist. Do you want a job?" And I was honored at that time, but I guess this might be a general greeting you give to people. I guess you said the same thing to Greg Cork, or maybe you're just hard up for electrophysiologists. But I was honored. I mean, to go work for my scientific hero, this was an incredible opportunity, and I took it. So I've been scooped on a joke and also on this picture. But we had these dress-up days, and I knew as soon as I started in the lab that this was really going to be an amazing and special experience. And I think that the secret sauce that made Joe's lab so successful was his ability to bring together really the best people. And I remember that before I came to the lab, Tad had told me, there's Joe's world, the experiments that you talk to him about and he thinks you're doing, and there's the real world, which is the experiments that you're actually doing. And I think this was actually part of Joe's grand plan, because he really knew how to be hands-off and let brilliant people from different walks of life sort of synergize and come up with amazing ideas and do great science and really push the field forward. And I think we were all really benefited from being a part of that. So when I moved to Japan, Joe was incredibly supportive, and we continued some collaborations from the beginning. But at this time, he also published this piece, as you all know, Rethinking the Emotional Brain. And I think this was important and necessary to move the field away from using the term fear. But I think for a lot of the rodent scientists, this was a little bit unnerving, because I think there were some in the community that felt like in the rodent world, we couldn't study emotion anymore. And this was concerning to me. But through the years, I would come back and meet with Joe on frequent trips to New York, because you can't really stay away from this place. It's an amazing city. But also, I would really value these discussions that I had with Joe about the nature of emotion and how to study it. And what I came to understand from talking to him, and he's since published more recent articles on this, exemplified here, is that he didn't think of it as a sort of dichotomy between conscious experience of emotion and the basic survival circuits. That there are these intermediary types of representations, internal models, for example, that are pre-conscious, that possibly we could study in the rodent. And this is something during the pandemic that my lab really started to think a lot about. How are internal models of emotion represented and formed in the brain? And to take you through an example of sort of our thinking on this, imagine that you're in your neighborhood and you have an internal model of the neighborhood in your mind. You can sit in your house and think of kind of where your house is, where the supermarket is, where different places in the environment are. There's a sensory model. Now you go for a walk in your neighborhood and you come into the park, and otherwise pleasant experience, but on this day, not so much. There's this vicious dog that jumps up and attacks you. It's the PR people at my center made this for me. I love it. So bad experience, right? So if you see the dog, the visual representation of the dog is aversive, you're going to freeze and have fear responses and so on. But you may also know where this dog lives. And if you walk by the dog's house, you're likely to be afraid, even though the house itself has never done anything to you. So something about that experience has altered your larger internal model so that you can incorporate different indirect associations and perform inference in terms of your emotional regulation. And this is something that we wanted to look at. And so we thought, what are the ingredients that go into these internal models? You need the sensory model of the world, and then you need to incorporate or even separately encode the associative features of the internal model, the specific features that are directly or indirectly related with these aversive experiences. In terms of what could be encoded in these models, you need binding. You need to know the specific sensory aspects of the environment that are related to the aversive event in this emotional type of model. And you need to be able to use these to perform inference in uncertain situations or in new situations. And you also need to be able to segregate and maintain flexibility of the representations if individual aspects of them are altered. And I'm not going to go into the details of everything we found, but we'll touch on a little bit of this. So sensory models have been studied a lot in areas like the hippocampus, and Jeff Schoenbaum has looked at this in the orbitofrontal cortex, and other people have looked in the perirhinal cortex. What we were interested in is whether there are brain regions that specifically encode internal models of emotionally salient associations. And we decided to look at the medial prefrontal cortex for many reasons, from work by Greg Cork and others, some in the audience and many elsewhere. We knew that the medial prefrontal cortex is important for context-dependent learning and cognitive control of emotion. And we generated a simple hypothesis that the amygdala is doing the simple direct associations between like the representation of the dog and the bite, but that you need the medial prefrontal cortex is encoding this more internal model of emotionally relevant associations. And so we used a behavioral paradigm to kind of model the sensory aspects, the sensory coding, as well as the emotional aspects of this process. So it's called sensory preconditioning, and during sensory preconditioning, you form a simple sensory model. You pair an auditory and visual stimulus together. Then during the aversive learning phase, you pair, say, the visual stimulus with shock. And then during the memory recall, you present the visual stimulus that was directly paired with shock, as well as the inferred stimulus that wasn't directly paired with shock, but was associated with the visual event, sort of like the house in our example. And we performed calcium imaging from single neurons in the medial prefrontal cortex throughout the sensory model formation and the aversive learning and the inference phase. And we found that after the sensory model was formed, there was not a change in the medial prefrontal cortex population coding. There were specific cells that were tagged, but the population code did not change. And during aversive learning, this is where we saw everything happening. These representations all became merged. And at the time of the memory recall, now you had this associative bundle where there was a larger increase in the population overlap between the auditory representation, the visual representation, and the shock itself, such that when you presented the auditory inferred stimulus, you can now re-engage this representation in MPFC to produce inference. And we also found that the projection to the amygdala was important for producing selectively inferred memories, and it specifically encoded the inferred memories and not the directly associated memories. So this was consistent with this idea that you have encoding of the emotional model here that's separate from the sensory associations, and that it helps the amygdala in cases of inferred cues. And this, I think, sort of touches back to what Joe is talking about in his recent writings on these internal models that are preconscious between the simple threat detection systems and the higher order. Thank you to the philosophers for defining this for us. The higher order aspects of the emotional experience, which, I mean, let's face it, we can't really study in the rodents right now, and maybe not in humans. Okay, but enough about the science. I want to get back to, so Joe's work has been inspiring me throughout my career and is inspiring me to this day in the work that I'm doing now. I think that, being in the lab, I have a quote I want to read. So, being in the lab, what I was struck by was Joe's humanity. There's a lot of successful people in the field. I mean, we know some of them, work up, we won't name them, but they might work up by the George Washington Bridge, who are not the nicest people in the world. But Joe is always, regardless of your level in the lab or whatever, he always treated everyone the same, really fair, and he works quietly behind the scenes to support the people who come from his lab, and I really appreciate that. And he also creates, as other people have talked about, a real family environment in the lab. When I left, Linnea Ostroff, another postdoc who worked with me, gave me a book by MFK Fisher, which I treasure both for the prose and for what she wrote in the front cover. And what she wrote was, we have all known that uncommon magic existed in the lab, and it will be a source of strength for all of us in the future. The bonds of trainees are usually the bonds of survivors. Ours are the bonds of family. And that was really touching for me, and I think that all of us have been touched by Joe in this way and are all part of the extended family, and I think we all know that today. And Joe, thank you so much for what you've done, and I know I'm going to be seeing more of you, and it's not over yet. Thank you.
Hi, everybody. First of all, I'm exceptionally grateful for giving a chance to talk here, and specifically on the occasion of Joe's celebration. Unlike many of you, I don't go back a long way with Joe, and I don't have any joint papers. I don't have any joint memories. I may have a couple of joint parties, but not parties where any fancy disguise, although I would have appreciated that, was. So rather than harking back to a lot of great memories, I'm going to end with a job assignment for Joe. maybe this is back. I don't like to talk about retirement, certainly not mine and not other people's either. So I'll end up, as you see at the end of my talk, giving some maybe some ideas about how to deal with the new job for Joe. Good. So I'm going to present you actually some new data from a blindsight patient, which is a well-known patient, TN. TN was tested around the age of, was first tested at the age of 52, and he had 2 consecutive strokes with like a 2 weeks interval, which basically left him completely cortically blind. There was no, as you see, I'll show you the images later on. There was no, not a sliver of visual cortex, as far as we can tell. Of course, everything is relatively relative, but so in principle, this would be a perfect. Is and was a perfect case for studying, for making some progress in understanding blindsight. Now, I must say that Joe came into my life, so to speak, on the occasion of my work on blindsight. And my work on blindsight sort of started when I was turning firmly my back to philosophy of mind, having been a philosopher for a while, training, going from epistemology to philosophy of mind, and getting seriously disappointed with philosophy of mind. So I thought, I wanted to study. I had this intuition that emotional stimuli are so powerful that they don't need a lot of cognitive treatment, right? So that actually, that intuition was confirmed by my becoming acquainted with Joe's work, and that work seemed to beautifully map on what we were trying to do, what I was trying to do with the blindsight patient. And I thought, okay, if I'm going to leave philosophy like I was doing, then here is a kind of scientific research that I could live with, namely the kind of scientific research that does not need to depend on any kind of subjective appreciation, subjective involvement of the subjects you were studying. So Joe's work on subcortical non-conscious routes for me offered an opportunity to do scientific work and not to be committed to any theories, philosophical or para or meta-philosophical, about consciousness. So that was for me a very, very welcome opening to go by. So as Marco has mentioned, we found, to the surprise of many, that the subcortical route, which had so much been stressed by Joe, was actually active in these patients. Okay, and a lot of philosophical analysis has been given about that. But here I want to show you some data from our patient. This is the first scan, the first scan that was published on the occasion of the first studies. You see what's happening to visual cortex there. There is a much better representation of that, which was done in a paper we did in 2008, where you clearly see bilaterally that there is no activity, no connectivity in visual cortex. Of course, also more upstream, but that's less relevant here. Here you get another visualization of that, which again shows that what the situation is with this patient. Note that this is the only bilateral case in the literature. We worked for like around 10 years with this patient, and we documented all sorts of residual skills, including blindsight for emotional stimuli. I just want to point out that bilateral cases, unfortunately, are extremely rare, but they are very important because you can, in some unilateral cases, of course, depends on the method of experimentation. You cannot always rule out that there is an impact, that may be due to experimental settings, to instructions, to task demands, etc., to some kind of metacognitive, if you want, to interactions with blind and sighted fields in those patients. It's something we've worried about for a lot, and it's one reason why so-called indirect methods of testing are, of course, superior. So now I'm going to show you 2 clips. One is a short clip, or would you call it object recognition, and the one is on color recognition. So I get out of here. Where is my... Can you... I don't know where you put it. I mean, the text is in French.
Which one first? Which one? Cylinder?
It's the cylinder, yes.
Par ça a brillé, bon, et maintenant c'est passé, disparu.
So here you see the patient trying. There were no instructions except, is there something there?
Dis donc, est-ce que c'est le même métal là? Ouais. Pourquoi alors la fois dernière je l'ai mis?
So the patient suddenly says it's metal.
Et oui, vous avez allumé quelque chose dans le sexe.
So that's pretty pretty competent grasping. There is still no object. So it's not. Let's not call this normal vision, right? But it's certainly color recognition. The patient says it's metal, and then a bit later on there is this recognition. There's the grasping of the object, and once he does that, it's like without. Once it somehow had said click, it's without any doubt that the object is recognized. Okay, I have another one on color now. This one. Oh no, that's you don't want to see me stop it. Sorry, Peter.
We're going to see about.
So one difference with the normal experimental testing here is that we created a very casual, like observational situation where we could exchange. Okay, yeah, okay. How do I start it now? Play.
Touch too much white in there.
Yeah, it's still the beginning.
With those two red bellows, he picked them up.
I should, why
is that because picking them up allows zooming in and out of
I don't know.
You don't want to see
Or
this.
it's an intentional factor.
Yeah, he might catch on to the edge. Looking for something that's.
What colour smart? Where are those little blocks, children's
I
blocks?
get them.
Yes, maybe the children's blocks.
Because then we can have
So I get some color pages also.
Trouver des autres objets.
So actually we tested red with a book, with a scarf, with all sorts of things, casually walking by, and this is the color of the sweater.
Une rayure sur votre pull.
Oui.
Oui. De couleur.
Oui, bon. Ici comme ça?
Oui.
Il faut. Ah. En tout cas, il y a le rouge qui doit y avoir.
Le bleu, le rouge, le blanc.
Le blanc, je le vois facilement, mais je ne l'ai pas dit. Parce que le blanc, il y a beaucoup de blanc. N'est-ce pas?
Oui, il y a du blanc.
C'est beaucoup le blanc, non? Ou bien c'est égal?
Oui, il y a plusieurs blancs. Il y a plusieurs.
Sorry, I thought we had cut it.
Voilà, les blancs, c'est beaucoup. Alors, c'est pourquoi je n'ai même pas voulu.
What you always have in these patients is some mental imagery, a bit of confabulation, so you really have to cut that away from the. So here we decided to use Lego blocks.
Il y a des couleurs qui sont en train de se former.
A substantial amount of
Il y a des
progress.
couleurs qui sont en train de se former. Et puis aussi, moi, quand je regarde même comme ça,
So this is a very intelligent man. He's actually a medical doctor, so he knows how to reflect on his own situation. But he reflects in various directions that are not relevant.
C'est quartier davantage.
C'est carquillé, oui.
Oui, je vois que je suis les orbites, hein, le mouvement des yeux dans mes
So he is actually speculating on the movement of his eyes in the orbital space.
Et je suis libre de bouger les yeux. Et automatiquement, je vois un peu plus. Ça laisse passer plus de lumière.
Encore sur le table là, il y a deux blocs.
Oui, je vois, il y a ici, il y a quelque chose ici.
Oui.
Voilà.
There is something there. Notice the perfect grasping always.
Bon. C'est-à-dire, j'avais oublié de... Est-ce que c'est toujours les mêmes choses ou
C'est comme hier.
C'est comme hier. Il y a le rouge ici.
Oui.
That's red.
Yeah.
See?
Donc finalement aussi, il faut trouver le bon angle.
Oui.
Oui. Parce que vous voyez,
So we
d'emblée,
repeat,
vous
we
savez, on n'est pas
repeat the red
trouvé. Mais il a fallu
discrimination in different
tourner
contexts.
assez bien là. Et paf, ça pique là.
Yeah,
Et
that's what he
j'ai
says.
vu, ça
Suddenly,
pèse
wang,
là,
it
l'œil là.
hits me in the eye.
Okay, okay.
Voilà.
So you see immediately taking the red to that.
Oui. Attendez, je vais vérifier avec ça. Bon. Je pense que j'ai ici le rouge.
That's the red.
Oui, quand même. Pourquoi? Parce que là-bas, il y a quelque chose qui a piqué.
So he
Et là,
again
je
says,
réponds plus courageusement parce qu'il y a quelque chose qui a piqué le rouge. Mais ici,
okay. Now we get stuck. Can you, up? No. All right, good, let's just do this one in presentation. So here are some questions. I mean, obviously, we know from our previous studies that TN has normal blind sight. When in normal experimentation, like in the literature, we use forced choice tasks, we use indirect forced choice tasks, and there is just no awareness of anything, no qualitative experience or anything, but the patient gives the correct answer. So that's standard literature, a normal blind sight, okay? So you didn't see that here. What you see here is actually some evidence for residual visual abilities, but they are not based on intact striate cortex. So that's already a bit of a problem for the theories in the literature that attach a critical importance to feedback, for example, feedback to V1. I mean, because that's not what's happening here. On the other hand, as you see very, very clearly, this residual vision is not like normal vision, because it takes somehow, depends on how you read this. You could say, well, it builds up till there is like sufficient signal strength or something like that. But none of that is actually a very clear explanation, right? On the other hand, and that's an important point, his vision, like coming to the decisive point, he says, this is red. That kind of visual experience has all the signatures of normal vision, namely, there is just no doubt. I mean, once the correct answer is there is no, it's given without any hesitation, and it's a phenomenal experience. I mean, we have no reason that this patient uses the word red in any other meaning than the standard common sense experience that it's underlying state recognition of red, right? Also, in that, related to that, there is normal phenomenal awareness. I mean, there is no indication that for the cases where the color is correctly coded, that there would be anything special about the experience. Of course, you could say that in this case, the experience of red is built up via different pathways than it is for the normal experience of red. But that may very well be the case, and we don't know. We also don't know very well for a number of object recognition phenomena in normal people. The visual system is not as transparent as common sense takes it to be, of course. So there, in whatever the routes through which that answer is achieved, there is normal phenomenal awareness. Okay? So it's I leave it open to discussion, to future discussions, to see to what extent this clashes with, is combinable with various versions of higher order theories, of phenomenal theories, of primacy of phenomenal experience theory, so that's pretty. I just wanted on this occasion to add this piece of information to the discussion, and it certainly fits, I think, with Joe's interest in somehow bridging non-conscious processes and conscious processes, as we see very clearly that phenomenal awareness can originate via different routes than it does in normal people, and that we cannot have this kind of fairly radical distinction between conscious and non-conscious processes, as if there were separate. The notion of separate routes for the two is not very needs to be challenged a little bit more than it is currently. Okay, so over to Joe for enlightening us on this puzzle. Thank you very much.
Okay, hi everybody. Wow, what a great meeting this has been. Thank you, organizers. Yeah, just I feel I'm seeing kind of cultural, mental, personal structures, you know, coming out from Joe, like this fungal hyphae, but reticulate it all, and then these other nodes growing their own hyphal nodes going out, and it's just we're seeing this incredible structure, and it's conscious. It's more than being fungal. Well, so my name's Tyler Volk, and until a few years ago, I was a professor at NYU in the biology department and in the environmental studies department. My expertise is in the global carbon cycle. And in studying the global carbon cycle, the reason I went into that discipline is because it allowed me to get into multiple fields at once, all the way physics, chemistry, oceanography, atmospheric, but also human futures, energy systems. And in that way, what comes in is human psychology, like why people make certain choices. And even though I haven't done research on it, in some of my books I've delved into issues. And Joe has really helped me along the years to think about some of these things and have conversations. But I'm mostly here to talk about the music at this point, because Joe and I are I guess the elders of the band, not only in age, but also in elderliness and going back far in history with the band. And this has been an unexpectedly important part of my life, being in the band with Joe and doing the music for so long. And it's allowed me some other important friendships, Daniela Schiller for a really long time, a great friendship. And then other people that are here, Colin and Amanda, and also Joe's wife, Nancy, and family. It's just been a whole fantastic social world in the last 20 years. Now, Joe and I met at one of his big picture conferences in the early 2000s called, I think it was Self in the Brain, and some of the biggies were there, Dennett, Damasio, some other people, and my girlfriend at the time, Amelia, who's now my wife, sitting over there, she and I went to this, and at the end of it, there was the reception, and we're standing around, and Amelia said, why don't you go talk with Joe? He seems like a really open guy. And so I did, and we had a talk about, I think it originated with our science, both writing popular science books, putting our science into books. I had a couple at that point myself. And we got together for dinner and then had some other conversations and discovered we played guitar, both had Stratocaster guitars, decided to get together to jam. And we started doing that regularly, sometimes my apartment, most of the time his apartment. Had some beers, and we would go into long jams. I remember doing Bob Dylan, Like a Rolling Stone. We would sort of sing it, and half an hour of Like a Rolling Stone, okay? A couple beers, just enjoying ourselves. And for me, that was enough. Now, I'd had bands in high school and in college at the University of Michigan. The band was called, no, I'm not going to go into the names of the bands. They're not really important. But that actually will come up maybe a little bit later if you're all patient enough with me. But we were just jamming, and then Joe said, let's the two of us play at one of our Joe's lab parties in December, and with the party, postdoc Daniela Schiller came up and said, "Oh, I played drums. I played drums in Israel," so we invited her to play some drums, and then Nina Curley was there, and Daniela, I think, knew Nina that plays bass, and okay, there's a potential for a band here. But now Joe and I in our jams, I mean, we were really a good dynamic duo. I was very happy with that. Very compatible in terms of, you gotta have some things that are in common and some things different enough to make it interesting. And I think we had that compatibility as a growing friendship. So now what I want to do here is take you through some slides and to preview what these slides are about. I want to ask, are there any fans of the amygdaloids here? Okay. Are there any, I'm going to test whether there are some fanatics here of the amygdaloids. So there's going to be a quiz. How well do you know the amygdaloids? Okay, some are easy, some are hard, some are hopefully fun. We'll see how this goes. So, number 1. Title of the first Amygdaloids album. Mind Countdown, Heavy Mental, Neuroscience Rock, or Rock, Paper, Stressors. Okay. B. Perfect. Okay, got it. Heavy Mental. Joe, I think that was coined by the New York Times reporter. Is that right, Joe? Wow. Coined it. Cool. And then you've heard that said several times in the talk. Now, a couple comments here. The CD, the fact that this was a CD. When we first started jamming, half hour like a Rolling Stone, there was no CD being talked about, really. And we played a gig at Union Hall in Brooklyn, and Joe started writing songs, and wow, holy smoke, and we're practicing these songs, and then Joe's starting to think of other gigs. And I remember, Joe, there was a moment where you and I were talking, and I just said, Joe, you're really serious about this. And you looked at me and you said, yeah. This was really unexpected and great fun. In fact, the producer who mixed it and did this first album, Jeff Peretz, he's going to be performing tonight on guitar. He's a professor here at the Clive Davis Institute, so you'll get to see him later tonight. Number 2. Okay, one of Joe, okay, by the way, so the number 2 is a little bit tougher, but for people that are maybe over 70 or really music aficionados. One of Joe's song's inspirations, Mist of a Memory, it's the title, came from lyrics to a song called Deep Purple, specifically the 1963 recording. So imagine Joe's teenager years glued to the transistor radio here. It was a duet sung by Nino Tempo and his sister. Chalmers? No? Anybody? Yeah, this is a tough one. Joe? April Stevens. Okay. Now, this seems like, why should I put this in? I knew it's going to be really difficult to get. But so I was thinking, well, one thing I found out, Joe, they were Niagara Falls. These people were Niagara Falls, and near where I grew up, near in North Tonawanda, near Buffalo, New York, and that will come up later. But Joe has got a big interest in memory and the way music can be very important at certain stages of one's life, and then mist of a memory. And Joe's been writing about how his own memories of certain songs have played such a role in his life. And so in a way, it's a mist of a memory lyric and a song. So there's kind of a meta thing here that I think is very interesting that Joe is exploring. Number 3, there's going to be 6 questions in all. The Amygdaloids were known for playing anytime, anywhere, even without pay. Even without pay. I mean, once we made some money, Joe, right? But it was, so anyway, which venue did the band never perform at? The Bitter End, Madison Square Garden, Otto's Shrunken Head, or the Kennedy Center? B? C. So, Joe? Wait, A. Yeah, we never performed at the Bitter End for some reason. But Madison Square Garden, man, I got to tell you, it was NYU, but it was so cool being up there like Eric Clapton, with my face up there on the giant screen of Madison Square Garden. 8,000. It was like so cool, Joe. Thank you for scoring that gig. And the Kennedy Center in Washington, D.C. at Otto's Shrunken Head. You felt like it was like Halloween every night walking into that place, and it still exists. So it was a place, so we played a lot of places. And the reason I brought up the pay thing is because even if you're playing for free, it's hard to get gigs in New York City. I mean, there's a lot of bands that want to be playing, but Joe, and occasionally some of the other band members, but mostly Joe, was finding these gigs, getting these gigs. So Joe was not only the band leader, but the manager. And when he was writing the songs, he was like sort of Pete Townsend of The Who. He would do a whole demo and would have the bass and the drums, and he was learning this software. He also, how did you have time to do all this stuff? So learning the software, doing these demos, and putting in nice guitar parts that I can then use and riff off of. Because my thing was really the improvisation sections of the songs. That's what I loved, coming up to improvisation. So, manager. Now, here's number 4. Which 2 of Joe's books were also exactly titles to his songs? The books are listed in chronological order. B and C? Joe? Wait, which are titles to your songs? Which are? Emotional, Brain, and Anxious. So A and C. Now this is really showing where the fanatics are. So there's not really many fanatics out there. But a couple comments here about it. So Joe, there's 2 more books that need to go into songs. That's one thing here. And also, one thing important, when he gets interviewed about his neuroscience and then how does music tie in, he's reluctant to go into depth about what music means. He often says, I keep it as a separate side of my life, the music thing. And this shows some of Joe's ability to expand himself and do some big thinking. But in terms of the deep history of ourselves, there is some interesting work by a musicologist at Yale called Tomlinson, wrote a book called, if anybody interested, 1,000,000 Years of Music. And he hypothesizes that making Acheulean hand axes and pounding hand axes was maybe the origin of something in the brain. At first, it seems like a little wild idea, but when you read the full book, it's really pretty good. So Joe, maybe we can work on that song sometime and start the beginning will be like a hand axe starting there. Okay, number 5. The amygdaloids. What else does the word amygdaloids refer to? Elements in an early cellular automata model of emotion. B, geological term for a type of igneous rock. C, chain of oblong islands in Malaysia. D, a kind of nuts eaten in ancient Rome. D, B. Joe? B. Yeah, it's actually a geological term for a type of igneous rock that has bubbles. The fluid goes into the bubbles, and then lava goes. Then they form minerals within those, and they look like almonds, kind of. So, which is the, as you know, is the word amygdala. But the kind of nuts eaten in ancient Rome. The reason I threw that in to try to fool people, well, it's not exactly the word amygdaloids, but it's very close to that. And I was reading, and I also found out that it's in Rome. It was almonds were thrown at newlyweds to kind of the way we might throw confetti or something like that. Okay, and here's the final one. Oh, and by the way, the elements in an early cellular automata model of emotion. If any of you want to use, who are into mathematical models of the brain, if you want to use that word and call a model of yours amygdaloids, I give you permission to do that, because I made that up. And number 6 and final. Finally, 4 photos of bands from the mid-1960s. This is an opinion question. Public opinion, a fan opinion question. Which group has the coolest overall look? A. These kids look all right. Yeah, so I don't have to think about my generation there. Okay, B. Now, if time keeps being on their side, they'll probably get some satisfaction sometime. C. North Tonawanda, Buffalo area, mid-60s, my band. I'm second from the left. Those shirts were like popular for Polish wedding bands at the time. But there was some kind of like influence, so that football, I don't know what was going on. But we were called the ID. And I named the band, and that explains some of my interest, I think, in neuroscience, even though it wasn't my field. And last but not least, D. And that's the band The Countdowns. Joe LeDoux, second from the left. Both of us are second from the left, Joe. Both of us are rhythm guitars. Okay, so A, B, C. Okay, let's. Okay, so Joe and I, hopefully will there be many jams in the future. It's really been a fantastic ride, Joe. You've been so important to my life and everybody I've known through you. I can't thank you enough, and may we continue for many decades ahead. Thank you so much.
Okay. Hi, I'm Jun-Sik Choi. I was Joe's postdoc. And it's Woody Allen who said 90% of life is showing up. But not only showing up, I'm given 10 minutes for this special event. Such an honor and privilege.
but I understand that Karim and I are the only thing standing between you and drink and great night of music. So I'm gonna try to change the plan a little bit. I prepared about 8 stories, how Joe LeDoux influenced my science and life, but I dropped them. And I'm gonna tell you a few stories that might show different sides of Joe. This is a title, but that's the only thing that looks academic, because I have to submit a official paper to university to justify canceling the classes this week. From freezing to active avoidance, facing fear with Joe LeDoux. So when I first studied, everyone was doing freezing, which was discovered by late Bob Blanchard and popularized by Joe LeDoux. But I kind of thought that, well, they freeze because there's nothing else to do in a situation like this. And Joe suggested doing active avoidance because he was thinking about active avoidance as a coping mechanism at the time. And it really worked out between him and me. So after that, he kind of let me build my 2A active avoidance and everything. So with the help of Chris Kane, I published a paper, Active Avoidance, looking at the role of different subdivisions of the amygdala. And also, Joe supported for the cover illustration of learning and memory, which I feel even more proud than the study itself, because I always considered myself as not an artsy person. So I thank you for that. And when I first became a faculty member, I didn't have an infrastructure for doing rat research. So here's this sort of an irony that we're in the business of scaring rats when everyone else is scared of rats. And everyone thinks that humans are expensive, but it's a lot of money to maintain rats in the lab. But I'm in the psychology department, so I have freshman undergraduate students who would voluntarily receive skin shocks for $10 gift certificates. So I started out with human fear conditioning, and of course, my sort of psychological barrier doing a human research was a little low because I read papers by Kevin LaBar and Liz Phelps. So I was able to publish some papers, human fear conditioning, and I realized that people were using Ekman face when people in Korea doing studies with Korean subjects. So I built Korea University Facial Expression Collection, QFAC 1 and 2, composed of about 10,000 photographs taken in my room with the setup I built. And then that got me some research money, so I was able to return rat research. And again, I was interested in some more dynamic stuff. So instead of using a skin shock, I built a robot predator. This is one of the renditions of robot predator we call RoboGator, in collaboration with Jin-Seok Kim, who, by the way, is the one recommended me to Joe as a postdoc with Ted Blair, who's not here. So I was going to show a video of RoboGator, how it scares a rat, to go back to, but the movie doesn't seem to be working, so I'm gonna skip. But that's a RoboGator. That's so since then I do everything with robots in my lab, different renditions of predatory robots. Oops. What did I do wrong? Just next slide. Okay. Another story is The Emotional Brain, which is the book that got me to work with Joe in the first place. And here's a story related to the book. So when I first got the job, on the first day, Joe bought me a lunch. So we'd sit together in a cafe. And there was kind of, you know, Joe, a quiet person. So to break the ice, I told him a story that people says that The Emotional Brain was a million seller, and Joe LeDoux made so much money that he drives Ferrari in New York City. I told him that story as an icebreaker, but he said no, very briefly. First, I didn't make that money, and whatever money I made from the book, I used them for my kids' private school. That's it. The ice didn't break, and the awkward silence afterwards. And I think that's the only lunch I had with him. So when I went back to Korea, I translated a book myself called 느끼는 뇌 in Korean. Whenever people ask me, and the book was pretty popular in Korea too, thanks to Joe's fame, people ask me if I made a lot of money out of that book. I told him no, and whatever money I made, I used for my kids. So, so here's, and also, a lot of people in Korea asked me if I could invite Joe to Korea. And Joe's answer was again, no. Because one time he visited Japan, and the trip was a little too harsh for him. So he decided not to travel to Far East. So, you know, Tony once mentioned that Joe was able to like Teflon coat it smoothly navigating, but sometimes Joe can be very, very strict and straightforward. But after many years, instead of inviting Joe, actually TV program crews actually came to New York City to film his lecture called Great Minds. So a lot of Koreans were able to actually attend his lecture through TV program. I think now it's online, so you can take a look at it. And show different sides of Joe, Joe as a band leader. So thanks to him, I was able to featured in another different TV program talking about amygdala and fear. And to celebrate this occasion, I asked one of my talented students, told him all these stories, and she came up with actually this nice design of Joe in a Ferrari with his guitar and leaving the amygdala behind. And although that guy, amygdala, has been used as a model of fear, you know, he knew that it was just an acting. But this time, amygdala is really scared because he's terrified because he never imagined a life without Joe LeDoux. But Joe's saying, no, I'm leaving the amygdala behind. So, and this is in Korean saying goodbye amygdala. And that's the end of my talk. And I brought these t-shirts for Joe. So I thank Joe for all those support and for wearing my t-shirt at least once. I know this is not your style. Okay, thank you.
All right, so this is our final talk of this conference. We're going to have a few videos after this that people sent, so please hang out a little bit longer if you can before the next break, which will be right outside. And just to make the announcement, Joe, exactly how did they get to the venue for the music? How did they get to the venue for the music? Okay, well, no, but how did everybody else get to the venue? Yep, down the hall. Yep. Okay, fine. Claudia can tell people. Okay. Awesome. All right, so it's such an honor. Kareem asked me to read some words for him, and it's such an honor for me because Kareem asked me, but also because I know what a special place he holds in Joe's heart. So I'm going to read some words from Kareem. This has been so much fun. What an incredible 2 days celebrating a man who has been etched into the very fabric of neuroscience. To me, Joe is not only an extraordinary neuroscientist, the greatest mentor, and a true friend. He's also the kindest, sweetest guy, as sweet as the powdered sugar on New Orleans beignets. I came to New York because Joe was a neuroscience god, and I thought, that's the place to be. I wanted to work with someone whose chill factor was as remarkable as his intellect. What an amazing time. I always say working with Joe was like riding a roller coaster. Thrilling, slightly terrifying, and full of screaming, although, come to think of it, maybe that was Joe's experience working with me. But it was always a wild ride. Joe's lab wasn't just a place to work. It was a place where lifelong friendships were forged, careers were launched, and history was made. Joe, thank you. Thank you for always having my back, even when you told me not to waste my time and money on the memory reconsolidation experiment because, quote unquote, it would never work. So much so, we even bet a bottle of tequila on it. So off I went and did it anyway. And when it actually worked, my jaw just dropped. I couldn't believe it. I ran into your office saying, holy bleep, bleep, bleep. Can't bleep believe this happened. I mean, the probability of this happening is like 0, right? So needless to say, Joe showed up at my door with a bottle of Patron. I had no idea at the time how that paper would affect my life, my career, and the number of events it would set into motion, starting with the Cold Spring Harbor Conference. Thank you for having my back and standing by our convictions, even when others relentlessly tried to discredit us. It was the craziest moment of my life, and I'm so glad I got to share it with you. Working with you has been nothing short of a privilege. You left an indelible mark on each of us, and you are truly the best guy to make history with. Thank you. Beautiful, beautiful. Thank you. So if everyone wants to stay, we're going to show a few more tribute videos, and then there'll be a reception outside, and then we can all go hear the amygdaloids.
All right, we have a few more videos to show. And for those who weren't here yesterday, we're going to replay some of the music video that Roseanne Cash sent us. Here we go. This is Itai Shapira.
Dear Joe, thank you for your groundbreaking work, for expressing your razor-sharp intellect with such simplicity and poetic beauty. Thank you for encouraging me to connect music and medicine and for insisting that I do it in a way that is genuine and meaningful, reminding me that science, like music, requires accuracy, imagination, patience, and consistency. I realize that you think that you are retiring, but I intend to keep you very busy in connecting two of your favorite worlds, music and science. Congratulations, Joe.
Do this one first.
Hi, Joe. We want to send our congratulations to your retirement.
We ourselves have retired many times, and it's something we can very strongly recommend.
And today, we really want to say how much we owe you in influence and inspiration. And I'd like to pick out just one thing that really made a huge difference to our thinking. And that is your wonderful demonstration of the showing that the response to threat in a physiological sense is not the same as the feeling of fear. And this is something that really encouraged us to think that we don't have to be entirely reductionist.
And the other thing that this result makes clear is that we should think of the brain as acting as a hierarchy, with the lower part being largely unconscious and the higher part being where consciousness resides. And we ourselves have talked about a three-level hierarchy, which of course is very similar to your more sophisticated version of a four-level hierarchy, which has an even lower level than we consider.
So what about the future? Of course, we would like you to read more books because we want to read them. And so we can only end by saying, welcome to the club.
And write more books.
That was Chris and Uta Frith. And this last video is from Trevor Robbins and Barry Everett. And then after that, I'm going to play on a loop some of the music videos that we played yesterday.
Hello there, Joe. On this momentous occasion, this is Trevor Robbins and Barry Everett from Cambridge. Remember us? Yeah. Well, we remember you coming on sabbatical leave, I think in 2009, to Downing College. And we enjoyed a lot of interesting discussions and social events, including you reenacting your famous group, the Amygdaloids, in Cambridge. Now, Barry and I were never members. We're not particularly musical enough for you. But I was going through our email correspondence the other day, and I remember a while ago you sent me a link to the lyrics of a new song called All in Your Minds. And I tried to explore this link, couldn't find it, it didn't work. So Barry and I thought we'd put some lyrics together for it and just kind of reimagine this song as all in our minds. So here we go. All in our minds, that's what they say. The synaptic self is here today.
All in our minds, deep histories too. The emotional brain, from ourselves to you.
All in our minds, but who are you? Find out by reading Joe LeDoux. There you go. Now, after a bit of light entertainment, we're going to proceed to a more scholarly interpretation of your works, Joe. And Barry's going to tell us a bit about when we first met up and interacted.
And as I remember it, Joe, our first, let's call it intellectual engagement that we can remember anyway, was over 25 years ago. And it was a bit of a spat in the pages of the journal Trends in Cognitive Science, when you and dear Karim Nader took exception to the heresy of our paper with Kilcross, suggesting that the basolateral amygdala and central amygdala had dissociable roles in explicit versus implicit fear, Pavlovian conditioned suppression versus punishment avoidance. But this we resolved in many subsequent discussions in bars, and the last time I think was in Waterloo in Belgium. And I still wrote later that year a hugely positive review of The Emotional Brain, which had been published during 1997. At least I think that's right. And I fished out my review, and I'm not going to read large swathes of it other than to say I was extremely complimentary. I also drew attention to the fact that you didn't make much mention of amygdala projections to the ventral striatum, but there we are. But what I did say towards the end of my review was, who should read this book? Frankly, I can't think of anyone who should not. And I still think that's true today. A remarkable book, very scholarly, exploding myths and presenting new ideas. Tour de force, Joe.
And Joe, beyond the amygdala and the ventral striatum, you've delved into really important, deep things like the evolution of consciousness. And I remember doing a synoptic review of Deep Histories of Ourselves, which is all about the evolution of consciousness. And I'll just quote that review, which I still hold to. Joseph LeDoux has provided a remarkable, personalized synthesis of zoology, neuroscience, psychology, and philosophy. His main theme is the emergence of consciousness through the evolution of nervous systems and the behaviors they control. Not the tree of life, but the tree of consciousness and where it may lead us. An amazing, mind-expanding read, Joe. Thank you so much.
And I think that's our message to you, is it not, Joe? Thank you for your huge contribution and the wonderful interactions we've had over the years. And I hope it's not too long before we see you again, either in the US or back in Cambridge, where the good people live. Bye.
Goodbye.
Congratulations.
Congratulations, Joe.
Sorry. So Joe asked me to end this session on one of the music videos from yesterday, and I'll see if I can find that. Yep, here it is. And then we're going to proceed for the reception, and then after that to the music. Do you want me to stop? Just good?
Hey, Joe. I just want to thank you for the lives you've changed, for your inspiration, for your knowledge and generosity, and your research and your love. And I know you're retiring, but I doubt you'll retire. I think you'll probably still be giving those things out in the next phase of this journey. So John has agreed to join me and play this song for you. I think you'll recognize it. Mind over matter, it's something I'm trying to do. It's just a little physics that keeps me apart from you. Mind over matter, it's something I'm trying to do. Break down space and time, be together with you.
trying to will you here, to find you wherever you are. Locate your body, your soul, pull you in from afar. Are you still in by time, or have you slipped away? Have you gone forwards or back, living in another day? Mind over matter, it's something I'm trying to do. I guess it's only physics that keeps me apart from you. Mind over matter, it's something I'm trying to do. Break down space and time, be together with you, be together with you, be together with you.
So that's not the end of our program. The amygdaloids is next, and if you want to know how to get there, ask Claudia. Thank you all for coming.